Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner.
Lovingly known as SBIA program, which operates within the Division of Drug Information, which in turn resides in FDA's Center for Drug Evaluation and Research's Office of Communications. It is my great pleasure, along with the SBIA team and our colleagues, Elias Malice, Director for the Division of Industry and Consumer Education from the Center of Devices and Radiological Health, and Dr. Larissa Laptova, Associate Director for the Division of Clinical Evaluation General Medicine from the Center for Biologics Evaluation and Research, to be with you here today. And I hope you guys can feel the excitement, I certainly do, that this is in fact our first hybrid REdI post-COVID. So thank you very much for making the effort. Yep, I think we deserve a thing. So it's really heartwarming to see so many of you here with us today. At this juncture, I need to say we are extremely pleased to announce that this two-day conference offers a total, over the course of 2 days, of 14.25 contact hours to physicians, pharmacists, and nurses. The CEs are available to all real-time attendees who have registered and who use the login links to attend. So note the emphasis on registration and real-time. And what this translates to is that no healthcare professional CEs can be given to those who attend via YouTube or for post-event viewing. All faculty are expected to use generic names. If trade names are used, those of several companies should be used rather than only that of a single supporting company. CE faculty are required to disclose to the attendees when products or procedures being discussed are off-label, unlabeled, not FDA approved, and any limitations on the information that is presented. We do have a spectacular lineup of speakers today and tomorrow, but for today, the planning committee, speakers, and CE consultation and accreditation team have nothing to disclose. Participants must attest to their attendance. Am I having an echo, or I'm just too close? Participants must attest to their attendance and complete the final activity evaluation via the CE portal, and that's ceportal.fda.gov. For multi-day activities such as this, participants must attest to their attendance and complete a faculty evaluation for each day. Final activity evaluations must be in 2 weeks after the activity, and in this case, the deadline will be June the 13th, that's 2 weeks from tomorrow. And we apologize in advance, but we are definitely unable to make exceptions to that deadline. Pharmacists should note that failure to provide the correct NABP and date of birth information in the required format may result in the loss of credit for this activity. Your NABP profile number should be the 6 to 7-digit profile number assigned by the CPE monitor, and your birth date should be month, date format. So example, that will be 0529. Do not provide your pharmacy license number. Please click on the My Account tab and then navigate to Edit Contact Information to verify that the information is correct. Physicians and nurses may then view or print their statement of credit. Pharmacists should log into the CPE monitor 6 weeks, please allow 6 weeks before inquiring, after the last session of the activity to obtain the CE credit. And to simplify things, SBIA will send one email to all the attendees of each day the following morning. The email will have information on both the healthcare professional CE claim code and the survey link. So I strongly encourage everyone to complete the evaluation upon receiving that email, as again, as much as we would like to, we won't be able to assist you if you miss the deadline of June 13, 2024. So, my slide is moving. Here we go. As of this morning, we have over 12,300 registrants. Of course, they're not all here in the room, otherwise we'd be good. From over 125 countries, actually, my calculation says. And over 49% of these registrants have self-identified as small businesses. And of those, over 26% have reported having 1 to 9 employees. So as you can imagine, these statistics continue to be extremely reassuring to us at SBIA, as they confirm that we are indeed reaching the small business sector of our audience. We know our audience is pharmaceutical industry at large, but it confirms we are reaching the small business sector. And they also validate our mission and the very existence of the SBIA program. And what you see here is a snapshot of a geomap for today's event. And this map is always quite exciting for us as well, because it provides a powerful visual as to the impact and extent of our outreach for those 125 countries. Okay, let's run through some logistics very quickly. These points apply to everyone. Here is the link for a one-stop shop where you'll get answers to your technical questions, downloading the files, accessing the survey link to obtain the certificate of attendance, and so much more. For our online attendees, this is the same page that you used this morning to access the event. So please bookmark that page, as you'll need it over these 2 days. If you have any technical questions about this conference, please check there first, that website, and you will most likely find the solution. On our one-stop page, there are session evaluation links and also a final evaluation survey link, which will be activated this afternoon and which will be included in a thank you email sent tomorrow morning. Upon completion of the final evaluation, an attendee can then download a certificate of attendance, which may be used in support of RAPS, SOCRA, ACRP, and SQA credits. The trick is that the certificate must be downloaded immediately after completing the survey, otherwise that opportunity is lost. And if you don't even need a certificate, please take a few minutes to complete anyhow, because the feedback, your feedback will help guide the design of our future events. Again, I'm stressing both the evaluation and the certificate are only available for 2 weeks, which means the deadline date is also June 13th. As a reminder, all the sessions are being recorded. It will take us a couple weeks to process the recordings, but you will soon be able to access them from SBIA's homepage. And hopefully everyone has already bookmarked that page, which is fda.gov/CDER SBIA. In the interim, the YouTube link from both days will be posted on the One Stop webpage at the end of the day, and then will also be posted on SBIA's webpage. For online attendees, you can submit your questions using the on-screen QR code, which is in the lower right corner of your screen. It's there all the time. And here are some logistics for the in-person attendees on site. Again, the Wi-Fi information. Please line up at the standing microphones for your questions for any speaker. Please silence your phone. Use the internet in-house sparingly, as it may negatively affect our online delivery. You're on your own for lunch. We don't provide lunch or even dessert. You're on your own for lunch, and there are 3 establishments in-house. You can pre-order via the QR codes, and the information with the QR codes is outside at the desk. So please do that for your convenience, so you don't spend most of your lunch hour standing in queue. And parking, for those of you who have parked in the hotel's garage, please also pick up a discount sticker at the registration desk, and that will allow you to pay $10 for the day. Also, please join us for the networking event, which begins after we adjourn for the day, which should be around 4:45 p.m. This is what the pre-order QR codes look like for the in-house establishments. And at this juncture, I would like to acknowledge those individuals instrumental in making this event possible. Many are listed here, but so many more deserve credit, really. First and foremost, we'd like to thank our center directors for their endorsement of this combined effort of expertise representing our 3 FDA centers. We have Dr. Patricia Cavazzoni from CDER, Dr. Chef Shuren from CDRH, Dr. Peter Marks from CBER. Then I'd like to go through and thank my own OCOM management for their support, which is, without the program wouldn't exist without their support. We have J.D. Wiley, who is the OCOM's director, Morgan Jarek, OCOM deputy director, Mary Kremsler, director for Division of Drug Information, Catherine True, deputy director for Division of Drug Information. A special thanks to Elias Malice, Joe Tartle, and Tanya Wilborn from CDRH, and Larissa Laptova, Stacy Rivett, and Lonnie Warren Henderson from CBER for coordinating their individual tracks. Our heartfelt appreciation to the faculty for their commitment and dedication in sharing their expertise with us all and being here in person over the next 2 days. Sincere thanks also to OCOM, our fellow DDIers, for their support in myriad ways. But most of all, our acknowledgement and thanks to you, the audience, who took time out to attend this event regardless of the inconvenience of various time zones. We do not take this lightly, and without you and your commitment and your interest, this program would not be the success that it has been. So at this time, I'd like to introduce our keynote speaker. Unfortunately, Dr. Califf could not be with us in person today because of prior commitments, but we do have his recording. Dr. Robert Califf is the Commissioner of Food and Drugs and was sworn in on February 17th, 2022. Previously, Dr. Califf served as Commissioner of Food and Drugs from February 2016 to January 2017. As the top official of the FDA, Dr. Califf excuse me, It's committed to strengthening programs and policies that enable the agency to carry out its mission to protect and promote the public health. Dr. Caleb served as the FDA's Deputy Commissioner for Medical Products and Tobacco from February 2015 until his first appointment as Commissioner in February 2016. Please let us welcome Dr. Robert Caleb.
I'm delighted to once again help kick off this important conference. Its focus, innovation in medical product development, is at the core of the FDA's mission. One of our primary goals to support innovation and scientific advances that can be translated into viable, safe, and effective medical products that make a meaningful difference for patients and their families. At the outset, I should point out that the FDA's support of innovation is not limited to medical products, but also includes the many other areas in which we have oversight, including food safety, nutrition, cosmetics, dietary supplements, and tobacco regulation, to name several of our more prominent responsibilities. For the next few days, however, you'll be focused on medical products and some of the many ways we're working across the entire agency to support you and your innovation efforts. The companies represented at this conference are a key focus of the support and guidance we provide to businesses, especially smaller businesses involved in the development of new medical products in the areas we regulate. As you know, we have 3 different centers whose work encompasses these issues: our Center for Drug Evaluation and Research, our Center for Devices and Radiologic Health, and our Center for Biologics Evaluation and Research, as well as many other offices across the agency who play a role. You'll have the chance to hear and exchange information from representatives of all 3 centers during this conference. In fact, during the next session, you'll have the opportunity to participate in a discussion with all 3 directors of our medical product centers. That conversation will reinforce the fact that while each center has specific responsibilities, their work also overlaps. This collaborative, cross-cutting approach is central to the FDA's success, and is based in our agency's underlying commitment to biomedical science, clinical research, and inquiry with a focus on research, data collection, and analysis that forms the evidence to support decision-making. Moreover, as developers like you embrace the latest scientific and technological advances, our capabilities to support your work and engage with you as appropriate only grows. This morning, I want to give you just a flavor of some of the initiatives we've undertaken to support the work you do and advance medical product innovation. Throughout the meeting, you'll have the opportunity to discuss in greater detail many of these and other programs and supports available to you. Let's start with CDER and with a topic near and dear to my heart, clinical trials and some of the work CDER has been doing in this area. Over the years, CDER has championed clinical trial innovation with programs such as model-informed drug development, complex innovative trial design, drug development tools qualifications, and other initiatives. Each of these efforts has enhanced the design and conduct of clinical trials intended to generate evidence of safety and effectiveness of therapies to meet the growing demands of drug development. I'm excited to announce that last month CDER launched its new Center for Clinical Trial Innovation, or C3TI. Using targeted projects, the C3TI demonstration program will explore scalable improvements in the adoption of several clinical trial innovations, expand opportunities for sponsors of innovative clinical trials to engage with CDER staff, and enhance the existing body of knowledge and precedents for clinical trial innovations to improve the efficiency and effectiveness of drug development. There are several sessions during the meeting focusing on clinical trials, so you'll be able to explore this topic in greater depth. CDER is not alone in its work in this area. For instance, it is collaborating on a new pilot program initiated by CBER to accelerate the development of new treatments for rare diseases, particularly the rare genetic disorders that affect children and result in severe disability or death in the first decade of life. In addition to working together on this effort, called Support for Clinical Trials Advancing Rare Disease Therapeutics, or START, Both centers also continue to participate together in the ongoing Rare Disease Endpoint Advancement Pilot Program. One of the topics that will be included in C3TI is artificial intelligence and machine learning. But this is just one of the ways we're incorporating AI, which is one of the most exciting and promising areas of science, into our work across the agency. I know many of you are also beginning to find application for this technology, and we look forward to supporting those efforts. The FDA in each of our centers has been working for years to anticipate and prepare for the challenges of AI and to harness its potential to enable major advances in the development of more effective and less risky medical products. Consider, for example, since 1995, the FDA has received over 300 submissions for drugs and biological products with AI components, and more than 800 submissions for AI-enabled devices. These submissions have included drug discovery and repurposing, enhancing clinical trial design elements, dose optimization, endpoint and biomarker assessment, and post-market surveillance. They also cover a growing diversity of medical devices that leverage AI to improve clinical workflows and patient experiences or outcomes, in addition to sophisticated prediction algorithms. We've issued several helpful papers in our work and role in this area. The first of these, Artificial Intelligence in Medical Products, how CBER, CDER, CDRH, and OCP are working together, outlines and reaffirms our commitment to promoting the responsible and ethical development, deployment, use, and maintenance of safe and effective medical products that incorporate or are developed with AI, as well as prioritizing collaboration, consistency, and mutual learning. Another discussion paper, Using Artificial Intelligence and Machine Learning in the Development of Drug and Biological Products, seeks to foster dialogue with interested parties in the medical products development community on using AI, ML in drug and biological product development and the development of medical devices to use with these treatments. As AI landscape continues to expand and evolve, we'll continue to develop new ways to use this technology. I know there are several sessions of this conference that focus on this exciting area. I also want to mention the Drug Development Tool Qualification Program developed by CDER and CBER, which provides a route to qualify novel drug development tools, or DDTs, for use in drug development programs. Examples of innovative DDTs accepted into the biomarker qualification program include AI and ML-based readings of liver histopathology slides in NASH and MASH, also known as non-alcoholic steatohepatitis and metabolic dysfunction-associated steatohepatitis, and an AI ML-based multi-component biomarker to assess risk of cardiovascular outcomes. Let's now move to CBER, and although I've already mentioned CBER's work several times, an indication of just how closely our centers can work together, let me offer a few other exciting CBER-focused developments in the center's continuing development and approval of innovative therapeutic products. In 2023, CBER approved 23 biologics device applications, 20 biologics license applications, and 17 biologics license application supplements. There were many highlights, including 2 new gene therapy treatments. One is the first product using genome editing technology for the treatment of severe sickle cell disease, and a second indicated for the treatment of children with different forms of a rare disorder called metachromatic leukodystrophy, or MLD, which affects the brain and the peripheral nervous system. CBER is also considering ways to leverage data related to platform technologies for cell and gene therapies to further promote development of treatments for patients with rare diseases. This includes and complements the ongoing collaboration with the Bespoke Gene Therapy Consortium. And CBER also continues to support global harmonization efforts related to the evaluation and regulation of cell and gene therapy products to help facilitate more efficient clinical development for these and other product categories. The international collaboration of regulators and the clinical community of medicine is extremely important and has spurred many important developments, including another new pilot program, the Collaboration on Gene Therapies, or COGEN-T, global pilot. In 2023, CBER published numerous draft and final guidance documents for developers on topics including blood and blood components, tissues, vaccines, and cellular and gene therapy. For example, to further promote incorporation of gene editing technology into new treatments available to patients, CBER published a guidance for industry, human gene therapy products incorporating human gene editing earlier this year. CBER has also continued to strengthen its patient engagement efforts to help facilitate the development of medical products that make the biggest difference in patients' lives. In 2023, CBER participated in 35 patient-focused drug development meetings and patient listening sessions, in addition to 4 CBER-led or co-led public meetings with patient groups. Now let's turn to CDRH, our center that is responsible for assuring that medical devices on the U.S. market are high quality, safe, and effective, and that works to facilitate timely patient access to these technologies while advancing medical device innovation. Last year, CDRH gave marketing authorization to 124 novel devices, excluding EUAs, the highest on record in the center's history. It's worth noting that one of the most important factors in CDRH's continuing evolution has been the importance of medical device user fees, which provide critical resources to support program improvements, including new hiring targets, greater engagement with developers of innovative technologies based on lessons learned from the pandemic, broadened international harmonization efforts, and expanded opportunities to ensure patient perspectives are an integral part of medical device development and evaluation. One essential component of MDUFA is the Total Product Lifecycle Advisory Program, or TAP, pilot, a voluntary program intended to help innovators avoid pitfalls in early product development and better ensure a clear, predictable path to market from development to bedside. TAP will provide industry with earlier and more frequent interactions with CDRH, more strategic input from stakeholders, and proactive strategic advice from CDRH to spur more rapid development of high quality, safe, effective, and innovative medical devices, first in the world, that are critical to public health. You'll be hearing in much greater detail TAP from CDRH Director Jeff Shuren a little later in the program. Before I close, I want to mention a few other important CDRH-related developments. The first is last month's final rule on LDTs, or laboratory developed tests. As this audience understands, LDTs are today used more widely than ever before in everything from newborn screenings to helping predict a person's risk of cancer or aiding in diagnosing heart disease and Alzheimer's. But even as 70% of today's medical decisions depend on laboratory test results, some of these tests perform poorly or don't work at all. As a result, patients may initiate unnecessary treatment or delay or forego proper treatment altogether, which could result in harm. I'm particularly concerned right now about the, quote, diagnostic odyssey, unquote, experienced by patients with rare disease, as they often go to multiple medical centers over multiple years before the diagnosis is made. The FDA's oversight and enforcement of the requirements for LDTs dates to a time when tests were generally simple, often made to address local individual needs, and mostly manufactured in small volumes and often in a single lab. In contrast, today's LDTs can be far more complex and offered nationally. Hence our decision to craft a new rule to ensure the safety and effectiveness of LDTs today. After seeking extensive public comments, we announced a final rule in April that will better protect the public health by amending the FDA's regulations to make explicit that in vitro diagnostic products, or IVDs, are devices in the FD&C Act, including when the manufacturer of the IVD is a laboratory. The second recent CDRH initiative I want to call to your attention is the Home as a Healthcare Hub project, which is designed to help reimagine the home environment as an integral part of the healthcare system with the goal of advancing health equity for all people in the U.S. In today's world, home care is an essential aspect of healthcare, but very few care options consider the structural and critical elements of the home that will be required to absorb this change in the location of care delivery. CDRH is collaborating with patient groups, clinicians, and provider organizations, the medical device industry, and housing design experts to build the home as a healthcare hub. Once this concept is established, it will provide a systematic approach to enable people to get the care they need when they need it. Stay tuned as this exciting initiative takes shape. As I hope this brief outline of recent developments has made clear, support for innovation in medical products is at the heart of what the FDA does. And I hope you'll take the time during the conference to connect with our outstanding FDA staff and leadership on these and other initiatives. Working together, we can truly achieve the best possible outcomes for patients. I want to add one final note in closing, which goes to a related responsibility of the FDA: how we communicate trustworthy scientific information to the industry and the public. As we know, misinformation is being used today to undermine public confidence in science generally, and biomedical science in particular, and to subvert the credibility of our agency's work. As we focus on your efforts to innovate, I want to underscore just how important it is that we and you get it right. We're committed to doing our best to produce reliable scientific work, to communicate facts and evidence, and to use the most scrupulous and sophisticated scientific methods and tools to achieve this result. This communication challenge includes a daunting responsibility to communicate uncertainty while also maintaining the public trust. In so doing, we will increase the likelihood that the public and developers will trust and rely upon the FDA's decisions in support of innovation in medical products. Thanks again for your commitment to this process and for your participation here.
I think that was a very nice overview of the three centers. What do you guys think? Did you learn something? You got to learn something, otherwise we're missing our mark here. All right. It's now my pleasure to formally introduce my colleagues who will lead the plenary session, and they're waiting here in the left. Elias Malice. Elias, you can come up first. Elias Malice has co-hosted this REdI conference event with us since 2012, and he will be the moderator for the session. Mr. Malice has held many varied leadership positions throughout the years and currently serves as the Director of the Division of Industry and Consumer Education in the Office of Communication and Education in the Center for Devices and Radiological Health, a position he has held since 2011. For those of you who are not used to FDA, we use a lot of acronyms. So, if you look on your agenda at the top, there's a link that takes you to the explanation of those acronyms. Then we have Dr. Larissa Lapteva, who will facilitate the online questions for the plenary. Dr. Lapteva is the Associate Director in the Division of Clinical Evaluation, Office of Therapeutic Products, Center for Biologic Evaluation and Research. Since joining FDA in 2006, Dr. Lapteva has held review and supervisory positions in both CDER and CBER, and has provided scientific and regulatory advice for clinical development programs with investigational products across different therapeutic areas. Let us welcome them, and I hand it over to you, Elias.
Thank you, Brenda. And now, before we go any further, I want to express my personal thanks to my two very distinguished colleagues who help us to put on this incredible conference just for you. First, I want to really thank Brenda Stoddard. Brenda, you were the force that brought us all together way back in 2012 with a new idea to partner together with a joint conference. And look where we are now, all these years later, 13 years. And second, I want to thank Larissa. When Brenda reached out a few years ago to you and your team to expand the program to add a third track that truly made us whole, you and your team's response was very simple. Absolutely, let's do it. What do you need from us? So Larissa and Lonnie and Stacy, I really want to thank you for just making this a great conference. This is our 4th year in a row with CDER. And so I want to give my colleagues a nice round of applause for them. So for everyone in our audience online, welcome, and in person, I have a question for each of you. What were you doing 5 years ago? Do you recall where you were? Now, if we're being really literal, 5 years ago to the day was May 29th, 2019. And some of you might have raised your hand to say, I was in Boston. And if you were, I thank you because that means you were at REdI 2019 with us in person. That was the last time we were together in this hybrid format, in person and online. So if you were with us in Boston in 2019, and now 5 years later, you're with us here, I thank you for your continued participation and engagement as we put on this conference just for you. Now, for many of us, 5 years goes by very fast in general. And if we think about it, so much has happened in our lives during these last 5 years. We've endured a global pandemic, and many of us in the regulatory business learned to say the acronym EUA a lot, especially in our world here. In the workplace, we learned new communication platforms like Zoom and MS Teams. Definitely, those have become part of our parlance. And at home, many of us took to heart the spirit of online shopping and delivery for just about anything that you can imagine. The one constant through this all is change. The world continues to change and move forward. And when it comes to technology, this is no more apparent than the explosion of artificial intelligence that has made ChatGPT a household name. Now, one of the more interesting statistics about the FDA is that we regulate 20 cents of every gross domestic product or GDP dollar in the United States. Now, that's 1/5 of the US economy. An incredible stat that really illustrates the vast impact of the FDA. And not surprisingly, the FDA is at the epicenter of where change meets growth, of where medical innovation occurs, and with the ultimate aspirational goal of bringing medical products of all kinds to patients in need. So it's only fitting that this theme forms the topic of this year's conference and this year's plenary session, that is that of innovation, and specifically innovation in medical product development. And to bring this forward-looking topic to you today, we have 3 very special guests for you, which I'd like to invite to join me on stage for today. Dr. Patrizia Cavazzoni, Director of the Center for Drug Evaluation and Research, or CDER. CDER regulates a whole range of drug products, including those that are over-the-counter, prescription, or generic, as well as biological therapeutics. And a fun fact, now that we're heading into the summer season, especially with me with my head, this includes greens. Dr. Peter Marks, Director of the Center for Biologics Evaluation and Research, or CDER. CDER regulates various biological products, including blood, cellular, and gene therapies, and of course, vaccines. Products that are derived from living sources. And Dr. Jeff Shuren, Director of the Center for Devices and Radiological Health, CDRH. CDRH regulates a wide range of medical devices and, interestingly, electronic products that are not medical devices, but which emit radiation, such as TVs and microwaves. That's the RH part of our acronym. Please join me in welcoming our esteemed faculty for the plenary session titled Innovation in Medical Product Development. So, for those of you familiar with our plenary session, You might recall that we historically start with a presentation by our faculty and then follow this up with a question and answer segment with you. Well, in the spirit of innovation, we're changing things up even for our plenary session. We're going to make this very interactive with you. We'd like to really engage with you for this session. So we will not have an initial presentation. We will immediately begin a roundtable discussion with our esteemed faculty here, covering a variety of topics on the theme of innovation. Now, this is where you and our audience, both online and in person, come in. So first, let me speak to those of you who are online. Welcome again for joining us. Please send us your questions online. You can do this by using the QR code that you should be able to find on your screen. Click the code, which should take you to our Q&A website. From there, you should be able to select the session Day 1 Plenary, see the names of our esteemed faculty here, and then please type in your question. Larissa out in the back will be receiving your questions and sharing them during our program. Now, for those of you who are joining us in person, let me speak to you for a second. As you can see, we have some microphones that are set up throughout this room. So you're not standing for our full hour segment. I'll give you a heads up when we're ready to start receiving some questions. And when we do so, please, I welcome you to come in and join us and share your questions. Now, speaking of the question, I do have just a couple of asks. Number one, if you'd like to direct your question to one of our center directors, please just let us know if it's specific to devices or drugs or biologics. You are invited to ask a broad question and invite all of our faculty if it's a broader question there. And then the second note, I appreciate that this is a very priceless opportunity to engage with our center leadership, FDA leadership. I do ask that you focus your questions on the theme of innovation, on the topics that we're covering today, and that they may have already discussed. So I appreciate that. So with that, let's get started with our session. Thank you again for joining us, and we'll get going. Our name tags. Okay, we'll put it here. Well, thank you for joining us. So as we did a little bit of an intro with our center directors, I'd like to kind of continue the introductions a little bit. So for Peter, I'm going to start with you for this first question. We'd like to get to know you a little bit more. So prior to you coming to the FDA and in your current role as the center director for CDER, can you share what role you might have had in your prior life that involved innovation?
Yeah, so prior life, which prior life? I think when I worked in industry, I actually did work on drug development in the oncology area, and for a time we were working on a targeted therapy that was basically targeted for a very large indication. And in generally, innovation for me has come by necessity. Necessity is mother. That saying that necessity is the mother of invention. We had a very large indication that we were trying to use this targeted therapy in, and the trials were going to take a very long time, and it was a very competitive field. And so the question was, how could you speed things up, get to market, and not be last to market, but instead be first to market? And so the innovative approach was rather than using a very, very large indication, to go for a very, very small indication where there was a tremendous unmet medical need. There was not any other therapy in that particular area that was effective. And it took what was going to be a 5-year development program and brought it down to about 1.5 years to 2-year development program. That was done out of kind of desperation because otherwise I think we would have just not gotten there. It turned out that the large indication failed when someone else did the trial. That innovative approach, which was successful, was a reasonable way to go. But I think that type of use of innovation out of necessity that has also paid off during the COVID pandemic and at other times as well.
Excellent, thank you. So you've been in the shoes of our audience with being on the other side of the FDA.
Correct.
Here you are now. Jeff, I want to ask you the same question. Any thoughts on your prior life prior to coming to FDA?
Well, before coming to FDA, I was in academia. So I was a user of the innovations, but I also saw the impact when needed innovations were not available, what that did for patients. And then so coming to then the FDA, I came about to CDRH 15 years ago. And at that time, developers of innovative technologies did not bring those innovations to the US first or second or third, sometimes not at all. They went to other countries. And that was in part due to us at the FDA. And credit to my team, we did a lot of soul searching and analyses on why that was the case, and we made a commitment to change. And now, fast forward 15 years later, more than 50% of developers of innovative technologies for US market do now bring it here first or in parallel with other countries. And we did it 3 big things I'll highlight. 1, a culture change for us, built on the pillars of safety and innovation. We saw it as part of our mandate to advance responsible innovation, innovation that's safe and effective, it's got value proposition for patients. And a culture of engagement, customer service, and collaboration. We're gonna work with people, and that's industry, but other customer groups, patients, providers. We changed our programs, streamlined them, made them more predictable, and introduced new ones like breakthrough devices. And we advanced the science for supporting the design, development, and evaluation of technology, regulatory science, because a lot of innovations, the science isn't there to understand what they do. We really invested collaborating with others for building those kind of tools like bench testing, modeling, biomarkers, clinical outcome assessments, and made that available to innovators. And part in collaboration, and the last thing I'll mention, we helped co-found public-private partnership, now a 501c3 organization, the Medical Device Innovation Consortium, which is the only such entity here in the US devoted to advancing regulatory science for medtech. You don't know that group, they have a lot of great tools. Check them out, mdic.org. I do get part of their proceeds, so please.
Thank you, Jeff. Patricia?
Very happy to chime in. What really resonated from what Jeff said is the concept of responsible innovation. I really like that. I came from FDA after spending time in academic medicine. I was also an investigator in clinical trials and then spent many years in industry. And I think it's really important that we make a differentiation between what drug developers do and what we do at FDA. We certainly have a very important role in regulating and to the extent that we can, obviously streamlining and helping drugs getting through development, but we're not drug developers. We're not collaborators of drug developers. We're fundamentally regulators. Now, we do have some very important tools that can facilitate drug development, that can sometimes expedite drug development, and if not, and tools that could actually potentially also slow it down, right? So it's really very important that we regulate drug development in an agile fashion, looking for ways to to cut unnecessary sort of bureaucracy or hurdles and et cetera. But I think it's very important that we make that differentiation. And my many years in industry where I have seen certainly heroic efforts and very innovative efforts, but also decisions, development decisions that were largely driven by financial considerations. And we see a lot of Me Too's in drug development as well, which when we have many areas where there's a lot of unmet medical needs. I mean, rare diseases is one of them, but still lots of cancers that don't have treatments and so on. So this concept of responsible innovation from a regulatory standpoint, I think it's very important and fundamental. And I see that getting a little blurred these days.
All right, thank you. Thank you for the introductions and hearing about your background and your journeys coming to this point now. Patricia, I'll stick with you with this next question. This is under the auspices of product approval considerations. What types of drugs are eligible to be considered for accelerated approval? And what distinguishes accelerated from traditional approvals?
Well, this is a big question, and I'm hoping that Dr. Marks will chime in at some point. So we can use accelerated approval in situations where we're dealing with a serious life-threatening disease that also does not have alternative treatments. Accelerated approval is possible if we can identify a surrogate endpoint, being usually a biomarker of the disease, or an intermediate clinical endpoint. And obviously, the most notable example is oncology, where they use a progression-free survival as an intermediate clinical endpoint to support accelerated approval. When we look at these endpoints in accelerated approval, we still apply the substantial evidence standard, as we do with the more proven clinical endpoints that we use for full approval. It is important to understand, however, that substantial evidence standard is applied in accelerated approval, is applied on endpoints that reasonably predict clinical benefit. And that's very different from having been confirmed to measure clinical benefit, and which is why we need to have confirmatory trials after we approve a drug with using accelerated approval. These confirmatory trials are very important. It's important that they be adequate, well-controlled clinical trials, and it's important that they be conducted in a timely fashion. And unfortunately, we still see some sponsors who, I'm sorry, I can't say it any other way, that really drag their feet, that take years and years and years to do a clinical trial that may take much less time. The other thing that is important to understand is that when we use accelerated approval, we accept a level of sort of residual risk that is much greater, or greater, maybe much greater, but greater than what we accept when we use full approval. And that explains why sometimes the confirmatory trials don't confirm clinical benefit. And we have seen this in many areas. We have seen it in oncology. We have seen it in rare diseases. We have seen it in women's health, a very notable example last year, and so on. So hopefully I was able to sort of unpack something that is rather complicated and causes a lot of discussions even at FDA. And I'm going to turn it over to Dr. Marks to see if he has anything to add.
Yeah, no, I think the only thing I would add is that the last year we did get some additional authorities to help those essentially move along those who might be dragging their feet by being able to insist that people have some timeliness in how they go about getting confirmatory information for accelerated approval. It is this concept of residual uncertainty that needs to be resolved, and some people like having uncertainty. It's very good for the bottom line sometimes, and unfortunately, that's not good for public health. The faster we can resolve residual uncertainty, the better for public health. So I think the ability to move that along is probably an important piece of us being able to continue using the accelerated approval pathway to be able to hopefully get products to people in need faster. So I think hopefully people will take that seriously. We're taking it seriously. Also, I think the other piece, one last thing, is that the ability to have a more structured process for withdrawing products when they don't meet their their confirmatory endpoints. I think it's also helpful. It used to be a lot of appeals that would take a lot of time, and I think we now have a much more structured and hopefully faster process in making that happen.
And we have already used these two authorities once each, one that Dr. Marks knows very well. And we also recently issued a complete response to a company, to a sponsor who, when we determined that the confirmatory trial was not sufficiently underway. So.
Thank you. Thank you both for that. Jeff, we'll go to this next question with you. In April, so just about a month ago, you published, or the center published, a report on medical device innovation. Can you tell us a little bit more about this report and its intent and what you were looking to accomplish?
Yeah, certainly. So as I mentioned, we've been on a bit of a journey for the past 15 years. And, as Dr. Califf mentioned, last year we had that banner year of, 124 novel device authorizations. But 15 years ago, it was 25. So there's been a 5-fold increase, and that's been increasing over several years, kind of building up over time. And so we kind of thought it might be time to kind of tell the story a little bit. And that's what the report does. Very high level, lots of pictures. You can read it in 5 minutes if you haven't already. It's wonderful read to your children at bedtime. It goes very quickly. So it was an attempt to connect the dots, but also we highlighted some of the key actions we're taking in 2024. Some pertain to the TAP pilot and home as a healthcare hub, Dr. Califf mentioned, but another one is on a complete redesign of our pre-market program. We're really completely reimagining the enterprise with the goal of significantly reducing burden both to sponsors and to ourselves at the FDA, and make this a much more expeditious process, far beyond what our commitments are in the user fee program. And already we're setting up to do several pilots. Some are expansion of our triage program that we've done in IBDs, where kind of the lower risk 510Ks with a high quality submission, those will do a review in 30 days. One we did recently on a fentanyl test strip was in 16 days, to give you an idea. A second is to be able to do the review, if you will, in one seating. You can get up because it could take several days. We let people go to sleep, but it really is stay focused. And the third is on agile queuing. So things kind of are moving through, they're not sitting in the queue for someone to get to them. That's just a few things that we'll be piloting and a lot more that we'll be talking about later in the year.
Thank you, Jeff. And we have a printout of the innovation report. I have a copy of it at our booth outside, so check it out later today, as well as the CDER statement on this report.
Elias will be autographing it.
I will be autographing it and auctioning it off to the highest bidder.
Will be worth something.
Well, I don't, maybe for my mom. Let's continue this discussion. When we're talking about innovation, obviously we're talking about also advances in technology. So Peter, I'll come to you for this next question. How would you describe the current landscape of first generation transformative innovative products to reach the market?
Yeah, so maybe I'll take that as in terms of the gene therapies that we're seeing coming along. So the first generation of gene therapies that we're seeing, it's quite exciting because they're offering people a lot of promise in the rare disease field, and for some conditions that have had treatments, but now gene therapy potentially offers a much more convenient or a much fuller treatment. I think in some cases, it's really quite transformational. For instance, for patients with severe sickle cell disease, having the cell-based or these cell-based gene therapies, 2 of them now, one the CRISPR-Cas9 genome edited product that Dr. Califf mentioned in his talk. Those are really potentially major transformations, and although there probably will be better successor products to those, I think those may be around for a little while to come. On the other hand, there are probably some other first generation products, like some of the products in the hemophilia space, where I suspect for hemophilia A, we'll see better products coming down the line that might have better durability, et cetera. I think we're still getting a sense of how these products work. The first generation of products that we're seeing or that are cell-based, or the ones that are being directly administered using the adeno-associated viral vectors, are slightly problematic because you can't currently redose those products. So if they stop having effectiveness, you have to move to something else. That may change, and I think what may change, and it's just kind of over the horizon, is the use of CRISPR-Cas9 genome editing tools, which can be potentially administered using non-viral vectors such as lipid nanoparticles, something we've become very familiar with during the COVID-19 pandemic in the form of the mRNA vaccines. And so we may see second generations and then third generations of these gene therapies coming along in the next 5 to 10 years. So I think this first generation, extremely exciting. It's going to really improve a lot of people's lives right now, but I think more to come as we see the innovation build. And that's not uncommon. I mean, I think it happened in monoclonal antibodies. You know, the first generation monoclonal antibodies were a lot of these were murine antibodies that had their own issues of human anti-murine antibodies. It's all sorts of issues that came up, and then you know, the successive the development has really brought them forward. So I think that'll happen in gene therapy as well.
Yeah, there's so many examples, you know, elaborating on what Peter just said of the journey that, you know, where gene therapies are sort of embarking now. If you think about the HIV drugs, and what the first generation of these drugs were and where we are now. Think about Alzheimer's more recently. You know, you know, we have some, for the first time, some drugs that seem to have an impact on progression, but they also have, you know, some side effects and so on. And, you know, these, we can consider these, you know, these stages of development in major diseases as you know, a progression towards better and better drugs. We've seen it with cancer. Think about how toxic chemotherapies were only 20 years ago and where we are now, which is why it is important, despite the fact that some of our approvals or decisions may be viewed as controversial sometimes, to keep the field going, right? Because drugs get better over time, right? But if we don't allow the first, maybe not perfect, not entirely perfect wave of drugs to be tested and to be used, and for evidence around their use being generated, then we really, you know, cannot advance the field, right? So it is a difficult decision from a regulatory standpoint, because it comes down to accepting some residual uncertainty to Dr. Mark's point. So.
Absolutely, the benefit-risk calculation.
Exactly,
absolutely.
and benefit-risk calculation. That's always overarching, right?
Yes. So. Thank you for these remarks. I think we're now ready to start to get some questions. So for those of you in the audience, I welcome you and invite you to come to a mic. Meanwhile, we'll start off with our online audience. And Larissa, I want to turn the floor over to you for our first question from online.
Yes, we have some. We have some good online questions here. The first question goes to Dr. Cavazzoni. What role does artificial intelligence play in safety assessments, in your opinion?
I couldn't hear the end of your question. My apologies.
What role does artificial intelligence play in safety assessments, in your opinion, from the perspective of the Center for Drugs?
I can tell you about you know, today's role. I think the role will definitely expand, and you know, Dr. Shuren and Dr. Mark are really, I'm sure that will want to chime in on this. So to give you an example, we have been using for now several years, we have been using machine learning to triage and deduplicate adverse events that are reported to MedWatch. That has been ongoing for for now several years, and we used to have a team of contractors who would do this manually. Having said so, I think that machine learning and artificial intelligence as applied to safety will be increasingly important in identifying trends in signals that may point to safety issues. I think that, there's a lot of discussions about how in silico data can be used in the evaluation of real world data and real world evidence. And I think that safety is probably one of the prime areas where we have used real world data and real world evidence. So there will be a lot of new applications. And I'm sure that we will be confronted with this very soon, if not now.
Thank you very much. Let's take our first in-person question.
Okay, great. The FDA Modernization Act talked about reducing animal testing. And I'm just wondering where you've seen innovations in that regard. Has there been a reduction in animal testing? And when you're talking about residual risk and potential, is it more in the pharmacology side? Is it more in the safety side? Where reduction in animal testing has been seen in drug development? It's an IND thing, so a lot of it isn't public. So I'm just wondering if there's some examples you could give on where animal testing has been reduced in all three of you.
I can start that at least in the vaccine world, release testing has changed from certain tests used to require, for instance, non-human primates for release testing. Now that has transformed. I mean, they still some of these still use animals, but instead of using non-human primates, they can use rodents. In some cases, we're starting to see people looking at during developing gene therapies where they're using organs on a chip. Because especially if you're developing a human genome editor, it's much more useful to have a human organ system such as has been developed at NCATS at NIH on a chip, and to look at that than to look at even a non-human primate, because the sequence isn't the same. So I think we're seeing a transition here. It's not night and day yet. And there still are things where we still need animals, where animal experiments are still bringing important information to the field. For instance, mouse experiments in COVID-19 vaccine development still are very useful. But I think there has been this shift from higher species, non-human primates, dogs, cats, to rodent models, and then from even some of the rodent work to in vitro or in silico work.
And I think all the centers are intentionally looking for ways to reduce animal testing.
The 3 R's, right, and to the extent possible. And for instance, non-human primates, there is a program that they're either reused or rehomed at this point.
And the science needs to get there, right? So we'll be able to reduce animal testing even more as the science gets there. But the reality is that today we still need to do animal toxicology studies in many instances before putting a drug for the first time in humans. Certainly the organ on the chip technology is advancing. It is not there yet when we're talking about complex multiple complex physiological systems that are sort of interacting. But, the science will continue to advance.
I think too, we're also looking at how we reduce testing on people as well. So we've got, in silico clinical trials, phantoms used, with radiological imaging. If folks are interested, certainly on the medtech side, we have some of those tools available, regulatory science tools. We've got a catalog up on our website. So it is something to check out. There may be something up there that's relevant for your technology.
Thank you. Thank you for the question, and thank you for the responses, my panel. Let's take a question from this microphone.
Thank you. This is directed at Dr. Sherman. So Greg Cornever, Avanos Medical. It's been my experience that that there's this reluctance among the decision makers in medical device management level people to avoid a de novo at all costs. And PMA 510K, but de novo seems to be this fear of the de novo. Can you address that and assuage that kind of fear?
Yeah, I don't, so I've not experienced the fear of a de novo. Years ago, there was the issue around there because the de novo program was broken. It was really hard. We needed changes in the law. So we used to try to shoehorn technologies that really were for de novo into 510K because the de novo path was just terrible. So we actually have the highest numbers of de novos coming through in the past few years than we've had, for previously for the history of the program. So they are coming through. I think there's a different issue with de novo, is that there's more of them that don't get authorized than what we see for the rates for PMA and for 510K. And we've been looking into it a little bit, and a number of times these are issues around, also because it's so new, sometimes the developers too don't seek out advice and they come in the door, or they're thinking they're coming in one pathway and haven't had the conversations with us. So I would say if you're looking at de novo, one piece of advice is make sure to come talk. And we provide lots of opportunity to get on the same page. That's the most important thing to do.
The question. Larissa, I think I want to go back to you for another online question for our panel.
Sure. Sharon, last month, CDER launched the Home as a Healthcare Hub initiative, which is aimed to help home environment as an integral part in this program, and how it may impact the advancement of innovation.
This is part of our strategic priority on advancing health equity. I think, as everyone knows, there are millions of Americans live in primary care deserts. And in part, one of the challenges, we built our healthcare facilities administrators. It's not designed to meet the needs of patients. I'm a patient myself. I think the healthcare system sucks, quite frankly. Drive down costs. A lot of costs come out of dealing with things through brick and mortar facilities. Then we have to rethink how we deliver care and design it around people and bring it to people rather than expecting kind of where they are, like the home setting writ large has to be designed for it. And technology is a bridge. It's not just to connect a person with their provider, to provide interventions and disease management. You've got to design it for people. So this initiative on Home as a Healthcare Hub is to do just that. We've partnered with it to build a virtual reality prototype. And we're doing this collaboratively with for underrepresented populations in mind, and a start with diabetes. So we're partnering groups, provider groups, medtech, innovators. And that prototype is designed, will be an idea lab to help innovators then kind of think about how they would design technology, because we really need not what happens today, a lift and shift from the hospital to the home, which is, and then jerry-rigging it. It doesn't work that well. It's a Band-Aid, and it's what you do when you got nothing else. But also, no one wants the hospital in their home, quite frankly. I don't want a nurse waking me up every 2 hours, and I'm not eating green Jell-O. It's out of the question. But we need integrated technology. We need it that's medical grade, but a consumer kind of product. That's for people to use. So that's what that prototype will help too. And our goal is to have that kind of launched and out there, at least a prototype later this year.
Thank you, Jeff. Thank you for the question. Let's go back to this mic for a question from our audience.
Thank you so much for this opportunity. So this last decade was an innovation for oncology, and I think we are entering the decade of innovation for neurodegenerative diseases. You touched upon Alzheimer's. I wanted to get your thoughts on Parkinson's disease in particular, because biomarkers are still being researched, and then digital tools and wearables and endpoints. So I think this question is for all three of you. What do you see innovation in Parkinson's disease in the future?
I'm happy to start us from sort of talking about the drug side. Parkinson's disease is another area where there's a lot going on, beginning with moving from a diagnostic construct based on symptoms to a diagnostic construct, as I'm sure you know, based on actual sort of biology, biomarkers, and so on. And it's the same shift that we have seen with Alzheimer's. And so once that shift is made in how a disease is diagnosed and characterized, then it opens tremendous opportunities for drug development, right? Because then it is possible to use biomarkers in drug development. It is possible to develop drugs that have more precise or targeted mechanism of action, and so on. And so what's going on in Parkinson is very exciting, and it goes sort of hand in hand with what we have seen with Alzheimer's, and what we hope that we'll be seeing more and more in other neurodegenerative diseases as well.
I don't have a whole lot to add except to say that we are starting to see cellular therapies where people are using induced pluripotent stem cells, trying to deal with trying to address this, but they rely a lot then going back to what Patrizia was talking about, assessment methods to try to see whether they're working or not.
And I think complementary to that, also looking at other kinds of measures in partnership with others like Michael J. Fox Foundation that can be used in the home setting. So we can also increasingly move towards decentralized clinical trials and broaden participation. That's critical. And then, of course, MedTech has its role. There are, stimulation devices that we have today, but also thinking about the roles with other kinds of technologies and AI that can be assistive in Parkinson's disease patients, because certainly things around gait visual cueing plays an important role, and technology is a way to help provide that to folks. So there's a lot of innovations underway, and all of this is, and I think the exciting part is not to think in individual spaces, but thinking holistically for people, and kind of that combination in the drug, biologic, medtech space, and provide holistic solutions for people with Parkinson's and other neurodegenerative diseases.
Thank you, Jeff. We're very rapidly running low on time. This has been great. We could probably spend 3 hours talking, and it'd be so much fun. So we'll try to get through as many questions as we can in the next 5 minutes we have left. Larissa, I want to turn back to you for any questions from our online audience.
Yes, there is another question, and this question goes to Dr. Marks. So someone says, I heard you recently said that we need a $4,000 kind of computers before we get to $300 computers, and that was a metaphorical expression that was said in the context of the development of cell-based therapies. Can you please talk a little about this and tell how this metaphorical expression relates to innovation in the field of cell and gene therapy?
Yeah, so in the area of cell and gene therapy, and it could be either, but we've started with a first generation of products where the first generation of chimeric antigen receptor T cell therapies were about $400,000 to $500,000 for the product, let alone all of the other care that went around them. And I think one could say, well, that's crazy. It's an incredibly high price, but that was what it cost to get that innovation out there. But what we're starting to see now is the successive rounds of innovation, where now the innovation is in how can you make those products more accessible by bringing their price down by an order of magnitude. And probably within the next few years, we'll see chimeric antigen receptor T cells out there for a tenth of that cost. Either because of decentralized manufacturing efforts, or because some manufacturers, that's precisely what they're trying to do, is develop a product in a much more efficient manner for less cost. So I think, like many things, as we have more use, more development, we will see these issues come down. Part of it too is that the people making them initially have had to make them by hand largely, and then as the market has expanded, we're seeing fabricators who make devices to manufacture products making systems such that we actually we have like closed system devices that can make these products, again bringing down costs. So I think that was what that comment was meant to explain.
Thank you, Peter. Let's take a question over here.
Thank you for taking my question. Actually, it's to Dr. Marks as well. I mean, I heard you, and I fully agree with you that what we are seeing for the gene editing aspects is the growth pains that we got through the ABs. But I have a specific question. I was able to see the advisory committee for the XSL product and I was quite interested that out of the questions that are being asked by CBER, there's a need for standardization, meaning that I would like you to elaborate a little bit more on what will be the agency's future in terms of defining risk assessments aspects on gene editing risk, of errors. So please elaborate on that.
If I understand the question correctly, it's how do we deal with the risk-benefit assessment here in a more standardized manner?
Sure, and particularly that aspect of gene editing errors.
Yeah. So right, outstanding question. So the issue for genome editing is this concern about off-target effects. There again, some of that may have to do with the generations of genome editors. The first generation genome editor, CRISPR-Cas9 constructs, we've seen the ex vivo approach was very helpful because you could sequence afterwards and figure out what had happened. You could use in silico analysis to look for off-target effects, and you could use the cell-based analysis. And so right now for these first generation editors, we're using essentially two different methods, at least, or asking people to use an in silico method and a cell-based method to try to de-risk things. I think ultimately what will de-risk them further is moving to new generation CRISPR constructs, for instance, CRISPR base editors and CRISPR prime editors. The construct itself doesn't cause a double-stranded break like CRISPR-Cas9 does. Double-stranded break's bad because you've got DNA flopping in the breeze. Base editors and prime editors. Prime editors are basically a CRISPR construct which has been grafted to a reverse transcriptase, so it can actually essentially read-write essentially from the RNA template. It doesn't cause a double-strand break. Again, less potential problems. Still, you have to worry about off-target effects. Idea here is to look by essentially two methods to try to minimize off-target effects. And to date, at least the data coming out of some of the non-human primate experiments, as well as some of the initial, very initial experiments with initial CRISPR directly administered to humans, is at least looking encouraging.
Thank you, Peter. Thank you for the last question. And I think that will conclude our session for today. I want to thank our panelists for joining us on this great topic. Again, we could talk for hours on end. Thank you for interacting with us both online and in person. Let's give a final round of applause for our. Thank you very much. Thank you. So this will conclude our plenary session. We're going to take about a 20-minute break. For those of you in-house here, where I'm standing here is the CDER track, so please stay here. Over to the other side of the room is the CDRH track, and then around the corner is the CBER track. So please go to where you wish to be for the next segments. And thank you for joining us. Enjoy the rest of the conference.
Thank you very much. I hope we have not all defected to other to other tracks. A lot of people are probably still outside. So, but let's keep ourselves on schedule. And you know what? I got notes. Hold on one second. Okay, sorry about that, everyone. All right, so welcome to the Drugs Track. I hope that you enjoyed the plenary and are ready for the next 2 days of learning the newest and the latest from CDER, Center for Drug Evaluation and Research. As mentioned earlier, and I need to do this, as mentioned earlier, my name is Bernard Stoddard, and I'm with the Small Business and Industry Assistance, SBIA program. We will also welcome Lieutenant Commander Nora Lim from the SBIA team. Lieutenant Commander Lim will be facilitating the online questions from behind the room for today and tomorrow as well. We'll be using many acronyms throughout the 2 days, as I alluded to earlier on. If you need to clarify something, click on the online agenda. In the upper right-hand corner, you'll see a link to FDA abbreviations and acronyms. We entreat you to make the most of this experience. For those on site, please interact with the subject matter experts when they are available. Attend all the sessions that interest you. Learn all that you can learn. Submit your questions and network in person as well as online. And we're also offering YouTube streaming. So please go ahead and, you can text your colleagues if they're not able to be here in person and they can join by YouTube. But YouTube, no CEs. So let's do a quick poll question. For the online people, please, do it online. We'll capture your responses for records. For the in-house people, please show by your hands. So this question is, how many conferences have you attended? And your choices are, this is my first one, 2 to 5, 6 to 10, more than 10, and I attended almost all since they started in 2010. So for in-house, A, first one. Very good, thank you, thank you. I hope it's not going to be your last. B, 2 to 5. Thank you. C, 6 to 10. One person, 2 people, okay, in the house. Online people, please continue to vote. D, more than 10. Kevin's been with us for quite a long time. E, most since 2010. Nobody here? See me afterwards. I have a nice appreciation token for you. Well, I think it's nice. I don't know what you'll think. All right, excellent. Thank you all very, very much. So let's take a couple minutes to call your attention to our upcoming events. First, we have quite a few events in June. They're all in June. June the 13th, we will host our OSIS workshop, CDER Bioavailability Bioequivalence Study Sites and Inspections of Good Laboratory Practice. And in this presentation, our Office of Study Integrity and Surveillance, OSIS, will provide an understanding of the OSIS BA/BE program. And then on June 18th, we will present on the OTC Monograph Drug User Fee Program, or MUFA, describing the key elements of the program as it relates to MUFA user fees. On June the 26th, we will discuss the prescription drug promotion and the clear, conspicuous, and neutral final rule, providing background information on prescription drug promotion, including different categories and types of promotion, as well as regulatory requirements for each. Now, this slide here introduces our newly formed Quantitative Medicine Center of Excellence, which aims to facilitate and coordinate a continuous evolution and consistent application of QM for drug development and regulatory decision-making across CDER. Please go ahead and click on the link on the side, and that will take you through to the website for more information. Moving on, this slide provides the QR codes of SBIA's social media avenues. You can scan them now with your phones and be taken to the appropriate platform. And they will also be advertised across the screen banner during the breaks if you miss it now. Be sure to subscribe to your email updates so you can keep abreast not only of upcoming SBIA events, but also see the regulatory action, guidances, pilots, and initiatives. We will also post that information to our SBIA LinkedIn, and we have some postcards out there talking about SBIA LinkedIn. So be sure to follow us there as well. For day one, actually, I have another slide here. This slide looks kind of busy, but my colleague, Lieutenant Commander Renu Lal, was asking for volunteers in the hallway there. If you scan the QR code, it'll take you to a survey. We're trying to get your feedback on the different types of graphics and which one would be most engaging to you as a prospective attendee. So we would appreciate if you could take that for us, please. Plus, in-house, we have some of these flyers out there and also online. For day one presentations, we will cover a variety of topics, which include clinical trials safety data, clinical trial innovation, diversity in clinical trials, ClinicalTrials.gov. Are you sensing a theme here? Artificial intelligence and combination products. Then we'll finish up the day by offering a one-to-one open session where you can ask your questions in a little less public environment. But whenever you ask questions, whether you come up to the mics or whether you ask questions in a one-to-one, please keep in mind that we are unable to address specific application questions. And each speaker will entertain questions during their presentation. At that time, we'll have the mics, one to the left, one to the right. Please step up to the mics in-house, and then we'll alternate with the online questions. As you saw in the plenary, we'll take one question from each person and go around until we exhaust the questions all the time, whichever comes first. So now, without much further ado, let's go to our first session for the day, which will address selective safety data collection in clinical trials. This presentation discusses the recent ICH E19 guideline and its adoption by several regulatory authorities, including FDA, in an effort to facilitate the conduct of important clinical trials designed to answer questions about the long-term efficacy and safety of drugs. Our speaker today will be Dr. Mary Than Hai, who is the Deputy Director for Clinical within the Office of New Drugs at CDER. Please let us welcome Dr. Mary Than Hai.
Thank you, Brenda, and thank you for the organizers of this meeting for inviting me. So I'm Mary Than Hai, Deputy Director for Clinical, and I'm really excited to talk to you about this topic, selective safety data collection in clinical trials. I was also the rapporteur for the expert work group that worked together with many regulatory agencies and industry representatives as part of the International Council for Harmonization to develop the ICH E19 guideline. So what I'm hoping that you will actually get out of my presentation today are the following things. First, to understand what is selective safety data collection, to understand the importance of ICH E19 guideline to selective safety data collection, and particularly in global drug development. And finally, to describe situations where and how selective safety data collection can be implemented in drug development. I'm hoping that there actually be other learnings in here as well, but I'm only going to cover these three here as the learning objectives in the final questions and answer polling question, excuse me. So the outline of my presentation will first cover defining what is selective safety data collection. I'm going to give you some background for the development of E19. I'm going to talk about the scope of E19, and when I talk about the scope of E19, it's specific to the guideline or the guidance, the FDA guidance. An important part is actually what is the purpose of safety monitoring clinical trials. This is going to be that balance between safety monitoring and what E19 or selective safety data collection entails. And then I'm also going to talk about what we mean when we talk about reduction in data collection, or what data really should generally always be collected. And then I'll also talk about possible trial scenarios and methodologies for implementing E19. Oops, why is this going so quickly? And then I'll conclude remarks in the next steps. So first, what is selective safety data collection? I'm just going to say SSDC. SSDC refers to the prospectively planned reduction and collection of certain types of data in a clinical trial for drugs with a well-characterized safety profile where continued collection of comprehensive data may provide only limited additional knowledge of clinical importance. So a couple of things I want to emphasize here in this definition. Prospectively planned reduction. That's really important. You have to think about this, whether or not it's appropriate in your clinical trial. The other thing that's important is a well-characterized safety profile. So this is not a one-size-fits-all. This is not every drug, every clinical trial can actually implement SSDC. And really the last part is that once you get to that stage, you've already planned it, you actually know that this is a well-characterized safety profile for a drug. What is the gain that you have in this? Because if you fit those criteria, continued collection of all safety data that is comprehensive may actually not add so much more to the knowledge in terms of doing an assessment with respect to benefit and risk of that therapy in the indication in the clinical trial that you're investigating. So SSDC or E19 is really an innovative approach to clinical drug development, conduct of clinical trials. It's innovative, but it's not something that hasn't been done before. And so it's something that I really want to emphasize, that while this is innovative, it is not so novel that we need to be concerned so that we don't have experience with it. And I will talk about how we do have experience, and I'm not sure why it's moving forward. And the reason why I say that, some of you may have actually heard about SSDC, particularly at FDA, because we actually had issued a final guidance in 2016 entitled Determining the Extent of Safety Data Collection Needed in Late-Stage Pre-Market and Post-Approval Clinical Investigations. And I remember when this guidance came out, I was at that time the Division Director for Metabolic and Endocrine, and we had a lot of clinical trials that are being conducted to evaluate cardiovascular safety, both in the pre-approval and post-approval. Most of it was in the post-approval stage. And speaking with industry representatives and saying, have you heard about this guidance? Because you're focusing just on cardiovascular safety. You can implement some degree of focused, targeted safety data collection. And some of the things I heard back was, yes, Mary, we're aware about that guidance. However, not everybody in FDA actually is aware of that guidance. But the other thing here is that it's an FDA guidance. Our drug development program is global. And so, we can do that here for you, but we can't necessarily do that in Japan or in Europe. So that leads to the question, why was E19 proposed for development as the ICH guideline? It was proposed because, for the reason I just referred to, is that drug development is a global endeavor. And there really was an important need to have alignment between regulators and industry. It was essential to advance those principles that were laid out in a previously published FDA guidance on this topic. And just to talk a little bit about the ICH process, it started in 2017, and it involved not just the founding members, regulatory members, so that would be the US, EMA, and also Japan, but there are others since then, regulatory agencies that are part of it, and also their industry representatives. The expert work group was comprised of about 30 people working together over a period of about 5 years, meeting on an annual basis. And initially it was in Montreal, and then to Geneva, Switzerland, and then we came back to North Carolina, and we were actually also in Japan and Singapore. So It wasn't a painful process. It was actually a wonderful opportunity to work with many of our colleagues to actually get this across the finish line. This is moving too quickly, to actually get a harmonized guideline. And what the objectives of E19 was, first, to provide recommendations regarding appropriate use of a selective approach to safety data collection in some late stage or post-marketing studies of drugs where the safety profile with respect to commonly occurring adverse events is well understood and documented. And the thought here is that by tailoring that method of safety data collection, it may enable greater efficiency in the conduct of clinical trials, and that this may facilitate the conduct of large efficacy and safety clinical trials with a large number of participants and long-term follow-up. And as I mentioned, it was a 5-year process. We crossed the finish line. We got to step 4 of the ICH process in September of 2022, when we actually published the finalized ICH E19. And shortly after that, we're now in step 5. Shortly after that, FDA issued its final guidance in December, and this is signaling that FDA is now implementing ICH E19. Other regulatory agencies have done so as well. And the scope of E19 are the following. First, it's to interventional clinical trials. Most often it's in the post-approval trials. But in some circumstances, it can also be considered for pre-approval trials. It's not applicable to gene therapy or rare orphan disease clinical trials. And what I'm saying here is that the scope of the guideline, what we focus on when we wrote that guideline, we did not actually target gene therapy or rare orphan disease clinical trials. Certainly, I can entertain questions if that comes up about applying it here. And then finally, SSDC needs to follow the principles. If you follow the principles of SSDC in the guidance, it must also need to understand it does not alter your local regional safety reporting requirements. So what is the purpose of actually safety monitoring? It has 2 purposes. First is to protect the safety and well-being of individual trial participants. And the second is to obtain safety information to be used in the assessment of the risk profile of the investigational medicinal product. Now, at this point, you may ask, well, there seems to be a conflict when you define SSDC, and now you're saying that the whole purpose of monitoring safety in a clinical trial is to protect safety of the patients and obtain safety information. But if this is actually a prospectively planned reduction in collection of safety data, how do you achieve that? And you achieve that because, remember, you need to understand that your drug has a well-characterized safety profile. It does not mean that the investigator or the healthcare provider's responsibility to the patient is in any way at all removed. They still have that responsibility to ensure the safety of the patient. So that's the monitoring aspect of it. But the collection and the reporting of safety information that would not add any further to the understanding of the benefit risk of the drug does not need to be collected onto the CRFs and reported to regulatory agencies. So that's where there isn't a disconnect. It's just to make it very clear that this does not in any way at all take away the obligation to the investigator to ensure safety in the patient population. So when is the safety profile of a drug well understood and documented? The guidance does list several factors for consideration, but it's not considered to be an exhaustive list or determinative. So as a starting point, what is the regulatory status of the drug? So as an example, has the drug already been approved? That makes sense because if it's already been approved, it has actually already gone through a regulatory review process and an understanding of the safety profile. Now, one is the mechanistic factors of the drug. Do we know the mechanism of action, including any untoward effects? Is it one of many in a class, or is it first in class? If it's one in many, you may actually know a lot about the class of drug itself. What about the clinical safety database of the drug? What are the number and duration of exposure, the dose, intensity of the safety monitoring of that clinical safety database? And what's the similarity of the planned clinical trial where you intend to implement SSDC to previous trials? So how similar is it with respect to the patient population, the dosing regimen? The further away you get from the further away, I'm sorry, Note to self, animation sometimes can be problematic. The further you get away from the knowledge or the evidence that leads you to make the conclusion of a well-characterized safety profile, the harder it is to implement SSDC. The other thing that to consider is the clinical pharmacology. What do you know about how that drug is metabolized, how it's absorbed? Drug-drug interactions is important as well. The non-clinical data from the safety and pharmacologic effects, are they well characterized from animal studies or non-clinical studies? And if it's already an approved drug, what do we have with respect to post-authorization data, the extent and the quality of the post-marketing safety data? So when it has been determined and agreed to with regulatory authorities that SSDC may be appropriate for clinical trial, collection of certain information may be limited or reduced in frequency of collection. And this needs to be specified in the protocol. Another point to emphasize here is that really selective safety data collection is something that you propose in a protocol and you get agreement with the regulatory agencies. So things that you might be able to reduce or eliminate or reduce in frequency of collection, some of the non-serious adverse events, some laboratory monitoring, physical exams and vital sign data, or concomitant medications. And I have an asterisk here to point out that if a serious adverse event actually does occur, or an adverse event of special interest, some of this information that's actually that you can reduce, you would probably want to collect it in the surrounding of a particular serious adverse event to better characterize or understand that event, to investigate that event. The other thing to understand is that even if you're going to reduce some of those events and collection during the trial, they really need to be collected at baseline. So at baseline, comprehensive safety data collection is important because you need to understand the patient's profile. Should something happen, there can be a better understanding in terms of change from baseline. Also, importantly, generally, one should expect that the following will be collected: serious adverse events, important medical events, medication errors or overdose, intentional or unintentional, adverse events that lead to a study drug discontinuation, pregnancy and lactation exposure and their outcome, and adverse events of special interest, including lab abnormalities. And this, again, is identified in the protocol. So what are the situations where selective safety data collection can be considered? An approved drug that is seeking a new indication in a similar population to the one that is already approved. An example here could be a drug that's actually been approved for reduction in risk in atherosclerotic cardiovascular disease, and now the company comes in and they want to expand the indication for reduction in risk of peripheral vascular disease. Another situation could be it's an approved drug that's seeking to expand the label to include additional endpoints in the same patient population. So an example, let's say a drug that's actually approved for the reduction in hospitalization due to heart failure. And now they actually want to come in that same population to get some claim with respect to a patient-reported outcome, some quality of life claim in the label. Another one is a safety trial with an objective to investigate specific safety concern. This is particularly in the setting of, on the FDA side, FDAA requirements and PMRs. I mentioned that when we had the 2016 guidance, there were a lot of cardiovascular safety trials being conducted in the diabetes space. Well, there was very, very focused on one particular or particular safety events, and that's where it could be targeted. Some of the other non-serious or safety issues may not be as important to collect, and you can reduce collection in those other events. And finally, you can also consider it where you have a trial that's designed to provide additional evidence of efficacy when the current available data support a well-characterized safety profile. Now, this one here needs a little bit of explanation here. Let's say, for example, you actually have a product, an application before the agency, and it had two trials in order to demonstrate substantial evidence of effectiveness. One trial was positive, the other one was not. And there may be a variety of reasons why the other one was not. But either way, because of that, it could not be approved because it had not met the statutory requirement of showing substantial evidence of effectiveness. However, in both those trials and the whole program, there was enough information to understand the safety profile of the drug. In this situation, the company would be told you need to provide do another study to actually demonstrate substantial evidence effectiveness, but you can reduce some of your safety collection. And that's an important consideration, because it just means that important second trial that they have to do, a third trial they have to do, they can focus on actually showing efficacy. They've already convinced us, at least of the safety profile. Now, so what are the possible approaches to implementing SSDC in a clinical trial? The first one is just selective safety data collection in everyone, probably the easiest way of implementing it in the protocol and operationalizing it. Another one is to do a comprehensive collection for a specific subset, but you can have SSDC in the rest. A situation here would be if in your safety database, you don't have enough information on a subset of the population, let's say the elderly patient population. You might want to have a comprehensive safety data collection in the elderly population, but the rest you can do SSDC. You can also have comprehensive collection everyone initially and SSDC thereafter. A scenario here would be an LDL cholesterol lowering drug. It gets approved just on LDL cholesterol lowering, but pre-approval, they've actually initiated their cardiovascular outcomes trial. Since the drug hasn't been approved yet, they are going to implement comprehensive collection, but as they get to approval, they begin to know a lot more about the drug. After approval, they may consider implementing SSDC after approval. And then finally, another approach is comprehensive collection and representative subset and SSDC for remainder patients. So this can be done a random sample of patients or a random selection of regions where you can have SSDC in some and comprehensive in another. Either way, the approach really, again, needs to be spelled out in the protocol. So as I mentioned before, SSDC or E19, well, E19 is novel in the sense that it was just recently finalized and being implemented. SSDC itself is not necessarily something that has not been done before. It actually has. And what I have here on the slide here is a publication by actually two of my colleagues on the expert work group, Yukiko Watabe and Hironori Sakai, the senior authors in this paper. They are from Japan Manufacturing Association, Pharmaceutical Manufacturing Association. And they actually wanted to evaluate the landscape in clinical trials in New England Journal of Medicine to see whether there was any mention in the publication about the study that could have been implementation of SSDC. And this was done in advance of us actually, I think it was step 2, when we actually issued a draft guideline for public comment. Their approach, they did a literature search of trials that were published in the New England Journal of Medicine from the period of February of 2016 through December of 2019. This was the time frame before the guideline was actually published in draft form. And they reviewed the text and protocols and the SAPs of these trials to identify if there were any terms that would suggest it was adoption of selective safety data collection. And how they defined it was that these were trials that would not necessarily collect some of the adverse events that were commonly collected in clinical trials. And what they found was that out of 459 papers, the results of clinical trials, 9.6% of them actually had some terminology identified as adopting selective safety data collection. What's also interesting about the 44 trials in this time frame was that in terms of the therapeutic area, more than half of them were in the cardiovascular space, and most of them were cardiovascular outcomes trials. So they were very focused on identifying either a benefit or evaluating a safety concern. It was followed by infectious disease and then oncology. And then the other interesting thing is that over 90% of these trials were actually conducted before 2017. So well in advance of ICH E19 actually being published. So some key considerations before implementing selective safety data collection. First, is the safety profile of the drug well characterized? Do the study objectives and design of the trial support selective safety data collection? And then finally, has agreement been reached with regulatory agency on the protocol implementing selective safety data collection? So where are we right now, the next step? E19 is at step 5 of the ICH process, which means that since the guideline is finalized, it's all the different regulatory agencies and will now have to implement it according to their own protocol within their specific region or country. And I list here the members and regions where ICH E19 has been implemented. I haven't looked at the site, but you can go to the site to actually see if anything has been updated. But this is really exciting because, as I mentioned, in 2016, there was just the FDA guidance. And in spite of just being FDA, there were already some trials, which I'm willing to bet that if you look at those trials, they were global trials as well, where it's been implemented. But to have this actually signed on by many different regulatory agencies, this is really an opportunity for us to continue to promote this more broadly. I know that when we actually reach step 4, and essentially when you get to that point, the expert work group is disbanded. It was really a wonderful work group to develop this guideline on. But as a group, we really felt that this was something that was important as a guideline or guidance that shouldn't actually just collect dust on the shelf, that we need to really get this out and encourage companies and sponsors to actually consider this in their clinical drug development program. Hence the conversation today I'm having with all of you. And on that note, I do want to actually talk a little bit about the efforts that the FDA has to further promote selective safety data collection. As you may have heard, we announced in April the establishment of C3TI, the Center for Trial Innovation, which you'll hear a little bit more from my colleague in the next presentation. But E19, or selective safety data collection, was identified as a demonstration project. So if you on this particular slide here has some information. You can learn more about the guidance itself through a podcast. There's a summary of the guidance snapshot. But importantly is that any company that thinks that there's an opportunity for a clinical trial there's to implement SSDC, we would really encourage you to reach out to the agency to have a discussion on whether or not it would be appropriate to be part of a demonstration project and engagement more closely with the agency, and also to help facilitate any interactions with other regulatory agencies that would also need to sign on to an E19 approach for the trial. So in conclusion, ICH E19 guideline will have a significant impact on the feasibility and efficiency of clinical trials designed to yield important new medical knowledge and advance public health. So now let's see how I did in the learning objectives. So your first challenge question, selective safety data collection refers to A, eliminating safety data collection entirely in a trial with a drug that has a well-established safety profile. B, prospectively planned reduction in collection of certain types of data in a clinical trial for drugs with a well-characterized safety profile. And finally, C, only collecting safety information in patients who experience an adverse event. Not A and D. So I don't know if, is there a voting mechanism now? Will people raise their hands? Just raise hands? Okay, so let me just go through again. So who says selective safety data collection refers to A, eliminating safety data collection entirely in a trial with a drug that has a well-established safety profile? Excellent. B, prospectively planned reduction and collection of certain types of data in a clinical trial for drugs with a well-characterized safety profile. Excellent, very good. That was the right answer. And obviously the last one, only collecting safety information patients. No. All right, second question. I hope this one's a little bit more challenging. Which scenario would not be appropriate for SSDC? A, an already approved drug for reduction of cardiovascular disease risk in type 2 diabetes is now being studied for reduction in peripheral vascular disease and type 2 diabetes. B, a phase 2 dose response study of a first-in-class immunomodulator being developed for the treatment of pediatric dermatoses. And C, a post-approval safety trial evaluating the risk of renal failure where the only safety signal of concern pre-approval was imbalances in serum creatinine elevation. So again, the question is, which scenario would not be appropriate for SSDC? It's always those negative questions. So Is A not appropriate for SSDC? Anybody raise their hand? B, is a phase 2 dose response study of a first-in-class immunomodulator being developed for the treatment of pediatric dermatosis. Is that not appropriate for SSDC? Okay, got some hands up. And then C, a post-approval safety trial evaluating the risk of renal failure where the only safety signal of concern pre-approval were imbalances in serum creatinine elevation. Is that one not appropriate for SSDC? Okay, there were some folks who didn't want to commit, but the show of hands, the only show of hands was for B, which is the correct answer. It's a phase 2 dose response study of a first-in-class immunomodulator, so you probably don't know a whole lot about of the safety profile of the drug at the phase 2 stage, also first-in-class. I threw in there the pediatric dermatosis, the pediatric population. The guideline doesn't actually exclude pediatric studies, but I think that there's probably always going to be a little bit more caution in a more vulnerable patient population. So with that, I am happy to entertain any questions.
Okay, thank you very much, Dr. Tanhai. All right, so we know, I'm sorry?
Do you want me to sit down?
You can have a seat if you want to be more comfortable. Can she have a seat? She's not mic'd up. I've forgotten, yeah. I'm just delirious with happiness, yeah. Okay. All right. Okay, questions in the room?
Yeah.
Please, we have two mics, so please, if you want to come up to another mic, please.
Yeah, thank you for that. I'm happy to see ICH 19 moving forward that way. I know there's been trials back in 2012. I worked on trying to get FDA convince them to reduce collection. My question is really a logistical question: is how do you manage a protocol, like a free-form response of how you feel? Because they're always going to answer maybe something that was not part of what you're restricting in terms of data collection. So that's my first thing. The second thing is, when I thought of reducing data collection, it was more like data reporting. So we would have the data, but we wouldn't actually report it to the agencies. It would always be in the case report forms or somewhere. So I'm just curious about the differences there and how you actually implement that on the case report form too. You just, if there's a free form part of that to answer about how do you feel?
Thank you for that question. So actually, specifically in the guideline, and I didn't put it in the slide, but when we talk about SSDC and E19, it's about the collection of the information on the case report form and then to actually report it to the regulatory agencies. And it was very specific in that setting there because we know that if sponsors are telling their investigators that you need to actually collect and capture this in the case report forms, that is actually additional work that you do. You have to actually solicit that information. The question you really have to ask is that why do you need to solicit that information? That's very different from monitoring. And whether or not, if you're going to collect it, then you're going to have to feel the obligation of actually reporting it to the regulatory agencies. So that was the distinction I was making with respect to safety, selective safety data collection. If you don't think that this is something, whatever the safety, I shouldn't say the adverse event, whatever the safety information is, whether it be EKGs or physical exams, that's not necessarily a safety information, but it's a test or a procedure that you're doing. If you don't think that that's actually necessary to continue to ensure the safety of the patient, then that's not something that needs to be put in the protocol to collect in the CRF. And if you don't have it in the CRF, you won't be reporting it to the agency. So I hope that actually answers your question. And I forgot your first question.
No, it just had to do with, if you ask the free-form question, the investigator says, how do you feel? Then some of these issues may come up. Do they not get put on the case report form then if the investigator is asking a question, how do you feel, which is pretty typical in a clinical trial?
So I'm not sure if I understand. So you're asking the investigator, how do they feel, or you're talking about the patient?
The patient. So the investigator asks how you feel, and the patient writes down some of these things that are not part of data collection. They go into some other are they not then reported, or are they just, I mean, they're in the case report form at that
Consequences, yeah.
point? We've limited reporting on some of the adverse events I've worked on. I'm just curious, how you manage that.
Well, so I think how I'm going to answer this without trying to actually get into advising on a particular drug development program, because I don't know the details of it. I'm going to assume that as an investigator, when your patient or the participant comes in, it's just, I would assume that's just a good patient physician practice, is to ask, how are you doing? How have you been since the last visit? And they may, talk about a lot of things. Again, it's protocol defined. Remember, it's prospectively planned decision in terms of what's going to be collected and what's going to be reported. If the patient actually said, well, since the last time I was in, I had such and such, and as the investigator, you are the investigator, by the way, it is a requirement that they have to be trained and have the expertise to be able to carry out the objectives or the protocol and the objectives of the trial. If they determine based on their scientific knowledge that this is not an essential part of ensuring the safety of the patient and it's not in the protocol, then that wouldn't be something that's going to be reported to the agency. Does that help?
Yeah.
Next question to my left, please.
She's first. You want first?
We're alternating.
Okay. So I have a question about how do you define the safety collection for the trial with infant, children, and adult?
Okay, so selective safety data collection, infants.
For infant trial, children trial, and then adult trial.
Okay. Well, the criteria, or as I mentioned, the things that you have to absolutely be sure is you feel that the trial or the drug has a well-established safety profile. And then after that, then the question is, can that be applied to the trial and the patient population? So if you actually have a drug that has a well-established safety profile, but it's never been studied in the pediatric population, it's probably something that you would not be comfortable. And the other thing here is that you obviously need to have buy-in from the regulatory agencies as well. You would need to make that justification that the population that's going to be studied, and where in a trial where you want to implement SSDC, you have enough data from prior studies, prior evidence, and I gave you a whole long list that is not determinative but can be helpful. If none of those actually can be applied to the new patient population, it's probably not something that you want to implement in a pediatric or a neonatal population. Does that help?
Yeah, that's helpful. Yeah, that we experience with COVID vaccine also. Thank you.
Okay, thank you. Let's go online for a question, please, and then we'll come back.
How would a sponsor reach agreement with FDA on the protocol implementing SSDC? What is the best way, and how long would it take for FDA to reach a decision regarding if SSDC is acceptable?
Okay, so that's a very good question. So this would be implemented as part of a protocol, and most likely it will be, let's say, a pivotal trial. I suppose one could actually make an argument it doesn't have to be a pivotal trial, but more likely it will be a pivotal trial. Protocols would be submitted to the agency. You could flag it with specific questions, as a submission to the agency. And I can't remember the exact time frame, but usually the time frame in terms of responding to these types of questions is probably in a period of 45 days to 2 months. Because there's an understanding if it's a pivotal trial, we don't want to wait too long, because companies actually need to be able to get this understanding to move forward with that. Certainly if this is something that you want to implement in a phase 3 program, you can certainly bring it up in a phase 2 meeting as well, where you may not have the protocol all spelled out, but you want to flag for the agency when we're planning on implementing selective safety data collection, and we will also be sending in the protocol. So those are different ways that you can approach the FDA about this being put into the protocol and getting feedback on it.
Thank you. Let's go. If you want to step up a little bit close to the mic.
Can you hear me?
Yeah.
So this is actually very similar to the question that was raised online Considering that this guidance has been around since 2016, have you already received protocols with this SSDC? Second part is, now that it is part of demonstration project, how is it different? Could we just submit the protocol with the reduced safety data collection for a post-approval study under our IND? Or what is the pros and cons of doing it via the demonstration project versus to the IND? Thank you.
So very good questions. The first one, are we receiving it? We have not established a database or a process to systematically identify these as they come in. That's something we're working on. I think that's really important. It's nice to track to see that I'm hoping to see an increase on it. And I'm hoping that the efforts like from today or other meetings, by the way, I don't know if anyone's going to DIA in June, but there will be a session on E19 as well, that getting that message out there, we will see an increase on that. So the second question is, what is the benefit of being in a demonstration project versus actually submitting it to an IND and getting agreement? And I'm going to actually, I don't know if Dr. Bugan will respond to that in his talk. I think the benefit is that you'd have greater interaction engagement with SMEs within the agency at many different levels to give you feedback on that protocol. And particularly if this is one a very challenging one, you may have to engage also with other regulatory agencies. It would have to be accepted in the program. The other aspect there is that there has to be an understanding that if you're accepted into the demonstration project, there is a willingness on the sponsor's part to actually publicly share those learnings with the public. And that's really important. If you think about MIDD and CID and also the drug development tool qualification program, you heard that from Rob Caleb this morning. He's talking about all the different approaches that the agency has for innovation. It doesn't help if you have innovation, but you only keep it close to your chest and you don't share it. So it's really important to agree to share that with others so they can say, this actually does work, or it didn't work. What can we learn from? Why did it not work? So it's not always the success stories that we want to share. Sometimes we have to share things that didn't go well so we can make improvements on it. Does that answer your question? Okay.
Thank you. Let's go with the second question in-house because you've been standing there for a little while.
Thank you for the wonderful presentation and informative discussion on the selective safety data. I wanted to take things a little bit to a different level. We are seeing and experiencing a lot of activities in clinical trials that are happening remotely with the modernization of clinical trials, and with that, the use of technologies to enable these trials. So in terms of, in that context, are there provisions from the agency perspective to deal with the selective safety data collection centrally versus remotely, and in the context of E19 and others?
Okay, so how do we apply, if I can paraphrase, how would we be able to apply selective safety data collection in different novel ways of conducting trials, such as remote monitoring or decentralized trials or digital health technology? Does that sound?
Yes, or even real world evidence. I mean, yes.
Okay. Well, I think first and foremost, from selective safety data collection, you need to, I'm going to sound like a broken record, but you need to be sure that you have confidence in the safety profile of the drug and you know what you need to collect and what you don't need to collect. So once you have that, then the question is, the things that you are going to collect because you think that it's important, that's where you're going to implement more novel approaches collecting, whether it be remote monitoring or digital health technologies. That's perhaps a different area that you have to address because you still need quality data in that setting there. So if you're going to, let's say, still collect a particular adverse event, but your intent here is to collect it through some DHT, some tool that's novel, there still has to be an understanding that that tool can actually adequately collect that information, that it's reliable. So that speaks to the quality of the data. So SSDC does not absolve you of that obligation at all, and sponsors will still need to make sure that that new technology can actually deliver on that. I think where the two will really go hand in hand is that when you start to reduce collection of unnecessary information, you actually make it more feasible to do things such as decentralized trial, remote monitoring, because it may mean that patients don't have to come in every month. They can come in on a 6-month basis. And if some of those events that are still considered necessary to ensure adequate assessment of benefit and risk, if those fewer events that are being collected can actually then be done in a remote setting, it does facilitate, it does go hand in hand. Does that?
It's okay. We can discuss later. Thank you.
Okay, sure.
Thank you very much. This was our last question. If you have more questions for Dr. Tanhai, please get her in. You'll be around a little bit.
Yes, until lunch.
Okay, thank you.
Thank you.
Okay, our next speaker, I got so many mics. Which one? Our next topic is all about enhancing clinical trial innovation. And this presentation will address new efforts and share opportunities for stakeholders to engage with CDER on clinical trial innovation. Our speaker will be Dr. Kevin Bugan, who is the Deputy Director for the Office of New Drugs within CDER. Welcome, Dr. Bugan.
All right, good morning, everyone. Thanks again to Brenda and the REdI folks for putting this on, and great job at getting us back in person. As I think I raised my hand earlier, I think I did my first REdI conference probably 10 years ago, maybe 11 years ago. So it's just really great to be here. And then most excited that O&D is kind of taken over this morning, and maybe even filtering out into the rest of the day, because there's a lot of discussion about clinical trials. And I think clinical trials and clinical research is just so critically important to what we do at the FDA to help promote health and help improve the quality of life for patients. So what I'm going to talk about today is the new CDER Center for Clinical Trial Innovation. So I think you heard about this multiple times already today, and that's excellent. But I'm going to just reinforce it so that you leave today really remembering C3TI and what C3TI is, and it's not a robot from Star Wars. But hey, if you remember that, you'll remember C3TI. So C3TI, or the CDER Center for Clinical Trial Innovation, was launched on April 15th, and it was intended to enable and amplify innovative approaches to the conduct and design of clinical trials, ultimately to improve the efficiency and effectiveness of drug development. Now, there have been a lot of programs going on for many years within CDER and across the FDA, enable or intended to enable or promote clinical trial innovation. You heard about several of them also already this morning, such as the Complex Innovative Designs Program, which is focusing on new statistical methods or approaches to designing clinical trials, such as master protocols or building in interim analyses, et cetera. You also heard about, had a great question here too, about the use of digital health technologies. So these are things like wearables that can enable patients to more directly contribute to the data that's being generated and collected in a clinical trial. We also have programs to qualify drug development tools, which are intended to sort of give both the FDA and sponsors that reassurance of the reliability, verification, and validity of those tools that are being used in clinical development programs and model-informed drug development, and I could go on, many programs here. And really what C3TI is trying to do is help facilitate those existing programs, help them amplify the great message. I kind of say, like, I'm the cheerleader of all those existing programs to help get that message out to groups like this. You all are learning about clinical trials today and learning about the most innovative approaches, not the approaches that have been around for many, many decades, although those are tried and true and still excellent, but really helping to ensure that as we design the trials of tomorrow, that we are always using the most fit for purpose and appropriate tools and approaches. All right. Now, so yes, did I go the right way? I did. Okay. So how did we get here? this really kind of stems from a personal passion of mine. So in 2020, like many of us, we probably experienced something different during the pandemic, and maybe we stepped up and played different roles trying to help out because we all wanted to contribute to helping folks get through the pandemic. And I played a role in the US government's response on the therapeutics development side of things, what was called Operation Warp Speed at that time. I was not working on the vaccines, I was working on the therapeutics. So this is the drug track over here in CDER. And what we were doing was launching a lot of different trials. Most of them are master protocol trials in collaboration with the NIH through the ACTIV Consortium, which was the Advancing Clinical Trials, Clinical Therapeutics, and Vaccines. And through that process, we experienced all the challenges that every sponsor and CRO was experiencing during the pandemic, which was it was very hard to get sites ready. It was very hard to launch these new trials. All the trials and sites were trying to make their clinical programs more decentralized because patients were in lockdown. They weren't able to come in for visits as often as they needed to, particularly if you were in the outpatient setting. And so it was very challenging. And yet we had all these tools. We had these ideas around making trials decentralized. We had digital health technologies. We had more advanced, complex, innovative designs. But it wasn't quite getting the level of adoption that we had expected. Now, the great news is fast forward a couple of years, we had learned we could do some of these things. And we were seeing this tremendous amount of guidance, lots of new programs being released from the FDA. But then suddenly come around 2023, again, a year ago, and we look back and we started to see some backsliding. And we started to ask ourselves, why is this? So we opened up a public docket last October to try and understand from external stakeholders, sort of what are those barriers or facilitators, if there are things that work well, we want to know what those are too, that help promote the adoption of innovative approaches to clinical trials. We pulled all that information together, and just a few months ago in March, we had a public meeting to discuss what those were. And what we heard were many different things relating to, oh, we have great examples of this where it's happened. We have phenomenal guidance, but a lot of times we just don't understand how to do this at an operational level. Every trial is a little unique. Every patient population, every drug, every disease, it's always a little bit unique. And so the guidances, while are absolutely necessary, aren't always sufficient. Like, we need to have these conversations around how we implement these new approaches each time we do it so that we can all learn. And this is really where C3TI comes in. So the idea, again, to promote and enable and amplify all the great information related to clinical trial innovation, we want to do this in 3 key ways. We want to share lessons learned. So every time we hear about a great trial that's maybe implemented these selective safety data collection approaches, as Dr. Tanhai mentioned this morning, maybe some of the other programs that I'll talk about in a few minutes, we want to share that broadly, kind of put it together in different ways, thinking about it from an implementation science or dissemination perspective, because one trial in the context of oncology may not be super relevant to someone doing a trial in dermatology. However, there's absolutely lessons learned that can be shared if they're adapted and contextualized in different ways. So we want to sort of play a role in that space. Secondly, we want to just communicate and collaborate. Going out, I think I've been at a meeting every week now since we launched in April, and I'm happy to do it, especially when they're local and arranged by Brenda. But the idea that you can engage with the FDA, and we are open to engage with you, and we want to talk to you about these programs and these trials, especially if you have questions around implementing something novel, is really important. That level of openness and interaction and collaboration, I think, is critical as you move into these novel spaces. It helps to kind of de-risk those areas for something that's not new, or for something that is new. And lastly, we want to expand opportunities for innovation. We recognize we have a lot of programs, as I showed on the slide a few minutes ago, but we also have these new areas such as selective safety data collection. We also have new approaches to making trials more pragmatic or integrated at the point of care. And we need to be able to very nimbly launch sort of new efforts to promote those without having to stand up large new separate entities or separate programs. And this is where C3TI comes in. We sort of have a central operations or infrastructure that allows us to very quickly identify the new area and put together a program where we can interact with sponsors on these new trials trying to apply these new approaches. Also, in addition, underneath what C3TI is doing, we have these 4 key activity areas. I've already mentioned the internal and external communications and engagement. So we have an excellent communication shop that puts together these wonderful slides with fancy things like QR codes that I think are really great when you're out at meetings such as this. We also have a central mailbox, the CDER Clinical Trial Innovation at fda.hhs.gov mailbox, where you can send in any question regarding anything that's innovative related to clinical trials. Doesn't mean C3TI is going to answer all these questions, because I don't know everything about everything that is innovative clinical trials, but I know all the people within CDER. Like, I'm very good at making friends and relationships within CDER, and I will sort of be your friend to the friends, and I will help you connect to them. And all of the different programs that we have across CDER, I sort of reroute, make sure if there's not a program for a particular question to go to, who is the most relevant SME or subject matter expert, and try to connect you with that subject matter expert. So we'll be your telephone operator, your kind of electronic yellow book pages, behind the scenes within CDER. Thirdly, we are launching, or we did launch last Monday actually, a centralized knowledge repository that is available to you as well on the external our website for C3TI, which if you click that link, you can kind of get into, and I'm going to also talk about in a minute. This is a phenomenal, very search-friendly, centralized one-stop shop for all of the different records or links out to information on various different clinical trial innovation activities going on, not just within CDER, but also at the FDA. It's not intended to have everything you need. It's not the encyclopedia of all things clinical trial innovation, but it's like those yellow book pages. You can find a link out to another web page or another program or to a case study or to our guidance, et cetera, that can sort of at least get you started on that path. And we're trying to make it very search friendly. We have a nice taxonomy and tagging, and that will improve over time. And then lastly, we have our demonstration program. This is how we will expand those opportunities for innovation by standing up a centralized program that can launch new projects in different spaces of clinical trial innovation whenever we believe we need a little bit more of a push or a nudge to get things going in the space of adoption. Selective safety data collections have been around for, again, probably 10 years. We've seen some pretty good implementation. I think as Dr. Tanhai mentioned, they'd seen about 10% implementation prior to the ICH guidance. It's hopefully ticking up since then, but we'd love to see it implemented everywhere it could be implemented. And so this is why C3TI will step in and help provide some additional assistance. So the Compass, this is the direct QR code link here to take you to the C3TI external version of the Compass. We do have an internal version as well. Sorry, you guys can't get access to that one, but that's the one that's got a lot more information. I think it's got about twice as many entries. And the only reason they're not shared with you is because they do contain more confidential or commercial confidential information. So we're trying to pull in links to actual protocol submissions, our reviews of those protocols, ultimately the study reports that come in that have the data from those studies, and then also our reviews, such as the marketing application reviews where we reviewed those studies, because how we internally interpret that information is really important and helpful. We also look for precedents. When we're doing reviews, we don't start from scratch. We go back and see, what did we think about this type of an approach in this type of a setting in a prior trial? And so our internal Compass has a little bit more information. But as that information becomes publicly available, and particularly, and I'll talk about this in a minute with the demonstration programs, as sponsors share that, it's no longer then considered commercial confidential. We can start to add it into the external version of the C3TI Compass. But the idea, again, behind the Compass is to sort of help share those lessons learned, create a centralized repository where you can find the links to all that information and go back to it on a regular basis. It can hopefully maybe be your one-stop shop for innovation in clinical trial space. So if you don't remember exactly what was that website for the RWE program, although I think we do a pretty good job with search engine optimization. So if you type in Google FDA RWE, you'll find the RWE program. But in case you didn't know exactly how to search something up, you can find those links through the C3TI Compass. So the demonstration program, as I mentioned, this is a centralized sort of infrastructure to launch projects in targeted areas to explore more scalable improvements in the adoption of these select approaches. I think the SSDC is a phenomenal example because you just had the sort of primer on that and you understand really what that is. But again, where we think this might be perfect in the appropriate settings where we have a well-established safety profile and it makes sense in the particular new setting of a new trial in that population, we're still not seeing the level of adoption that we think would be most appropriate. So we want to sort of launch a new project to do demonstrations to help give that a little bit more of a nudge. So as Dr. Tanhai mentioned, when you submit a proposal to be a demonstration trial to the FDA through C3TI, what you'll get is access to additional CDER subject matter experts. So of course, you'll be working with the review team. They are the experts on trials in that particular disease with those types of drugs. But we'll probably also give Mary Tanhai a call, and she might be a subject matter expert. She might even show up at your meeting when you come in to meet with us, so that you have the best and brightest who understand the guidance and the policy and its original intent, and helps that review team who may have not seen this particular approach. Again, if SSDC has not been used in a therapeutic area before, that review division just wouldn't have had that hands-on experience. They certainly will understand the guidance and have read that, but they may not, again, have that hands-on experience. Dr. Tanhai has had lots of experience, so we'll pull her in, or others across the center, and we're working to identify those who have built that experience. So it's not always going to be Dr. Tanhai, if I can help her calm down. Secondly, what else we'll do is, as these trials go through maturity and they're completed and the data is locked and cleaned up and it's sent in and we're starting to review it, guess what? Inspectors come. And now a great thing about REdI was they used to do this, and they will go back to that. They would go out to field offices and you would sort of be there, and you'd have compliance people in the room, or you'd have the ORA, Office of Regulatory Affairs inspectors in the room. And it was nice. You could really get to know who's going to show up at your site or at your sponsor and start to audit your trial master file and things. So what we're going to also be trying to do is think about prospectively, when it's time for inspections to take place, we're going to be working with our folks in the field and in compliance to have sort of a proactive plan, and also make sure that everybody's trained on what these innovative approaches are, because inspectors also may not have had hands-on experience. They may not understand some of the very good questions that we heard from the audience earlier that, oh, well, the patients were saying this, it's in the source, but I don't see it in what was reported to the FDA. Why is that? That was because that trial maybe used a selective safety data collection approach. In addition, trials are global, so we are also going to be working on some of the global harmonization aspects. Can't guarantee that we'll always be aligned with our global regulators, because they have different sort of regulatory frameworks or country type laws and regulations that they have to deal with, but we will at least work across the aisle to try and make those connections and help them understand what our perspective is, at the same time as that they can understand ours. Ultimately, this will hopefully support implementation. Sometimes trials fail or drugs fail, that's okay, but we still want those trials to have a successful implementation of the innovative approach. And then we want to be able to share the information from that and what we learned. And this will ultimately, hopefully, enhance the body of knowledge across the board on clinical trial innovation. As I mentioned, we have 3 project areas. Initially, we'll hopefully be able to launch more because there's sort of economies of scale. This is hopefully a rinse and repeat model for supporting clinical trial innovation. So after these 3, we have a few demonstration trials lined up. We can move into other areas. So the 3 are Bayesian supplementary analyses, and I'm going to walk through each of these so you get a little bit more information. As mentioned this morning, selective safety data collection, and then streamlined trials embedded into clinical practice or STEP. All right, so Bayesian supplementary analyses. So I'm not going to give you guys a training on Bayesian statistics, because I probably wouldn't do a very good job, but let's just say it's different from the traditional approach to statistics that's used in clinical trials, which is a much more frequentist-based approach. And what we're trying to do here is increase experience with clinical trialists, sponsors, their development or product development teams, and FDA review staff on using Bayesian statistical methods in their clinical trial settings that are otherwise what we would consider simple. And they're by far anything but simple, but the difference between simple and complex is sort of a terminology that we use to refer to trials that maybe have adaptive elements, multiple arms and agents, like in a platform trial or basket trial, et cetera. But the idea is we'll work with the sponsor, C3TI, and our demonstration team will work with the sponsor and the review team and the reviewers who are going to be reviewing that trial's data on implementing some additional supplementary, or maybe what you might consider sensitivity analyses prospective in the clinical trial using the Bayesian approaches. And what this does is it doesn't change your primary analysis, so you don't have to worry, will FDA be able to accept my decision because it's a different approach that we've never used before. You've kind of de-risked the use of Bayesian by making it a supplementary analysis. And the benefit is you get experience. You get to use sort of the different programming methods. You might build up some libraries of different models on your computer. Apparently, I'm not a statistician, but apparently they build up all kinds of different programs, and they like to rebuild those and put them back together in different ways. But if you've never done Bayesian methods before, you won't have any of those libraries. You don't have anything to start with. So this is a great way, again, on both sides, sponsor side and internally, for us to build some experience and put those libraries together. Most optimally, the trials that we're going to be looking for are phase 3 efficacy and safety trials or non-inferiority trials. Again, not borrowing any data. That's a concept in Bayesian from any previous trials. So this is going to be a very much sort of frequentist approach for the primary analysis. However, I will say, if you are interested in doing a primary Bayesian analysis trial, you can definitely get in touch with us at C3TI, and we can potentially discuss how we could collaborate with you and with the Office of Biostatistics within CDER to help support that trial, because we would not be against that at all. Next is the selective safety data collection. I won't have to go through this since you had an excellent training on it this morning. But again, the idea with being a partner in C3TI is that you get access to those additional resources, both from the subject matter expert side on the policy front and those who maybe helped originate the guidance, but also some perspectives, planning and support when it comes time for inspections and potentially with any global regulator harmonization type conversations. I think Dr. Tanhai went over the eligibility criteria as well earlier this morning, but this would be a trial most likely for an approved drug that's seeking a new indication in a very similar population. So you're going to be able to kind of extrapolate that well-established safety profile into that new population, maybe approved, seeking an expanded label, or it's evaluating a very specific safety concern. So this might be something like a post-marketing required trial for a previously approved product. And lastly, it's going to be a trial designed to provide additional evidence of efficacy when the current data is to support a well-characterized safety, when the current data do support a well-characterized safety profile. I think I'll maybe use that point to illustrate with all three of these demonstrations and probably all future demonstration projects. We really want these trials in the demonstration to be a regulatory grade. So they are intended to be submitted to the FDA and intended to support some sort of regulatory decision. Because this is something we heard when we were discussing with external stakeholders, that when it's a regulatory trial, that carries a lot more weight precedentially than if it's a non-regulatory trial, because it means the FDA made a decision. They had to sort of weigh in on it. And that's something that we can all look at as helping us understand how are they going to maybe interpret the findings from a trial in a future scenario. Last but not least, and my most favorite, well, I don't have any favorites. They're all great. Especially with Dr. Tanhai here, but the streamlined trials embedded in a clinical practice or STEP demonstration project. So this is a program that aims to promote the adoption of more pragmatic trial design functions. There is no one single pragmatic trial, although I think Lung Pragmatica, which is an oncology trial, is probably pretty far to the most pragmatic you could possibly be. But when you think about creating a trial and making it more pragmatic, there's all sorts of different elements that you could be exploring. You could be exploring the eligibility criteria. You could be exploring the endpoints, ways to make those easier to be implemented. Ultimately, you're looking to see how can you simplify the trial such that it could maybe even be conducted in a more point of care like setting, because this is where most of the patients are. Again, if we have to bring all the patients into Johns Hopkins, which is a phenomenal academic medical center, it's going to be really challenging to reach out and get patients from the panhandle of Maryland, right there. That's very far away. So thinking about how we can streamline these trials and help us bring it back into the point of care setting, which has a lot of other benefits. As far as other advantages, obviously it can make the trials more resource efficient. While FDA is not in the business of making trials, cheaper or easier to conduct, I think kind of we all should be, because as you make trials cheaper and easier to conduct, you're able to sort of launch and run them more efficiently and more quickly. And that will ultimately get products to patients faster if they're safe and effective, or it'll help us sort of find the products that aren't safe and effective more rapidly, which is important as well. It could also help us attract broader study populations, as I mentioned, because we don't have to have our patients traveling, 100+ miles to get into our studies. And it can also ensure that all of the study objectives can be completed as robustly as possible. As for the eligibility criteria, the trials that we would be looking for in this particular demonstration would be those looking to test out or explore and incorporate any sort of pragmatic design element. It doesn't have to be completely pragmatic across the board. There's sort of all these different features of your trial you could be considering to make more pragmatic. Maybe you want to add some decentralized trial features, use a digital health technology, maybe even use selective safety data collection to reduce the burden of safety data that needs to be collected, which might enable that trial to be sort of conducted in a more closer to point of care type setting. Secondly, it most likely should be a trial later in pre-market development when the safety profile is reasonably well established. This just allows us to sort of kind of more focus on certain things and not have to worry about all the novelty that might be associated with a new product that we don't have a whole lot of experience with. Could also be a post-approval trial. This could be a product being repurposed for other uses, expanded indication, new populations, et cetera. I'm going to kind of just round out here, and then we can move into the questions. I think we've got plenty of time for questions. With, this concept of how do you promote clinical trial innovation? It's kind of nebulous. And first of all, most people like, what does an innovation even mean, right? Because some of these things we've been doing for decades. I think the pragmatic clinical trial design as a concept someone told me it was published in like the 70s. So this is not new stuff. Yet we're trying to promote it. And C3TI thought about this for a long time before we ultimately launched, and we put together sort of what we call a logic model or a program theory. And this will help us evaluate it down the road, maybe a year or 2 years from now, to see, did we make a difference with any of these new activities that C3TI launched with? So you can see just on the screen, I'm going to just highlight a couple of them. Initially, what we're primarily going to be looking at are short-term outcomes, such as, are folks finding the compass useful? Do they actually go in and search it? Can they find what they were looking for? If they go to our website, if they send an email or ask a question to get connected to a new program or to a point of contact within CDER, are we able to respond to those? So we track things like how long do folks stay on the C3TI website, on the compass. We are triaging and tracking all of the triaging on our inquiries to our mailbox. So this allows us to kind of evaluate over time if we're making progress, if we're doing the things we least said we would do. As you move a little bit further down the road, hopefully we'd like to see more adoption of these innovative approaches in clinical trials. So probably C3TI might redo that analysis that Dr. Tanhai mentioned this morning, where you went in and looked at the trials that were sort of published over a period of time and see, did they do more selective safety data collection? Hopefully we would like to see that they did. And maybe point that back to C3TI. I'll probably just shower all the praises to Mary Tanhai and all of her presentations for promoting that. But then, of course, in the long term, as clinical trials do use the most innovative approaches and improve over time, that should ultimately improve outcomes for patients. Like, at the end of the day, we want to be able to have drug development that's as efficient, as effective as possible, because that helps us get safe and effective medicines to patients. So with that, I think we can go ahead and turn it over to questions. And of course, if you have any questions that come to mind afterwards or something maybe a little bit more private, you can connect with me personally, or you can reach out to C3TI via the information on the screen.
Thank you, Dr. Bugan. So again, we encourage you to come up to the mics for in-house. We like to see some participation in real life. And then we'll alternate as fairly as I could. We'll alternate, and then we'll do it on online. So let's start with the first question here in-house.
Thank you for an easy way to remember the acronym, C3PO and C3TI. So the question is, you said that you're going to be a facilitator between the sponsor and the review division and help things faster, and then we can help innovation by making things be publicly disclosed. Operationally, right, meetings with our division, it's still going to be the same, or will we get separate meetings with C3TI? And what's the timeline for reviewing things that we want information? Thank you.
Yeah, great question. So I wouldn't have a lot of friends if I said we'd have to do more meetings or do the meetings faster internally. I'd probably have a lot of friends externally. But we also just didn't want to short circuit the regulatory review process. That's a lot of R's. What C3TI hopefully will be able to do, because we've identified a trial in advance of any of those meetings to discuss the trial as a demonstration, we'll know it's coming. We'll kind of have some advanced warning, and the review division will know it's coming too, because the proposal is going to go directly into their IND, and they'll actually be the first persons to tell C3TI, yes, that does look like it would be a good fit for the demonstration program. And then C3TI sort of affirms and acknowledges that. And then from that point going forward, when you request a meeting to discuss that trial, and FDA does meetings all the time with sponsors to discuss the protocol. So if you want to come in early, even if you've got like a 2-page synopsis of your trial, most likely FDA will grant that meeting and try to give you feedback, if there's enough in there to give you feedback on, to help you ensure you have the most effective and efficient design possible. So what C3TI again will do is sort of work in the space of that timeline to make sure all the relevant subject matter experts can be pulled in. So you get like a little bit of an extra support from C3TI in the coordinating function. It won't necessarily, again, short circuit the review team. They're going to still be there. It just hopefully brings in those extra experts. The other benefit is we'll be tracking that entire interaction. So, it takes, many years for a trial to be sort of designed and then launched and then the data to come in and then, finally be submitted and so on. So C3TI will hopefully have sort of a record alongside of the regular records that are already going to be in place to help ensure that all the subject matter experts are aware and are facilitating throughout the way. Secondly, I think there should be a higher level expectation when we say, trial X with sponsor Y is part of the demonstration. Like that kind of should give you a little bit of a sense that FDA is watching this very closely and really does want at least the implementation of the innovative approach to be successful, because we know this is going to be looked at as a possible case study, as an example of how well to do this. Again, can't guarantee a drug's going to work or that a trial will be successful. There's just so many different factors at play, but at least the way we're implementing the innovative approach will be.
I just love the passion of our speakers. You can tell they are fully immersed in what they're doing, and their sole objective is to help you guys. Thank you. Next question in-house, please.
Hi, so understandably, a lot of these examples focus on later stage trials. I'm wondering if you're envisioning an expansion of focus areas that could be applied in like proof of concept or some earlier stage trials.
Yeah, it's a great question. So since we launched, and even maybe before we launched, there have been other proposals internally for possible demonstration project areas, coming out of some of the other existing programs. I'll use the Model-Informed Drug Development or the Quantitative Medicine Center of Excellence that was mentioned earlier this morning. So in that space, there may be opportunities for something a little bit earlier or more upstream in drug development where C3TI could partner to help demonstrate. I wasn't aware of this, but our commissioner said it this morning, and he's the boss. So I guess maybe AI could also be a place where C3TI could potentially partner. I know there's going to be a speaker later today talking about some of our efforts to help put out some guidance around artificial intelligence and machine learning and drug development. And so as we get that guidance out, there might be opportunities to potentially partner with trial sponsors for demonstrations there. I think a lot more needs to be sorted out, but yes, I think that has been explored.
Thank you.
See, we learn as well. So let's go online for a question, please.
To what extent can C3TI assist with Pediatric Research Equity Act or PREA required pediatric studies? Will C3TI consult with the Pediatric Review Committee and vice versa?
Great question. So I don't think C3TI would necessarily get involved in working through a pediatric study plan or a trial intended to fulfill a pediatric study plan requirement under PREA, unless they were looking to explore one of these innovative approaches in the demonstration. Or maybe another innovative approach. And let's say, again, there was maybe a digital health technology or a wearable that might enable the trial to be more effectively conducted in pediatric patients. We might be able to help facilitate getting connections with the relevant subject matter experts within the center to help them with that process. But again, we're probably not going to play any sort of a direct facilitator role with the regular processes there.
Thank you. If you have questions in-house, come please take advantage. This is your time. Question, please.
Hi, thank you, Kevin. My name is Jess Ross. I'm from Unlearn AI. And speaking about AI and speaking about, these technologies that are coming into development, I think that you have a real opportunity at C3TI to incorporate these into clinical trials at a larger scale. I know particularly in diseases like Alzheimer's disease, there are many, many trials that are coming out, and then the patient pool is going to get smaller and smaller. So I think I'm wondering about, it seems like many of the programs are geared towards sponsors, and there are other opportunities and companies that are developing technologies that could be broadly applied across trials. And especially knowing that trials take so long to happen, and if people want to see an example of a successful trial or FDA acceptance of a certain technology, it could be a decade away. So I wanted to see if what your thoughts are on the opportunity here with C3TI and being able to incorporate some of these technologies that could be broadly advantageous to society.
No, it's a great question. I don't think it's initially anything C3TI will be able to cover, but certainly there is a lot of innovation occurring in the clinical research, CRO space or other service provider space that's helping to enable clinical trials. For example, the use of AI to maybe enrich enrollment. I mean, certain pieces of that we'll probably see in the context of the drug development program, but many pieces, like if let's say a CRO or a site is using Gen AI to help with informed consent, we might not see all of the detail. We would see the source of the actual informed consent document, the probably very 35-page plus long one that's for the lawyers most likely, but we wouldn't necessarily see the versions that were created with the additional Gen AI, maybe for translations, or if they were to say create a chatbot or something to help patients understand the informed consent document, just because that's not typically something that we're reviewing on the upfront side when the protocols are being designed. But I think it's a really important question, and it is something that we, FDA probably needs to think about how we can help disseminate some of those learnings more broadly, because otherwise I think potentially those become like competitive advantages for some of that industry that is helping to promote clinical research, and not something that could be shared more broadly.
Thank you. Next question in-house, please.
So thank you, Kevin. I have a question about you talk about the Bayesian software. So is it so do we upload the data and run the data analysis there, or can we download the algorithm and run on in Python or an R software, or how did it work? Can you elaborate about this?
If I understand the question, or maybe also let me just also demystify. So FDA is pretty unique in that we will reconduct the analyses pretty much ourselves. Even though we do have the summary data, we'll go and we want the source data and we want the analysis programs and we want to reconduct those internally. So when you submit to the FDA, you'll have to submit that data. And I think we also require the programs as well.
I see. Thank you.
Yeah.
Okay, thank you. Question online, please.
What advances do you or do you not see occurring with informed consent to ensure that decentralized trials are diverse and inclusive of participants with different levels of access to real-time electronic document receipt and transmission?
Wow, great question. This is the one downside of being the clinical trial innovation guy. You get like all the questions. But, I guess I'll just share my personal perspective on this. And I think maybe you can raise this later with the speaker on AI from CDER, Tala Fakuri, because I think AI could potentially be very useful in the space of informed consent. I mentioned earlier that we have to have the informed consent document. There are a lot of required aspects to it that we simply can't change. But once you have that required set of data elements and information within the informed consent, you can then take it further. You can figure out what are the best ways to say translate it, maybe put it in different education level ranges. And AI is very good about doing this. It still requires a lot of verification, and you have to trust, go through and make sure you can trust what's been created, but it can help accelerate that process. And then I think something else that's been published in literature has been the use of maybe a chat bot. So you can almost have the patients have a follow-up conversation with a bot that's pretty much all based in the informed consent document. So I think those types of tools will potentially help to improve the comprehension and understanding of the informed consent process. But I think at the end of the day, the basic requirements are still always going to be there.
Thank you. Nobody's up at the mics. Next question online, please.
Regarding Compass, do sponsors have a say on what becomes open to the public link before data, which used to be confidential, is released?
Oh, this is a great question. If I could have
All your questions
yeah.
are great.
Yeah, no, and I almost should have asked this one myself, because it gives me an opportunity to say, yes, so sponsors, particularly if there's going to be any information added to the Compass that's coming out of the demonstration programs, they will have the opportunity to say what is sort of off limits for disclosure during the proposal process. So if you look on our website where you have information around what needs to be sent into their IND to be considered for a demonstration, they have to outline, in addition to some of the other important eligibility criteria that are listed, they'll have to outline sort of what, again, is off limit for disclosure. Otherwise, we'll consider sort of the other top line information to be possibly available for disclosure. Beyond the demonstration program, the FDA is, as always, going to protect commercial confidential information. There won't be anything shared that's not already in the public domain. The good news is there's quite a bit nowadays in the public domain. Most studies that are being conducted under IND, they have to report to ClinicalTrials.gov. So a lot of the high-level study data elements are there, such as the primary objectives, the population, et cetera. So, I mean, there's a lot already. And if we know that a sponsor is implementing something innovative in one of these trials, assuming the sponsors agrees with that, that's something that we could simply just point to and say, hey, that's a trial. And then maybe the additional information could be gathered that way.
Are you good for two more?
Yeah, sure. I don't know when lunch is.
Online, please.
Okay, another question from someone trying to access Compass. Do you need permission to access this?
No, so the external version of Compass is available. You don't need permission. It's on the FDA website right now.
That was a quick one. Last question then for Dr. Pugan, online, please.
How can innovative study designs such as adaptive trials or decentralized trials affect efficiency and effectiveness of drug development and regulatory decision making?
Great question. So I think as noted in maybe some of the demonstration programs, so I'll use sort of the streamlined trials embedded into practice example. These different approaches can make the trial, I would say, more optimized, so we don't have unnecessary burden on either the sites or on the patients to participate in these clinical trials. And when you sort of decrease that burden on what the patients have to do, maybe how far they have to travel, how many visits they have to come in person for, suddenly you make the trials more accessible. And as we make trials more accessible, the number one reason trials take so long is like the enrollment. It takes a long time. You probably screen fail way more patients on an order of magnitude than the ones you accept into the trial. And so as you increase the capture and you have more patients potentially participating, you're hopefully going to have more enrolling and you're going to hit those enrollment targets a lot more quickly. And secondly, I think it also helps sites and the ability of sites to get activated more rapidly because there may not be as many stringent requirements. Now, I say that with a grain of salt because new things, innovative things can sometimes actually add more burden initially as we get more experience with them. So sometimes you might be doing the traditional way plus the innovative way the first time you do something at a particular site. But over time, it will build that sort of muscle memory and experience, and it hopefully will lead to more efficient clinical trials on the operational piece too.
Thank you. I see some people taking notes. That means we're probably going to have a quiz later on. Thank you very much, Dr. Bugan. So both Dr. Tanhai and Dr. Bugan will be hanging around for a little bit. If you have those questions that you want to have on a more individual basis, we will break now for lunch. I hope you pre-ordered your lunch. Hopefully, if you did not, the lines are not too bad. And we will reconvene here at 1:05 p.m. Thank you. I have a couple of gift bags for you guys. I have a couple of gift bags for you guys, so don't leave. Okay, good afternoon, everyone, and welcome back from lunch. How was lunch? Did everybody have something to eat? Good? Did the pre-ordering work? Okay, excellent. Okay, so now we're going to do something I know everyone is, waiting for. Every session is great, right? And with the theme of innovation, this, fits right in. So now we're going to talk about artificial intelligence in drug development. This presentation will highlight FDA's experience with submissions that have AI components. CDER has seen a 2 to 3-fold yearly increase in the number of drug and biologic application submissions using AI, with over 100 submissions reported in 2021. And to talk to us about AI in drug development is Dr. Tala Fakuri, who is the Associate Director for Policy Analysis within CDER's Office of Medical Policy. So please let us welcome Dr. Fakuri.
Hello. Thank you for the introduction, Brenda, and thank you for having me here. As Brenda said, my name is Tala Fakuri. I'll be talking about AI use in drug development. You may have heard about the AI work that's happening on the devices side of the equation, which is really exciting. Our colleagues at CDRH have really set the stage for policy development in that area, but today we're going to focus specifically on drugs. So there are a few take-home messages that I hope you'll be able to take from today's presentation. I'm going to go through these learning objectives. The first thing that we're going to do, we're going to talk about AI use across different stages of drug development. So we'll go through some examples in discovery, non-clinical, clinical, and so on. Then we're going to talk about FDA's experience with AI in regulatory submissions. You'll also hear more about that in one of the sessions tomorrow. After that, we'll talk about some policy milestones when it comes to AI in drug development. So we'll talk about what CDER has been doing to kind of promote the responsible use of these technologies. And after that, we'll talk specifically about the work that we're doing to address the executive order that was signed by President Biden on October 30th last year, which has specific mandates as it relates to AI in medical products, specifically for drugs. Then we'll talk about some immediate and long-term next steps or future directions for this work. You're probably all familiar with AI use. You probably use ChatGPT or some form of a generative AI in your daily life or in your work. But AI has also been used in medical research for many years now. And this is really just meant to illustrate this increase in the use of AI in medical research generally. On this slide it shows PubMed search results. If you just go in and type mesh terms for AI, generative AI, large language models, and you look at the number of publications from 2016 until 2022, you could expand it to whatever year you want, you'll see this really rapid growth in the number of publications in medical research using AI. And there are many reasons behind this phenomenon. We're not going to discuss that today, but briefly, a part of the reason is the availability of large data sets. So generally, AI needs large data. So you could think there's a lot of EHRs, claims data, data from registries, data from clinical trials, multimodal data, right? Data from wearables or genomics data. Proteomics data, data from imaging. The availability of these large amounts of data, different types of data set, really enabled that technology. Importantly, increases in computational power has really pushed the field forward, and advancements in methods. So if you think about methods, you could think about more traditional machine learning methods, and then on the other end, you have generative AI or large language models. This advancement in sophisticated methods has really accelerated the adoption of these tools in medical research. Now, we see what's happening in the outside world also reflected specifically for drug development. On this slide is some examples of AI uses across different stages of drug development, starting with discovery, moving to non-clinical research, clinical research, and pharmaceutical manufacturing and post-market safety surveillance. So when you think about discovery, AI use in discovery, it could be that AI is being used to identify a drug target, to select a drug target, identify protein folding, identify ideal or optimal targets to be tested in a clinical trial. There's so much work happening in that area, and it's really accelerating a lot of these upstream steps before you even enter into a clinical trial. In fact, during my graduate work, some graduate students would spend 6 to 8 years identifying how a protein folds, right? That would be their PhD dissertation. Now you can use some tools that may be able to do that for you in a year and a half. This is how fast this technology can move things forward. A lot of that work that happens in discovery is usually outside of FDA's oversight. We know it happens. Sponsors tell us about it because they're very interested in the use of the technology, but it's not something that we would be looking at carefully. After these drugs are identified, or targets for a therapeutic is identified, then it's going to enter through the traditional non-clinical research, identify, PK, PD, toxicology studies, dose range finding. That's also where we see applications of AI. And then the largest proportion from what FDA sees on our end is in clinical research. So we get asked, can you tell me more about how AI is being used in clinical trials? It could be things like identifying sites. So you might have a lot of data to identify what are the sites that are ideal for my clinical trial. Then you could also use it to identify the optimal or ideal patients to be recruited into your trial. You could also enhance adherence in a trial using machine learning or AI tools. Of course, for data collection, what that means, it could be a wearable, right? A lot of wearables, a lot of digital health technology tools are AI enabled, so they'll be able to process large amounts of information, collect information, and provide kind of a digital biomarker for a clinical trial. Of course, there's also analysis of RWD, or creation of EHR phenotypes from real-world data, from RWD. That's another use case. And as I mentioned, in clinical endpoint assessment. Additionally, AI can be used in these later phases of the drug development process in manufacturing, right? Advanced manufacturing actually is a prime target for AI because this is where a lot of automation can happen. So we know that machine learning is being used a lot in that area. And then, of course, for post-market safety surveillance, so AI can be used to detect or evaluate or process signals as it relates to adverse events. And that's one of the applications of AI. So this is kind of the spectrum of where this tool is being used, which makes it really interesting to be able to provide general frameworks that could apply across all of these different stages. That's why we always emphasize that your evaluation, our evaluation of these methods have to be really anchored in the context of use. How are you using AI? What is the scope and role of your model across the phases of drug development? And then, it also has to be risk-based or risk-informed, as I'll discuss in other slides. So in 2023, our colleagues in the Office of Clinical Pharmacology published a landscape analysis, a paper that described FDA's experience with submissions that include AI and machine learning components. And I want to emphasize this is what we receive in-house. This is not necessarily reflective of what's out there because we know that the uses are wider outside of regulatory submissions. The paper, as I said, described the uses across different parameters. On the top of this slide, you're going to see the submission that included AI ML by submission type. So whether it was an IND, an NDA, a submission to the Drug Development Tools Program, for example, or a CPIM meeting submission. From 2016 to 2021, we're updating these numbers, but the paper was published last year with the range of 2016 to 2021. On the bottom of that table, you'll see the submission by the drug development stage. So discovery, preclinical or non-clinical, clinical research, and post-market safety monitoring. What you'll see is kind of representative of that first image that I showed you, where you see this kind of exponential growth. In 2016, we had 1 IND with an AI ML component. In 2021, we had 128 submissions with AI ML components. So the number of submissions and the complexity of the submissions is increasing. When you look by drug development stage, most of what we get is in clinical research. But again, I want to acknowledge that a lot of what's out there is actually in discovery. But these are submissions that come into the agency, whether it's in an IND or a meeting request. We also looked in that paper at the submissions by therapeutic area. So here is kind of a distribution of different therapeutic areas where these types of tools are being used. And the largest proportion is in oncology, psychiatry, gastroenterology, and so on. But it really traverses multiple different therapeutic areas. So it's not restricted to just one thing. Now, we get asked a lot this question about how will FDA regulate AI? I just want to clarify that really our role is to ensure, and Dr. Cavazzoni mentioned this in the morning, is to advance the responsible adoption of these emerging technologies to accelerate the development of safe and effective drugs that can really help patients that need these therapies. So you can kind of think about this technology, artificial intelligence and medicine on one side, and advancing regulatory science, tipping the scale towards patient benefit. So we want to promote the responsible use of this technology to make sure that it is being used robustly in a way that we can rely on it to give us the evidence needed to approve a specific drug. Now, in the past several years, we've been working on AI, and I actually now wish I had another slide that shows the CDRH AI policy milestone. So that's for devices, but because we work very closely with our devices colleagues, they've kind of set the stage for a lot of regulation, not just in the US or regulatory policy, not the US, but globally, as it relates to AI in medical products. But we started this work in 2019. So in 2019, CDER established the AI Steering Committee. And the role of the steering committee was really to try to come up with a strategy, an internal strategy for AI use across the center, to have a unified voice in explaining the work that we're doing, and kind of developing our thinking over the next few years as it relates to AI in regulatory policy. A part of the AI subcommittee was the AI framework work group. And that group was specifically tasked with developing a regulatory framework, a regulatory thinking around AI in drug development. And that's the main focus of that group, was actually to write discussion papers that ended up being published last year in May of 2023, to solicit feedback and to learn from the outside world, not just sponsors, biotech, academia. A lot of this work is actually done by academia, by biotech, by smaller types of companies rather than large pharmaceutical companies. So I wanted to engage with external parties on how they're using that technology and on specific questions that we raised in the discussion paper that I'll show you briefly. So 2019, we developed these groups. At the end of 2022, we had an expert workshop with Duke Margolis, where we again invited speakers from academia, biotech, industry, other regulators, because we know that harmonization globally is a very important point. So we had a discussion around several topics, including governance, accountability of the use of AI models, issues related to data quality, So these models are just as good as the data used to train them. So we wanted to talk about the availability of data. Is there good data out there in the healthcare setting to help promote the use of that technology for drug development? And then we also wanted to ask about model performance. What are people doing to assess the performance of these models in their drug development programs or in their research? The Duke Margolis workshop and then the discussion paper, we published actually two. One that looked at drug development overall in all phases: discovery, non-clinical, clinical, post-market, manufacturing. Another discussion paper was just focused on manufacturing because the uses there are a little different, and with some unique challenges and considerations. Happy to discuss this further later. These two discussion papers, the expert workshop, and then our experience with regulatory submissions that I just showed you, kind of helped us set the stage for what's coming next, right? So now we've gained kind of this experience, and we got this information from external parties. We can now start developing a robust regulatory framework moving forward to advance the responsible use of that technology. And then the executive order came out. So it's perfect timing. The executive order came out, like I said, in October. I don't know if I have a pointer here. I think I do. Okay, great. So at the end of last year, and in it, there were specific requirements for CDER, for drugs. And we are currently this year responding to that. We were actually given 365 days from the time the executive order was signed to push out certain deliverables. I'll mention briefly what we've been doing to address the executive order, but primarily we published a white paper recently in March of this year. I'll talk about that briefly, that describes how the medical product centers are working together to advance the responsible use of this technology. And then we have a city workshop, so Clinical Trial Transformation Initiative workshop. If you actually Google City, FDA, AI, and drug development, it will be August 6th. It's going to be a hybrid workshop, so there are limited seats available for you to attend in person, but you could also join online. I encourage you to sign up. In the workshop, we'll have experts speak using use cases. So we want it to be practical and helpful. These are the specific unique challenges that we face with AI, and here are some proposed ideas or solutions. That will be the focus of that workshop in August. And then this year, we also hope to publish our first AI guidance for drug development. And I'll walk through some of these milestones in the next few slides. Okay. So Executive Order 14110 on safe, secure, and trustworthy development of AI. The executive order really launches a government-wide effort to guide responsible AI development and deployment across the federal government. And what's really great about this is that it's not demanding that the government's approach to AI is horizontal, meaning it's not going to be the same across all industries. Instead, each agency will develop its own thinking and regulatory frameworks. It's more vertical, which is great. It includes 150 requirements, and these requirements range from developing guidelines and frameworks to conducting studies, creating task forces, issuing recommendations, and even engaging in rulemaking. So a wide range of different requirements for all agencies within the federal government. Then there's specific language for drugs and devices. We work very closely with our colleagues at CDRH, with CDER, of course, and then the Office of Combination Products to make sure that our approach is consistent where possible. And I'll get into more detail on that shortly. Like I said, for drugs, we have 365 days to be able to develop our thinking, specifically as it relates to, this is language directly from the EO, define the objectives, goals, and high-level principles required for appropriate regulation throughout each phase of drug development. So the first thing that we did is we kind of wanted to explain on a high level what is the work that we are doing, all of the medical product centers, as it relates to AI. And this is very, very important. We actually get feedback on that. Like I'll show you in the next few slides too, based on feedback we received on the discussion paper. People want to make sure that we are consistent where possible with how our devices colleagues are handling AI in devices, right? The approaches may be different in regulation between the two centers, but generally you could think about principles for good machine learning practice, just general principles should pretty much be the same regardless of what the medical product is. So we wanted to be able to articulate how the medical product centers are working together, and we published this paper with 4 areas of focus. In the first section of the paper, we talk about how we intend to continue to foster collaboration to safeguard public health. Then in the second section of the paper, we talk about our plans to advance the development of regulatory approaches. In it, we announce the AI guidance that's coming out for drugs, but also there are 2 guidances from CDRH that will be published this year. One is draft to final guidance on predetermined change control plans. It's really great guidance. I encourage you all to read it. And then there's another guidance that will be coming out from CDRH on life cycle maintenance of AI models. And then, of course, there's the CDER specific guidance that we're publishing this year. We also talk about our efforts to develop harmonized standards. In fact, if you go online and type standards for AI, you will see a million different documents out there. And the question is, which standards do I follow? Do we which specific standards should I accept and adopt for my organization? So we're working on that to try to identify some sort of harmonized standards that we could all follow for medical products development. Then, of course, best practices and tools and so on. And lastly, support research related to the evaluation and monitoring of AI performance. And this is so important for specific use cases. I'll mention manufacturing again. The strength of these tools is they learn, they evolve, they adapt. Whereas generally regulations for drugs are based on a fixed point in time. So there are certain applications where this continuous evolution and training of these models becomes very important, and we need to develop methods to be able to monitor their performance after they've been deployed. Again, this is of critical importance for devices, not so much for drugs, but depends on, again, the context of use. There may be examples where the use in drug development would require this continuous monitoring. You could think about post-market safety surveillance or manufacturing as some examples. Okay, the second deliverable that we're working on is guidance that will be coming out this year. Of course, it's right now deliberative, pre-decisional. I can't share with you what's in the guidance, but I can give you just very high level that this guidance is in collaboration with all of the medical product centers. So we actually, in the writing of this guidance, we have colleagues from devices, from biologics, CDER devices, Oncology Center of Excellence, Office of Combination Products. We're all working on this together. It is informed by the experience of over 300 submissions to date with AI ML components, on over 800 comments that we received on the 2 discussion papers that were published last year, and on the best available or current regulatory science research. We understand that this is an evolving area. So this is going to come out as a draft guidance. We really want and need your feedback. This is how we can make sure that when that draft eventually goes to final, it is really informed by the community's feedback and experience, and thinking through the applicability of this guidance to the work that you all may be doing. So I wanted to end the next few slides with some of the comments that we received on the discussion paper, just to kind of forecast what we hope to accomplish in the future, but also to kind of summarize the feedback that we received for all of you, to see if your questions, your concerns are reflected in some of the comments that we've received. So the The discussion papers that we published last year were also written by a collaboration between the medical product centers. And we received over 800 comments from 65 different organizations. When we issued the guidance, it included 3 sections where we solicited feedback, one on human-led governance, accountability, and transparency. What does transparency mean for AI? What is governance? Who's responsible for monitoring the performance of the model after it's being deployed? The second set of questions were around the quality and the reliability and relevance of data used to train and develop these models. And finally, we ended with a set of questions related to model development and performance. And we got a lot of feedback that can kind of be summarized into these themes. The first questions that we got on the discussion paper were, FDA says that that AI is being used across the spectrum of drug development. What is within FDA's scope of regulation and outside of scope, right? So you could think about, again, discovery. You're developing an AI tool to predict protein folding. Will FDA regulate that? If I'm a sponsor and I'm using AI to write the first draft of a regulatory submission, is this under FDA's oversight? These are the type of questions that we actually got on the discussion paper. Then we got asked about, what do you mean by risk-based for AI? Can you actually tell us how you would operationalize a risk-based approach for AI? We got questions on transparency. What is the level of detail that will be required in terms of documentation that should be submitted to the agency? There were a lot of calls for harmonization, which is very interesting because we don't have currently global, guidance or regulation. So we need to start by developing some guidelines and guidances before we move to harmonization. But I think it's this expression perhaps of a little bit of anxiety about what's coming next. So we're keeping this in mind. And then, of course, a lot of comments about alignment with AI guidelines that were published by our colleagues at CDRH. And finally, which I actually find very interesting, calls for the establishment of public-private partnerships to create kind of big data lakes, if you want to call it, or a place where people can put their data so that everyone can use that data to develop better models. I think it's interesting that it came to the FDA because, when sponsors submit their data to us, this is protected, it's proprietary. So we are thinking of ways to enable data sharing. This would be something that would be of great value, I think, for all AI tool developers, because in fact, a lot of the data that's out there is kind of biased towards positive trials. But for a lot of machine learning, you need negative trials. You need data that did not actually produce good results to train better models. So these are some of the themes that emerged. We're going to try to address these different questions that we received on the discussion papers and the guidance that we're publishing this year. Okay, so for the short term, we're publishing our guidance this year. We really want your feedback. That feedback will help us develop future guidance or guidances. There may be guidances that are focused on specific areas of drug development. We're also working to develop practices that can be best practices that can be shared across the board. And another important thing is this consistent terminology. So defining what different terms mean is very important in a field like AI. We want to continue, obviously, engaging with external parties. Please sign up for the FDA City Workshop if you haven't already. For the long term, we want to continue our responsive risk-based regulation. We hope that AI guidance will kind of articulate that we are open to the use of this technology in a responsible way to advance safe and effective medicines. And then we plan to continue advancing regulatory science. We hope to be able to promote demonstration projects in the future that can help with that step, and continue our collaborative approach by engaging with all players in the AI ecosystem. Okay, now I have 2 very easy poll questions. They're true or false. The first one, Evaluation of machine learning model performance requires an evaluation of the data used to develop the model because these models are data-driven. Who thinks this is true? So if you're evaluating the model, you should evaluate the data used to train the model. Awesome. I see like 60% hands up. Agree. The second question, a risk-based assessment framework, and I skipped that slide, so this is going to be hard for you, but we could do it now, means that the level of oversight, the level of the rigor for the assessment of the performance of these models or the acceptance criteria for these models will depend on the risk that is associated with using AI. Does that sound like a reasonable yes? I see a lot of nodding. Okay. I think they passed, right? Great. I'll take questions now.
Okay, we have just about 8 minutes for questions, so please come up to the mics if you have any questions in-house. And we have one brave person here. Your question, please.
So guidance documents are current FDA thinking, they're not regulation. So in terms of, from the way I'm reading your, interpreting your presentation, there are no true guardrails right now, other than as a sponsor, we would present to you what our risk-based approach is and what our strategy is. And we would kind of do a mommy may I, or are we all in agreement? How's it work?
So the guardrails for drug development will be different. I just want to put that out there, different than guardrails for AI use in other industries, right? So if you think about AI in a self-driving car, obviously that's going to be different. In drug development, we've received over 300 submissions that we've been able to engage and interact with using our current regulatory framework. Our current regulatory framework is robust and is able to handle these submissions that we've received. So we feel like we have the guardrails in place. The questions that we get asked, though, is for AI specifically, when the agency needs to evaluate a submission that comes in with AI. Are you going to review the data used to train the model? Are you going to review the code? Are you going to try to replicate the finding using? So these are the type of guardrails or questions that we get. They're really more questions about, can I trust the results from the way that you use the model? And that's where we hope our guidance can provide some answers to that question.
Fair enough. Thank you.
Sure.
Thank you. We have one other person here in-house, please.
Hi, as you're creating guidance around the use and management of AI models, are you considering some of the recent ISO publications that have just come out to sort of align AI management systems with how ISO is thinking about it?
So specifically for management systems and ISO, I mean, like I said, there are so many standards out there, right? So when we think about eventually developing a set of best practices or standards, we would take into consideration all of these different standards that are out there. For the guidance that we're developing specifically, it's kind of our articulation of our thinking around what is the risk-based regulatory framework that the FDA will take to assess the credibility of these models.
Thank you.
Thank you. Online question, please.
Can you comment on the use of AI by FDA employees, for example, reviewers? How is FDA introducing AI to reviewers to support the review of incoming applications?
That's a great question. So the question is on internal uses of AI. I actually encourage, if you're going to be at DIA this year, there's going to be a session on internal uses of AI by different regulators, including EMA, FDA, and so on. So we do have a lot of experimentation of AI tools happening internally that is meant to really increase the efficiency of the work that we are doing. We are a big organization, just like a lot of organizations out there. But the uses of AI within the agency, we are government, so the uses are different. For example, we can't just bring in ChatGPT and use it within the agency. We house a lot of proprietary data, right? So we have certain set of requirements that are set in place to kind of guardrail that type of information that we have in-house. But this is, this is going to be changing because I didn't talk about that. But in the executive order, there's also a directive to agency to report on their internal uses of these technologies and making the use of that technology public. So we will be reporting just like everyone else within the federal government on how our staff are using AI over the next few years or so.
Thank you. In-house question, please.
To preface, by all means, I'm not an AI expert. I recently got to sit in a roundtable with Brandon Gallus from FDA, and we were discussing possible neural pathways being like another variable to algorithm variability. And I was wondering if there's any initiative from FDA's end to consider those on top of the guidances that are currently in progress.
To use what? I couldn't hear you. Sorry. Can you repeat that?
So I was wondering, as AI, digital pathology specifically in drug development, as they progress, there are lots of variabilities between different algorithms by developers. And I know as pilots for these digital pathology projects progress, there will be necessity for adding more variables, such as adding a pathologist into the algorithm. And I was wondering, that being a new neural pathway, is that also being considered in terms of FDA's initiative to harmonize these algorithms?
So in that landscape paper that we published last year, we discuss where AI was used in the drug development program. And actually one of the largest uses is in imaging, and one of the largest imaging for drug development is in pathology. For any specific technique that is being used in a specific context of use, our evaluation of that technology will depend on how are you using AI, how much am I relying on AI to come up with the evidence, right? To come up with the answer for the question, if it's supportive or if you're trying to replace a traditional method that is in place. Is it something that we would consider? Of course, we would consider any use of emerging technology that is responsible and robust. I can't give you a specific answer because it would depend on exactly how the technology is being used.
Thank you.
Okay, one last question online, please.
How is the FDA integrating AI with knowledge management, and how could that potentially impact life cycle management of drug products from development to post-marketing?
How is FDA using AI in knowledge management? I would need, honestly, more information on that. I'm not sure if this person is specifically talking about like knowledge graphs, but we receive a lot of submissions where AI is used in different stages of drug development, including AI to kind of predict different outcomes, that integrates information from a trial to use for predicting outcomes. But specifically for our review, I'm not sure I understand that question. So perhaps we can go to another one.
Or some clarification on that question. It's based upon the multiple sources of information they have on drug products, and whether an AI-driven knowledge management approach is something the agency is looking forward to adopting soon.
Yeah, I mean, like I said, right now, we're experimenting with different uses of AI internally within the agency. We have to go through different type of milestones to be able to adopt any specific use because of some of the restriction around cybersecurity and our access to these tools. I don't have a specific answer to this, but obviously one of the strengths of AI is this ability to synthesize information from different sources, different modes. So I can't see why we wouldn't be doing that.
Thank you. Thank you, Dr. Fakuri. Dr. Fakuri will be staying through the break to 2:45 p.m. So if you have some questions that you were too shy to come up and ask, please take this opportunity to ask. Okay, our next topic is all about ClinicalTrials.gov. and meeting transparency and reporting requirements. This presentation will describe the ClinicalTrials.gov registration and reporting requirements for applicable clinical trials and the actions that may be taken by FDA in response to non-compliance with these requirements. Our speaker will be Dr. Laurie Muldowney, who is the Deputy Director for the Office of Scientific Investigations within CDER's Office of Compliance. Please welcome Dr. Muldowney.
All right. Well, AI is a tough act to follow. I'm afraid to say that compliance with data transparency may not be quite as exciting a topic, so I appreciate you guys hanging in there with me. Again, my name is Laurie Muldowney, and I'm with the Office of Scientific Investigations within CDER's Office of Compliance. OSI is responsible for compliance and enforcement activities for studies involving CDER-regulated drug products. And so we have responsibility for oversight of ClinicalTrials.gov registration and reporting. So I'm going to focus on CDER trials today. I know there are obviously device trials as well as trials regulated by CBER that are on ClinicalTrials.gov as well, but I'll focus on the drug trials. And you may be asking yourself, what does this have to do with innovation? And what I would say to that is, as clinical trials evolve and innovate, transparency about those trials is even more important. Registering where readily accessible to patients and healthcare providers will result in trials that are more diverse and participants that are more representative of the patient population. And then, of course, reporting information from trials, including protocols and statistical analysis plans, as well as select study results, will help others to understand what has worked and what has not worked when innovating in clinical trials. Okay. So the learning objectives today, first would be to recognize the roles of the National Library of Medicine, or NLM, FDA, and study sponsors when it comes to ClinicalTrials.gov requirements. To understand what those requirements are for clinical trial registration and reporting, and to just be able to describe the potential consequences of non-compliance. So I hope that everybody is somewhat familiar with ClinicalTrials.gov. It is, of course, a web-based resource that's managed by the National Library of Medicine at the NIH, and it provides patients, healthcare providers, and researchers with easy access to information about clinical trials. The registration is intended to help patients and healthcare providers identify potential trials, and reporting requirements ensure that those subjects' participation is honored and that the trust is enhanced by having a public record of the clinical trial. It also helps researchers to identify unmet research needs, to find collaborators, and to stay current on developments in their field based on information about studies that are both ongoing and complete. The information in ClinicalTrials.gov is provided by study sponsors as well as principal investigators, and there are close to 495,000 studies on ClinicalTrials.gov currently. That covers studies across all states as well as over 220 countries. And ClinicalTrials.gov gets about 4 million visits monthly, so it's a very heavily trafficked site. So a little bit of history on ClinicalTrials.gov. 1997, the FDA Modernization Act required NIH to establish a registry for clinical trials. So this resulted in the launch of ClinicalTrials.gov in 2000. At that time, it was really just registration, so just registering when you were going to be starting a trial, and it was largely voluntary. The only requirements really at that time were for studies that were conducted under IND for serious or life-threatening diseases. Otherwise, it was entirely voluntary. The requirements were expanded in 2007 with the FDA Amendments Act. So this expanded the registration requirements to go beyond those serious and life-threatening diseases, and it added reporting requirements. As a result, in 2008, NLM launched their results database where you can now find select results information on clinical trials. In 2017, so things always take a little bit longer, 2017, the final rule was issued that clarified and expanded these registration and results submission requirements from FDA. And all of this can be found in 42 CFR Part 11. So you'll see that referenced on a number of slides here that really includes all of the important information regarding ClinicalTrials.gov requirements. So while the website has been around for about 25 years, really the requirements for registering and reporting have only been in the regulations for about 7 years, and government time, that's not that long. So I mentioned that only certain trials are still required to register and report. And the focus of the remainder of the talk is going to be on that subset. That's the subset that FDA has compliance and enforcement oversight responsibilities for. And we call those applicable clinical trials. So an applicable clinical trial is defined as a controlled clinical investigation other than phase 1 of any FDA-regulated drug or biologic product for any disease or condition. And the final rule considers all interventional trials that have one or more arms and that have one or more pre-specified outcomes to be controlled. So while we might not consider like an open label, single arm safety study to be a controlled clinical trial, if there are pre-specified outcome measures associated with it, then for the purposes of ClinicalTrials.gov, it is considered to be a controlled clinical trial. In addition, an ACT must study an FDA-regulated product. So if the study were conducted entirely outside of the United States, it would only be considered an ACT if either the drug is manufactured in the United States or the study is conducted under IND. I mentioned that ClinicalTrials.gov has close to 500,000 studies, and actually a minority of those studies are applicable clinical trials. So there are a lot of observational studies, behavioral interventional studies, phase 1 studies, and others that voluntarily register and report results information, but don't have those same requirements. So the responsibilities for ClinicalTrials.gov really fall into 3 buckets, which is a little bit different. We sort of split some of the responsibilities with NIH. NIH and NLM have implementation responsibilities for the website. The responsible party, which I'm going to talk about in a little more detail in a few minutes, is responsible for actually submitting and updating that registration and results information. And then the FDA, of course, is responsible for compliance and enforcement activities. So just a little bit more on the role of NLM. Again, they're responsible for the development, operations, and maintenance of the website. They actually did update the website, I think it was about a year ago, modernized it so that it would be more mobile device friendly, included more plain language, FAQs. They did a pretty good job with it, so I think it's a little more user-friendly now than it was previously. They're also responsible for actually processing and posting those registration and clinical trial information that are submitted to NLM. And then probably for the purposes of this talk, most importantly, they're responsible for like quality control. So they do a QC check of everything that is submitted before it's going to be posted publicly on the site. So study records are actually submitted through sort of this back-end PRS system. It's the Protocol Registration and Results System. And after things are submitted, it goes through a manual review by NLM staff to look for any apparent errors, omissions, or contradictions, anything that's sort of blatantly obvious from an initial QC review. And when they do that, they'll identify issues as either major, which would be something that must be addressed, or advisory, which is really just to sort of improve the clarity of the record. There's a lot of excellent resources on clinicaltrials.gov on registration and reporting, tons of information that sort of go into that process as well. So now on to the role of the responsible party, which is pretty important. So who is the responsible party? This is a key term in 42 CFR Part 11. And the RP is the person or the entity that's directly responsible for registering and reporting complete, accurate, and up-to-date information on ClinicalTrials.gov for any applicable clinical trial. We run into a lot of questions regarding who the RP is for a specific study, so I just wanted to spend a couple of minutes on this. The simple answer is that the sponsor of the ACT is considered to be the responsible party unless and until they designate a principal investigator for that role. And each ACT can have one and only one responsible party. So if it's usually the sponsor, how are we defining sponsor? Because things are defined differently in different parts of the regs, as we know. But 42 CFR Part 11 distinguishes if the study is conducted under IND or IDE or not. If the study is conducted under an IND or an IDE, there's no ambiguity. The IND holder, the IDE holder is the sponsor. If the study is not conducted under an IND, then it's considered to be that single person or entity who initiates the study by preparing and planning the trial and has the authority and control over the trial. So again, that the sponsor can designate a principal investigator to be the responsible party, but only if they really have the ability to meet all of the requirements of ClinicalTrials.gov. So it's listed if that the PI has to be responsible for conducting the trial, they have to have control and access to all of the data, and they have to have the rights to publish the data as well. If a PI is serving as the responsible party, they're actually going to use the PRS system of the sponsor. And we consider the principal investigator having their name listed as the RP on the study record in ClinicalTrials.gov to be their acknowledgement of that role. So last slide on responsible party, I promise, but these are actual true questions that we've gotten in just the last probably couple of months. So I recently took over sponsorship of an IND, which includes several ACTs, both ongoing and complete. Am I the responsible party? I see some head shaking. That's good. We recently acquired a small company that conducted a clinical trials in an indication that we are not interested in. We're no longer going to pursue it. Am I the responsible party? And I'm thankful that I see a lot of head shaking. And yes, the answer is, we always say generally yes, because everything is a little bit fact specific. So I don't want to give like a blanket, but in most situations, the answer is going to be yes here. So when you take over an IND, you become the sponsor and therefore the responsible party for all of the studies that are conducted under that IND. Even if the studies are completed, you're responsible for making sure that those studies under your IND are in compliance with ClinicalTrials.gov requirements. So it's important to know what studies have been conducted or are being conducted under an IND before you're taking sponsorship. Similarly, for the second one, again, this is sort of fact pattern specific, but usually the acquiring company you should confirm when you're acquiring a company what studies have already been conducted and what are underway, whether those are ACTs and whether they are in compliance with ClinicalTrials.gov requirements. So the acquiring company may be held responsible if they assume that sponsor role of studies, even if those studies have already been completed. So I think the bottom line is just it's critical to know the portfolio of studies under an IND or within a company's scope before assuming sponsorship or ownership. And this includes knowing whether ACTs are in compliance. All right, so let's talk just a little bit about some of those responsibilities that the RP actually has. The first is related to registration requirements. Responsible parties are required to register their applicable clinical trials within 3 weeks or 21 calendar days of enrolling the first human subject. Again, the main purpose of registering is to provide that information to potential participants and healthcare providers about studies that they might be eligible for. I've included a list of some of the required elements here. It's not an exhaustive list, but it's pretty basic stuff. The descriptive information is going to be the study phase. What is the intervention and control? What are the eligibility criteria? Recruitment information. Is it not actively recruiting yet? Is it currently enrolling? Is the study closed? Administrative data, location and contact information is pretty obvious. And as I mentioned, I've mentioned PRS once or twice. This is the Protocol Registration and Results System. So you're going to be submitting that information to that web-based portal, and NLM is going to be doing a quality control check. And they will get back to the RP within a couple of weeks if there are any problems, and you have 2 weeks to correct any of those major issues that have been identified as requiring updates before the information is published. So reporting requirements. You're required to report clinical trial information no later than 1 year after the primary completion date. So the primary completion date, this is the standard submission deadline, and it's defined as the date that the final study participant is examined or receives an intervention for the purposes of data collection for that primary endpoint. So we often have studies that have other endpoints, safety endpoints that maybe might go longer than the primary endpoint, but for reporting requirements, it's based on that primary outcome measure. So if you do have longer term adverse events that are being evaluated that are going to be part of a safety outcome measure, you still need to report at that 1 year deadline, but you may not have you may not be able to report everything. So, but you should still always follow that primary completion deadline. There are some opportunities to delay submissions of results information. The first is really the most common, and frankly, it's the only one that I've ever really seen, and that's a certification for delayed submission. And this is for ACTs that are studying an investigational product that's never been approved for any use, or it studies a new use, and the manufacturer is the sponsor and plans to file an application to the FDA within a year. So in those circumstances, you can request a delay in submission of your results. Very importantly, that request for a delayed submission has to be submitted before you hit that 1 year mark. So we have gotten requests 1 year and 1 day, and we do not have the authority to consider them if they're beyond that deadline. So it's really important that you get that request in by that 1 year deadline. And if you are granted that certification for delay, that pushes back the results submission until 30 days after approval of the drug for the use that it was studied in the ACT. Or if the application is withdrawn and hasn't been resubmitted in 210 days, seems a little arbitrary, but at that point it would be required. And no longer, regardless of the circumstances, you have to submit your results within 2 years of that request for delay. So that's the most common. We see that pretty regularly, actually, the certifications for delay. The other two, the extension requests for good cause and waivers for requirements, they're reviewed by NIH, and I have not seen them, so they're not commonly used. So I can't really speak to sort of what some of those, the criteria would be for those delays. So as with registration, the reporting information is going to be submitted through that PRS system. And the rule requires that the information be provided in a tabular format. So there's very little text. It's really just summary data, and that's intentional. There's no discussion or conclusions, the thinking being that it's a standardized format. It will prevent or decrease the potential for bias. So this lists some of the information that you would be providing in terms of the results information on participant flow, demographics and baseline characteristics, as well as primary and secondary outcome information. You are also asked to provide the full protocol and the statistical analysis plan. And then NLM is going to review it. They have a little more time to review the results information because there's a little bit more to review. They have 30 days from the time of submission to review that information, and it often goes back to the responsible party. We see that quite a lot. If you get it back, you have 30 days to address any of those major issues, and then it goes back to NIH. We do sometimes end up in a little bit of a circle. It will come back to NIH and they might identify additional issues or something wasn't fully addressed, we give that time. So if we believe that an RP is in good faith working with NIH to submit the correct information according to the timelines, we let that go before we're sort of going to pursue anything further. And then really the last thing in terms of the responsible party requirements is updating. And this is really important. Any changes to the ClinicalTrials.gov record must be updated within 12 months. So every 12 months, you should be checking it and making updates. There are certain data elements that are considered to be a little more time sensitive that you only have 30 days. For example, expanded access information. So if there's changes in the availability of an expanded access product, of course, that's very time sensitive. So you've got 30 days to update that. If your recruitment status changes and you go from enrolling to no longer enrolling, that's the information that obviously is important to get out there. The other one that I just kind of wanted to flag here is the responsible party. So if the responsible party changes because of one of the reasons I mentioned otherwise, or the point of contact at the company has just changed, it's really important to update that in the record. We will figure out who the RP actually is, but it's important to keep that updated in the record. And that shows that you sort of acknowledge and understand your responsibilities as well. So just a few more questions, I guess. These are real questions that we've received about whether or not RPs need to submit results. So we published our study results last year, so they are already publicly available. Do we need to submit our results to ClinicalTrials.gov? See some heads shaking. We terminated our study after only 6 patients enrolled. Do we still have to submit our results to ClinicalTrials.gov? And our product was approved based on the study, so the information is in the label. Do we have to submit our study results? And I'm glad I see a lot of heads shaking. So yes, yes, yes to all of these questions. When in doubt, the answer is probably yes. And I recommend, I will say, I keep referring to clinicaltrials.gov, but it's again this sort of separation. They manage the system and the actual submissions. They will answer your questions, and there are a plethora of resources on their site. But we really have received these questions. You know, publishing results in a journal does not remove the responsibility to submit results to clinicaltrials.gov. And if an ACT is registered, if a study's an ACT, it has to be registered, and any trial information needs to be submitted, even if the study is terminated. The only exception to this would be if the study is terminated before any patients are enrolled, in which case you would change the recruitment status to withdrawn, and there would be no requirements. And there are some good FAQs about, you know, it can get a little confusing because you might have outcome measures that have 0 patients, and how to sort of answer those is very, very study specific, of course, and there's some resources available for you there. And then, of course, the last one, if the product met the definition of an ACT, registration and reporting are required even after approval. So now on to our role, FDA's compliance and enforcement activities. So our program it's incorporated into our bioresearch monitoring, or BIMO program, that hopefully many of you are familiar with. The agency-wide oversight program designed to monitor all aspects of the conduct and reporting of FDA-regulated research. And our goal in terms of clinicaltrials.gov is really voluntary compliance. We have sort of 3 ways that we are evaluating for this. We will check clinicaltrials.gov compliance on certain inspections. We review complaints that we receive, and then our largest, our biggest approach, I guess, would be through surveillance activities. So these are some of our BIMO compliance programs. And the ones specifically where we're going to look for clinicaltrials.gov related requirements are going to be the Institutional Review Board, the Sponsor and CRO CP, and the Clinical Investigator and Sponsor Investigator CP. Some of the things that we will look for in all of these, really, we're looking at informed consent documents. And I didn't get into that in my presentation, but hopefully everybody knows there's a required statement on informed consent documents. We do look at that. We look for that on inspections. On the sponsor and CRO inspection, we will also ask the ORA investigator 2, confirm whether the study that's subject to the inspection is an ACT, confirm who the RP is, and whether or not they've met those responsibilities. It's not required, it's not necessary to conduct an inspection, obviously, to confirm compliance with this program. And this was sort of one of our initial strategies. We find that the compliance is quite high through our inspectional programs. We really haven't had a lot of problems from that perspective. Our second is through complaint evaluation, and we do receive complaints about ClinicalTrials.gov, but they're really few and far between, like a handful a year. We evaluate them on a case-by-case basis, really looking at the information on ClinicalTrials.gov, as well as our internal information about the trial in our internal database, and any public information that might be available. And we have not really found a significant amount of non-compliance through complaints evaluations. Oftentimes, the studies that we get the complaints on are not ACTs. So that is not really an avenue by which we've found a lot of non-compliance. Our primary method is really through surveillance. So we utilize an analytics platform that pulls information from ClinicalTrials.gov, as well as from some of our internal systems, DARTS in particular, which is an internal system that we use, to identify potential cases of noncompliance. We then prioritize those using sort of a risk-based approach, and we will manually review and confirm information in our internal records. Most of our efforts right now are really related to failure to submit results information, and we prioritize applicable clinical trials that we believe may pose a higher risk to human subjects, or where violations are likely to have a high impact on public health. So what do I mean by that? In terms of higher risk to human subjects, is there something that's inherently potentially higher risk about the investigational product? Is it an NME? It's never been approved for anything, so we don't know much about the safety profile. Does it have an unusual or a higher risk route of administration, intrathecal or intracerebral ventricular? Is the patient population one that we would consider to be vulnerable? So is it a pediatric trial, or is it a trial in pregnant women? And then in terms of high impact of public health, we do prioritize those serious or life-threatening diseases, as well as rare diseases, where we feel like getting that information out and having that out in a transparent way is even more critical. We'll also prioritize responsible parties that have a pattern of previous noncompliance. So if they've received a prior pre-notice of noncompliance, or if that noncompliance is in conjunction with some other type of good clinical practice violation, which would typically be found through inspections. So consequences of noncompliance, and I'll run through the first few in just a couple of minutes, but this is sort of the different choices that we have. We start with a preliminary notice to a notice, and then the last three are very infrequent. So if we identify potential noncompliance that warrants further action, then we'll issue what's called a preliminary notice of noncompliance letter. This is going to identify what that potential violation was and provide an opportunity to come into voluntary compliance. So we in that letter, it will say you have 30 days, and after 30 days, 30 calendar days, that we will begin to reassess that noncompliance to determine whether it has been rectified or not. I mentioned again that after that the RP submits the information to NIH, we can sometimes go through some cycles, and so that can sort of slow down the process a little bit, but we will begin reevaluating at that 30-day mark. And just recently, we started posting these on FDA.gov. So previously, pre-notices were not publicly available except through FOIA. In December, we started posting them. CDER's issued 130-something pre-notices in the last several years, and those can all be found here. And the goal of posting these pre-notices is really in the hopes that increasing transparency is going to encourage more general compliance across the board. So if a responsible party fails to come into compliance in that 30-day time period, then our next step would be to reassess and to issue what's called a notice of noncompliance, which looks a lot like a pre-notice, except it's a little more serious. So it's sort of more of an official declaration. A pre-notice is it appears that you may be in noncompliance, whereas a notice of noncompliance is sort of that next step. We've taken a little more time to more thoroughly evaluate. Very similar to the pre-notice process, it gives that responsible party 30 days to come into compliance. And after that 30-day period, we will reassess and make a determination. Again, goes through the same process with NIH when the information is submitted, and we will kind of give it that time. Our pre-notices have been very effective to date. So I mentioned we've issued 130 something in the last several years. We've only issued 5 notices of noncompliance. So in general, those pre-notices have been very effective. And this slide shows the website where those notices of noncompliance go. Again, it's on FDA.gov, as well as where any civil money penalty actions would go if we were to assess CMPs. And this includes a link to the letter and some other information. Our follow-up response letter will go there as well. If an RP fails to submit the information following a notice within 30 days, and or if we determine that they have submitted false or misleading information, then we're authorized to seek civil money penalties. To date, both the pre-notices and the notices have been very effective. So I mentioned the pre-notices 130 something, 5 notices. We have not had to seek civil money penalties to date. The statutory maximum for the CMPs are stated as not more than $10,000 for all violations adjudicated in a single proceeding. This is, of course, adjusted for inflation. I think it's like 14,000 or so right now. And it's actually can be levied each day. So at that 30-day point, if the noncompliance continues, then each day you can sort of assess that amount. I will say we're often asked, why don't you seek more CMPs? You could be making a lot of money. That's like a common question that we get. And I will say, first of all, our interest is in promoting voluntary compliance. And CMPs are not a trivial matter. You know, it's very resource and time intensive. It requires litigation in front of an administrative law judge and significant resources to prosecute. So we're actually quite happy that the pre-notices and the notices are working and that we really haven't had to go down this pathway yet. The other things I think I mentioned is like criminal prosecution and injunctions, I think, and we have not used those enforcement actions to date. All right, so I have my challenge questions. The first challenge question, when are results required to be submitted for an applicable clinical trial? There is no requirement to submit results information for ACTs. You guys can raise your hand if you think it's the right answer. I hope I don't see any hands raised on that one. As soon as the last patient completes the study, within 6 months of the primary completion date, or no later than 1 year after the primary completion date, unless qualifying for an exception. And I see some hands and some head nods. That is correct. So 1 year, unless the exceptions that I had mentioned previously have been submitted. All right, and the second challenge question, which of the following statements is not true? ACTs are the subset of clinical studies that are required by regulation to register and report certain clinical trial information. The responsible party is required to register and report results information for ACTs. All updates to the record in ClinicalTrials.gov must be made every 12 months. I see some questionable looks there. And the IND holder is the responsible party for studies conducted under IND. Anybody think it's A? B, C. Awesome. And D, it was a little bit of a trick question because I suppose the IND holder could have designated a principal investigator to serve as the responsible party. So it's a little bit of a trick question, but yes, you guys are all correct. That is exactly what I was thinking. Every 12 months is at a minimum. Those time-sensitive pieces of information, it's 30 days, 30 days. And please make sure that you're updating that record. So in summary, clinical trial transparency is very important for multiple reasons. Innovation and encouraging innovation being among those. NLM, FDA, and industry each have important responsibilities related to ClinicalTrials.gov. And finally, the sponsor or designee serving as the responsible party must ensure that applicable clinical trials are in compliance. And so I think that's it. With that, I will close. I appreciate you guys hanging with me there. Kind of a compliance heavy talk.
Do you think they passed, Dr. Muldowney?
What's that?
Do you think they passed?
Oh, absolutely. A+. Nice job.
All right. Any in-house questions, please?
I had a question.
Sure.
In my organization, I don't do this, so I'm sure our organization is familiar, but it's a curiosity question. Applicable clinical trial. What about drug-drug interaction, ClinPharm, QTC studies? I mean, the whole premise is transparency, making sure people know about these trials, but then there is obligation for reporting results and everything. So the cancer.
Yeah, so most of the time, I mean, you said a few different types, but ClinPharm studies are generally not going to be, unless it's a phase 1 study, you know, those are typically going to be phase 2, right? And as long as, if it's an intervention, if there's outcome measures that are pre-specified, then it's going to be an ACT. So it's more often than not, yeah.
Anyone else in-house? Okay, online, please.
If there is a long-term, multi-year study with interim analyses that interim data are used for regulatory filing, is there obligation to post interim results to ClinicalTrials.gov, or do we only need to post the final data after the study is completed?
Right, so it would be based on the primary completion date. And you know, you're going to be submitting the statistical analysis plan as well as the full protocol, other information along with that. So everything's really centered around that primary completion date, not interim analyses.
Thank you. And we'll just go with online questions unless anyone comes up to the mic. You know, I keep entreating you guys, take advantage of this opportunity. I know it feels kind of, you know, after lunch, but take advantage of the opportunity, please. Okay, online?
Could you please provide insights into FDA's current initiatives regarding the development of new guidance or the update of existing guidance to enhance clarity on trial transparency requirements, particularly within the framework of master protocols where sub-studies initiate and conclude at various time points?
Interesting question. So I hadn't really thought about the master protocol angle. I know that there are efforts to update guidance that are currently available, and you know, we're trying to expand outreach. It gets a little tricky with this program because whereas everything else that under our purview is really in 21 CFR, but ClinicalTrials.gov, of course, is in 42 CFR, and we work together with NIH, and so we sort of share responsibilities, and it can make some of these things kind of complicated. I always tell people when they have specific questions regarding applicability, really, because NIH manages the website and that QC portion, I think the resources that are available, which we vet, you know, which we share and we collaborate on those resources, that the information available on that site is really an excellent starting point. But we are working to sort of update our internal information and guidance as well over time.
Let's take one more question, and that will be the last.
Once a study is entered in ClinicalTrials.gov, can it be taken down? For example, if an initial IRB approval indicated it was a clinical trial, so it was entered, but then the IRB reconsidered and declared it not a clinical trial.
So I believe it would be withdrawn. I'm not 100% on that. It's probably an FAQ on the website, but I think that that is how it would be handled.
Okay, thank you. Thank you very much, Dr. Maldani. Okay, right now we're going to be taking a break, 20 minutes. We'll be back at 2:45 and come back, please. Don't escape anywhere. Come back. Thank you. Okay, my clock says 2:45. So nobody paid attention to me because not everybody else is back here. They probably went outside in the sunshine. Is that? I see these bright lights and I think it's sunshine. Well, maybe they went to dance in the rain or something. All right, so let's start so we could be on time. Our next talk will discuss diversity in clinical trials, drug trials snapshot perspective. And the presentation will highlight the importance of FDA's effort to make demographic data available and transparent with the drug trials snapshot. Our speaker will be Aidan Asifa, Drug Trials Snapshot Lead with the Office of Drug Evaluation Sciences in the Office of New Drugs at CDER. Please welcome Aidan Asifa.
Hello. Okay, great. Thank you, Brenda, for that welcoming introduction. Oh, yeah, thank you. I do need these. So as Brenda mentioned, my name is Aidan Asifa, and I am the Drug Trial Snapshots Lead. And for today's talk, I will be talking about diversity in clinical trials from the Drug Trial Snapshots perspective. Okay. So for today, we'll go over the importance to increase diversity in clinical studies, FDA's initiative to increase diversity in drug development program, and an overview of the Drug Trial Snapshots program, as well as the most recent findings from the 2023 annual report. So why is it important to increase diversity in clinical studies? Adequate representations of patients with disease or condition of interest from diverse U.S. populations may help to generate data reflective of the diverse U.S. populations in drug, biologic, and device development programs. Potentially identify differences in safety efficacy outcomes. Incorporate diverse patient voices and experiences of various parts of the U.S. Build trust among patients. Provide a more comprehensive studying about the disease or condition, and improve health equity and access to clinical research and innovative treatments. So first, I want to go over some statutory requirements to increase diversity in drug development. We'll start with the 1998 demographic rule, where FDA issued a final rule requiring presentation of effectiveness and safety data by age, gender, race, and when appropriate subgroup from the intended population, such as renal failure, for marketing applications. And in 2012, the FDA Safety and Innovation Act, FDASIA, directed FDA to investigate how well the demographic subgroups by sex, race, age, and ethnicity were included in clinical trials, and approve labeling for medical product applications to improve the availability of data to the public. And then in 2014, FDA formulated a 5-year action plan designated to boost the clinical trial participation by women, minorities, and older adults. And this 5-year action plan required FDA to work with stakeholders and standardize subgroup categories to improve the quality of subgroup analysis in new drug applications, which led to the implementation of the Drug Trial Snapshot program in 2015. And then finally, the 2022 Clinical Trial Diversity and Modernization under the Food and Drug Omnibus Reform Act was most recently with the most recent implementation of the diversity action plans. And then I'm just going to go over some FDA guidance documents that are addressing the diversity inclusion in the clinical studies. So collection of race and ethnicity data in clinical trials, that guidance was issued on October 2016. The evaluation of reporting of age, race, and ethnicity specific data in medical device clinical studies issued September 2017. The evaluation of sex-specific data in medical device clinical studies, guidance for the industry and FDA staff, which was issued August 2014. And in 2016, FDA issued new recommendations to industry to the collection of race, ethnicity, in clinical trials. And with respect to women and sex-specific considerations, the post-approval pregnancy safety studies issued on May 2019, as well as the clinical lactation studies, and pregnant women scientific ethical considerations for inclusion in clinical trials was issued on April 2018. And for age group considerations, inclusion of older adults in cancer trials issued on March 2022, E7 studies in support of special populations, the geriatric population in 1994, and ethical considerations of clinical investigations of medical products involving children on September 2022. And then finally, enhancing clinical trial diversity, the enhancing the diversity clinical trial populations, the eligibility criteria, enrollment practices, and trial designs issued on November 2020, and then most recently, the draft diversity plans to improve enrollment of participation from underrepresented racial and ethnic populations in clinical trials that was most recently issued on April 2022. So for the remainder of the talk, I'm just going to be focusing on the drug trial snapshot program. I am not an expert to any of the therapeutic areas that I will be discussing in my talk today. So in 2012, FDA Safety and Innovation Act FDASIA 907 was enacted to require FDA to report on diversity of participants in clinical trials and the extent to which safety and effectiveness data are based on demographic factors such as sex, race, age, and ethnicity. The Drug Trial Snapshots program was implemented in 2015 in part of FDA's response to the action plan Section 907 of FDASIA with 3 overarching priorities: to improve the completeness and quality of demographic subgroup data collection, reporting, and analysis, to identify barriers to subgroup enrollment in clinical trials, and to employ strategies and to encourage greater participation, and an overall effort to make demographic subgroup data more available and transparent. So Drug Trial Snapshots provides consumers and healthcare professionals with information about who participated in key clinical trials that support the FDA approval of the new drugs. They are only published for approved new molecular entities, NMEs, and original biologics, BLAs, which are medications that are made of new molecular structures that have not been approved by the FDA before. And demographic composition and the resulting analysis by the age, gender, race, and ethnicity in pivotal key clinical studies for FDA-approved medical products would be made available on the FDA website. And the race categories are defined from the OMB standards for maintaining, collecting, and presenting federal data on race and ethnicity. The information also highlights the trials, where the trials were conducted, and whether there were any differences in benefits or side effects amongst the different demographic groups. So this is just an example of what's actually in the Drug Trial Snapshots database. As you can see, the brand name or the drug name is on the left, and there are hundreds of drug trial snapshots, individual drug trial snapshot summaries in this database. And I have also provided the link below. So this is an outline of what's actually in a drug trial snapshot. Consumer information is presented first. How is the drug used what is the drug used for and how is it used? Followed by the information about the clinical trials, who participated in the clinical trials, including the number of participants, number of sites and countries, followed by the trial design, baseline demographic data, which includes sex, race, age, and ethnicity. And this is obtained from, and this is usually presented in table and pie chart format. And then the benefit and side effects of the drug, which is from the FDA review. And then lastly, participation in clinical trials with trials report, which report information on clinical trial participants by site location over the 2-year timeframe. And then the most recent 2023 annual report was recently published on May 14, 2023. So for the most recent 2023 Drug Trial Snapshot Annual Report, CDER approved 55 novel therapies for a broad range of diseases, with more than half approved for rare diseases. And about 44,000 study participants contributed through their participation in these pivotal trials. So I've included a section from the most recent 2023 annual report for the therapeutic area, infectious diseases. And then, as you can see, I was given an example, Beyfortus, the indication to prevent respiratory RSV, lower respiratory tract disease, had about 2,943 participants in the trials. 48% were female, 62.7% were white, 23% Black, 2.3% Asian, 15.9% were of Hispanic ethnicity. And then it looks like for this drug program, they adjusted for age for below 6 months, since it's a pediatric drug. 12.1% of the population was below 6 months, and then 24.5% of the participants were in the US. And then comparable to DefenCath, it looks like 100% of the US participants were in the US. So another table in the 2023 DTS Annual Summary Report, this presents percent participation in trials in the therapeutic area of reproductive, urologic, and the rare metabolic diseases. And as you can see, the drug Elfabrio and Lamzee, they have asterisks near their name, which indicates the rare disease. And this year we had a little bit more than half of the approvals for the treatment for rare orphan diseases. Lamzee, a drug to treat non-central nervous system manifestations of alpha-acidosis, had no participants in the clinical trials within the US, as well as no Black or Asians in the clinical trials with Hispanic information just not available. And the drug Veozah to treat moderate to severe hot flashes caused by menopause had only females in the trials, with 81% white, 17% Black, 1% Asians participating in the trial. And also, we included pie charts that reflect programs enrolling both males and females, excluding sex-specific indications for each therapeutic area, which I'll talk more about in my next slide. So the 2023 Annual Summary Report, the percentage of females participating in these drug programs range from 41 to 67%. For participation by sex, we summarized data for programs enrolling both males and females, excluding sex-specific indications, for example, prostate cancer or postpartum depression, as their inclusion would impact interpretability of equitable participation by sex trials for these diseases that affect both sexes. So I won't go into each of these, but as you can see, there are 4 therapeutic areas, and 3 on this slide enrolled more males and females, with infectious diseases almost equal enrollment. And on this slide, 3 of the therapeutic enrolled more females than males, noting the biggest difference of the therapeutic area was in neurologic and psychiatric disorders, with 67% females and 33% males. So some additional race findings from our report: whites comprise more than 50% of the trial populations enrolled for all programs except for 3. Asians comprise the second largest race category, with 6 drug programs enrolling over 30% Asians. 2 programs enrolled over 25% Blacks, and then 5 drug programs enrolled over 30% Hispanics. And unfortunately, we did not report any data in the report of American Indian or Alaska Native race category. Historically, the numbers have been very low, but under 1 to 2%, or just not available. So all of the pivotal trials supporting each of the novel therapy approvals were conducted at multiple sites, and majority were multinational. Percent of the participants within the US range from 0.4 to 82%, with 5 drug programs conducted entirely in the US: DEBUT, DefenCath, Miebo, Xemvi, and Zezapret. And then 2 drug programs enrolled participants entirely outside the US. So from 2015 to 2020, DTS annual summary reports provided aggregate data on demographics of the study population. And in 2021, we started to provide demographic data of study population by individual drug program. And it's important to remember that no prevalence information of the disease or medical condition targeted by the approved drug was provided as a benchmark. The interpretation of the racial and ethnic composition of study population across the novel therapies should consider the prevalence of the disease within each of these racial and ethnic subgroups. And then in my next slide, my colleagues and I, we actually did compare prevalence data of multiple sclerosis with the last 5 drug programs that were approved, and enrolling a study population in a drug development program that is representative of a patient population in the US requires an understanding of the epidemiology and the demographic characteristics of that disease or medical condition. So in this case, multiple sclerosis, it's a chronic autoimmune inflammatory demyelinating disease of the central nervous system that affects an estimated 744,781 adults in the US and is the leading cause of disability in young adults. And 5 therapies were approved for MS between 2017 and 2023. And then baseline demographics and race ethnicity is displayed on this slide. So as you can see, the prevalence data of US patients with MS by sex, race, age, and ethnicity is presented on the first half of the slide. And we compare that to the baseline demographics of race and ethnicity on the bottom half. So the first row is all sites, second row is US sites, and the third is non-US sites. And so as you can see, the x-axis, you see the racial categories, and each of the bar graphs are the MS drugs that was approved on that year. Oservis was the light blue in 2017, Mazenet 2019, Zeposia in 2020, Ponvory 2021, and Briumvi in 2022. And then we compared it to the prevalence data, and then that red line is actually what you see. That's the prevalence data of MS for that racial category. So what we found across all 5 programs, the percentage of female patients enrolled across all sites, about 60 to 67%, was similar between US and non-US sites, and slightly below the US prevalence at 76%. And in terms of race, enrollment of Asian patients in the MS programs was below US prevalence, below 2%, with the exception of Mazenet at all sites and non-US sites, and 3 programs enrolled no Asian patients in the US. And the enrollment of Black patients at the US sites, around between 6 to 21%, approximated or exceeded US prevalence of MS, whereas the enrollment of non-US sites between 0 to 0.5% was lower than compared to the US prevalence at 10%. Across MS programs, enrollment of white patients at all sites exceeded the US prevalence of this race group. And approximately 7% of the MS patients in the US are Hispanic or Latino. The percentage of Hispanic Latino patients enrolled varied across the programs. And in general, the US sites enrolled a higher percentage of Hispanic patients, by 15%, that approximated the US prevalence. So presenting demographic data by drug therapeutic area in the annual summaries will help track trends in diversity over time. It's important to identify programs that have achieved lower or greater enrollment of underrepresented patient populations. And in conclusion, efforts across many years and by multiple stakeholders have led to improvements to enroll diverse study populations in some therapeutic areas, and opportunities for improvement still need to be identified and addressed. And external engagement and open dialogue about knowledge gaps, underrepresentation, challenges, approaches are important to ensure diversity in drug development programs. And in summary, it's important to generate data reflective of the US population in drug, biologic, and device development programs. Drug Trial Snapshot Program is FDA's overall effort to make demographic subgroup data more available and transparent. Interpretation of racial and ethnic composition of these study populations across the novel therapies approved should consider the prevalence of the disease, and opportunities for improvement still need to be identified and addressed. And in closing, I have a quote. I think it's very, it relates to the Drug Trial Snapshot. The way we work in public health is we make the best recommendations, decisions based on the best available data. Yeah.
Okay, thank you very much, Aiden. So we're going to do, am I out of minutes for questions?
Well,
Okay, we have a good amount of time for questions here. Need I ask, any questions in the house? Okay, we will then continue with.
I had some poll questions.
Someone?
I had some poll questions. I don't
Oh,
know.
okay. Sorry, go ahead.
I didn't know if you're okay. So one of my poll questions was the drug trial snapshots program was in response to which FDA statutory requirement? Is it A, the 1998 demographic rule, B, the 2022 clinical trial diversity and modernization under the Food and Drug Omnibus, the FDORA Act, or the 2012 Section 907 of FDA Safety and Innovation Act, FDASIA, or all the above? All the above, you said? No. Yes, it's D. And then my second poll question is the drug trial snapshots are available for all drugs, devices, and biologics approved by the FDA. True or false? Yes, it's false. It's only for drugs and it's only for novel therapies. Thank you. Okay.
Thank you guys for answering, audibly. It's good to have that type of energy in the room. Oh, we got somebody. Good to have that type of energy in the room. Okay, let's take the question in-house, please.
Thank you. Thank you for your talk. And I'm really glad to see that this is happening. My name is Maria Waracha from UC San Diego. And one of my main focuses there is on diversity and patient outreach. So I've seen and throughout the research, 50% kind of the average for the white to be part of the clinical trials. But is there something with the INDs that CDER is looking for or to encourage or recommend for the outreach? Because, obviously we can't say, oh, it's difficult to get patients of minority background into clinical trials, but just looking to see what else is being done. I mean, it's great to provide the numbers, but what actions are actively being taken to put us on onus to go out and really try and increase that diversity in clinical trials?
Yeah, that's a really good question. So, FDA isn't a regulatory agency. And so, actually, the drug trial snapshot is a really good way to boost those numbers because it is transparent. So, patients and consumers, they see this data. And also the recent legislation that's requiring sponsors to submit diversity action plans, which as a result, that's going to have in turn sponsors submit data that's reflective of the US population in these drug trials. And then we do have like certain offices that do fund certain studies. I think it's the Office of Minority Health and Equity that helps fund studies that are of underserved communities.
Thank you. Next in-house question, please.
Hi, Yang Wang. I'm from EMD Serrano. Thank you so much for this presentation on a very timely and important topic. We all know that the FDA diversity action plans are a big priority for the agency. So one of my questions for you is, do you anticipate using some of the snapshots data to further help support, discussions and feedback surrounding how sponsors are getting, their diversity action plans, whether they're meeting enrollment goals?
Well, as you know, it is separate, a different timeline in the drug development program. However, I do sit in those meetings. And so my experiences in reporting this data, I do bring that experience to, our implementation on what is being required in the diversity action plan. So, it's related in a sense, and it's transparent. And so we try to encourage collaboration with those initiatives.
If I may, just a real quick follow-up to that. One of the things that I really think I have to compliment you guys on is the separation now of US versus global in the summary reports. I think that's very helpful.
Yeah.
And it'll be I think, ongoing topic of conversation as we get more multi-regional clinical trials and more involved in kind of this diversity conversation.
Oh, thank you. That was actually at the request of Dr. Califf, so that was something he wanted us to capture. Thanks.
Thank you.
As a reminder, when you do the survey, please put your comments such as this. If you have negative comments, we'll take that too. But we'll take any positive comments, any suggestions that you would like to see, or anything you thought was exceptionally done well and of great significance to you in your responsibilities. Please put some notes there for us, please, so we can get some idea of the impact that we've had upon the audience. Okay, online question, please.
The snapshot provides an overview of race/ethnicity and other demographic breakdown. However, how do we ensure that this is reflective of the population of interest or reflective of the epidemiology and balance it with clinical trial recruitment timeline and completion of the trial to bring drugs to patients with high unmet need?
Yeah, that's a really good question. So the Drug Trial Snapshots program's purpose is just to report on the data that we're seeing in the clinical trials. So, it's not compared to any prevalence information, but, the data is available to the public. And for what my colleagues and we've done, and anyone that is welcome to do this as well, is compare the enrollment of different racial categories in these trials with the prevalence data of certain medical conditions. For example, what I did earlier with multiple sclerosis.
Thank you. Next question, please.
The demographic data in drug trial snapshots appear to be for the globally enrolled population. Are there plans to share enrollment demographics for just the US population enrolled in a study, or does FDA expect that the population enrolled in the global study should reflect the US treatment population?
Yeah, so I mean, so this year was the first year that we actually did break down the percentage of participation in the US, and that was the reason why. Before we never really captured that, and so before we just did race, the other demographic factors. And so the first step to, finding more about, the outside versus the inside US is starting to report the percentage of US enrollment. Again, that could be done with additional research. However, right now, we're just reporting with participation in the US and the racial and sex and age demographic factors.
Thank you. Next question, please.
Do you have information on the number of diversity plans that have been submitted since the release of the guidance, and what types of programs trigger the FDA to request a diversity plan?
So I actually don't have any information on diversity action plans. Sorry, this is only the Drug Trial Snapshots program. I knew I was going to get that question.
Okay, next online question, please.
What are some of the biggest barriers that explain such underrepresentation of minorities?
Well, I think it's very complicated, and there's many. It could be location, it could be health equity, it could be standard of care, and it could be lack of trust. I mean, there's so many issues of lack of, and then also, one thing I've noticed when I was doing this is that a lot of the drug programs, they kind of follow each other from the previous years. For example, with MS, they tend to do this certain the same amount of enrollment outside the U.S. versus inside the U.S. And so when they do that, they're not really changing the diversity of their clinical trial. So I think a lot of it is just strategies from these companies on enrolling diverse patients.
Okay, do we have any more online?
Yes, we can do another online. With AI integration, will it help issues like this? How would it help break any barriers with diversity?
I'm sure it will. That is something that we haven't had talks about yet. I'm sure industry has, but I'm pretty sure that AI, especially with the communication efforts of enrollment of diverse patients, will definitely help.
Let's take two more online, please.
Diversity plans are currently recommended as per the guidance. Is it anticipated that inclusion of these plans will eventually be required with INDs and BLAs, et cetera?
Yeah, so I'm sorry I can't answer anything related to the diversity action plans. Out of my
so-
scope.
All right, so let's take two more online questions, please.
Okay. If mixed ethnicity, white/non-Hispanic, and white/Hispanic parentage, is there data for that inclusion? Is ethnicity self-reported? Sorry, that's the first part of the question.
Oh, I'm sorry, I can't hear. What did
Sorry,
you-
Can you repeat,
first
please?
part of the question is ethnicity self-reported, aka if mixed ethnicity, is there data for inclusion?
Yes, it's self-reported. And so that's also challenging with other countries that don't recognize race. So that could also be a reason why you see race categories that are not available.
Last question, please.
Recently revised Statistical Policy Directive number 15 now recommends using one combined question for race and ethnicity and encourages respondents to select as many options as apply to how they identify. How will these revisions impact FDA's expectations for collection and reporting of race and ethnicity data in clinical trials, subsequently drug trial snapshots data?
Yeah, so eventually that's going to make the drug trial snapshots reporting of race and ethnicity a little bit more expansive, which is good. However, we don't have any like near future plans on reporting these new categories, race categories, or race and ethnic standards. However, I'm sure that's something that will be in the future.
Okay, thank you very much. Thank you.
Thank you.
Great job. Okay, our last presentation for today, and let's keep up that momentum for the presentation so we can get Dr. Lauritsen here all excited to talk about combination products. She's going to talk about combination products, updates, and best practices. Combination products offer opportunities for development of innovative diagnostic and therapeutic products. This session will include a brief overview of combination product regulation, provide updates and recent guidance, and share best practices for submissions of FDA to FDA and engaging with CDER. Our presenter today is Dr. Christina Lauritsen. She is a combination product regulatory the combination product regulatory advisor from the product jurisdiction and combo product team within CDER's Office of Executive Programs. Let's welcome Dr. Lauritsen.
Be your last presentation of the day. Hopefully it will go smoothly. I do have the pleasure of speaking to you today about combination products. And, kind of before I dive into that, I just want to, let you think for a minute about what we used to do back before Google existed and we had a question where you went to the library, right? And so anytime we wanted to find out more information or get some ideas about how to proceed with a specific idea that we did have, we'd go in and research that in the library and we'd check out some books, get some ideas. So I'm going to kind of hopefully open a little bit of a book for you today on what combination products is, give you a little bit of lessons and some advice on how-tos on combination products. So I'm going to start by providing an overview of how FDA reviews combination products, then move on to giving you some updates and ending finally with best practices for submissions and engagement. So in terms of an overview, we need to talk about what the combination products are. So the combination product definition, center assignment, and then pre-market review of combination products. So what is a combination product? It is composed of two or more different types of medical products. So it has to be two different things, drug and a device, drug and a biologic, device and a biologic, or even all three combined together. And when you have a combination product with the two different types of medical products in it, each component is called a constituent part. So we talk about constituent parts of combination products being the drug constituent and the device constituent if you have a drug device combo. Now, there's 3 different ways you can make a combination product. You can be chemically or physically combined into what we call a single entity, co-packaged or a kit, or sold separately but labeled for use together. So let's dig in a little bit. Single entity combination products, again, chemically or physically combined. So things like an EpiPen, a pre-filled auto-injector with epinephrine, right? Antibody drug conjugates is a drug biologic example, one of the more common ones. Inhalers, IV bags pre-filled with the drug, or transdermal patches for delivery of drug. These are all considered single entity combination products. Co-packaged combination products are, pretty much exactly what they sound like. They're one of the easy ones to tell, where you have both a device and the drug, and they're just provided together in the same package. It can be something as simple as Tylenol with the oral dosage delivery syringe in it, or they can have, even more complex combinations where you have pre-filled syringes, plus additional devices, plus reconstitution devices and transfer things and fillable cartridges. And all of these things could be all combined together in one package. So that's a combination product. And then the rarer type of combination product, we don't see a lot of these, is the cross-labeled combination product. And this is when the constituent parts are sold separately. They're packaged and distributed separately, but they are labeled for use solely with each other. And one of the more common examples is a reusable light source and a one-time use photoactivated drug. And so in these cases, there's specificity in the label about using these 2 components only with each other. This is a little bit different than just recommending a general use device for use with the drug. That does not make a combination product. So if a combination product has 2 different constituents, how do you figure out where in FDA you're going to work on developing that product? Luckily, it's not a choose your own adventure book. We do have some regulations that tell us how you figure that out. And so what we do is we look at primary mode of action for the combination product. And that's defined as the single mode of action that provides the most important therapeutic action for the product. And then we assign based on which constituent part provides that primary mode of action. If it's a drug, the combination goes to CDER. If it's a device, it goes to CDRH. If it's a biologic, it goes to CBER or CDER, depending on the type of biological product it is, because some biological products like monoclonal antibodies are regulated by CDER and not CBER. And then there are times when it's a little bit difficult to tell which constituent part provides the primary mode of action. In that case, we use something called the algorithm and determine assignment based on first consistency. Do we have a product like this already that raises similar questions of safety and effectiveness? If yes, we will assign that similar product to the same center for consistency. If that doesn't exist, if it's really novel, we'll look at it and we'll try to determine where the most significant safety and effectiveness questions lie and which center has the most expertise related to those. And that can be really challenging to tell too, especially if both the device and the drug raise really novel safety and effectiveness questions. So it requires a lot of collaboration and discussion among the centers in those cases. So pre-market review of combination products. It's important to recognize that we assess the safety and effectiveness of the combination product as a whole. We'll look at the individual constituent parts and we'll look at them, how they're used together and how they're put together as a whole. When you're assigned to that lead center based on primary mode of action, that lead center is the primary point of contact for everything related to the combination product. And the combination product will follow that lead center's processes, procedures, applications, et cetera. So for combination products assigned to CDER, you're going to use the IND process, the NDA, the BLA, or the ANDA for your application type, just depending on what's most appropriate for the combination product. If you're in CDRH, you're going to use all of their application types. CBER is a little bit of a special snowflake here because they have both the device application types and biologic applications and drug applications. And so it's really going to depend specifically on what the product is in CBER and the type of application that's appropriate. And then the other thing is that once we have an application or a product assigned to a center, that doesn't mean that you are solely working with that center. The lead center will engage all of the other centers as appropriate to get their expertise so that we're bringing all the expertise to bear on the combination product as a whole. All right, so I'm going to move on to some updates. unfortunately, I don't think these updates are necessarily quite as exciting as finding a new book on the shelf at the bookstore, but we do get pretty happy when we finally finish hard work on something and get that out in the public eye. So starting with the guidance on regulatory considerations for prescription drug use software. This is something we affectionately call PDURS, which stands for Prescription Drug Use Related Software. And what it is software that is disseminated by or on behalf of a drug sponsor and produces end user output that supplements, explains, or is otherwise textually related to one or more of the sponsor's drug products, regardless of whether the software is a device. I know that's a mouthful, and we'll talk it through a little bit. So the draft guidance essentially describes how we're going to apply our labeling authority to the outputs of certain types of software. And then it also describes what we're going to do to determine whether some of that output gets described in the prescribing information or whether it's simply considered promotional labeling. If it's going to be in the prescribing information, how are we going to describe it? What are we going to do? And then how you should submit information about that end user output to the agency. So importantly, the guidance isn't changing our definitions of required or promotional labeling. We're just applying those definitions to the output of software. So required drug labeling is the labeling drafted by the manufacturer that's reviewed and approved by FDA, and it includes the information essential for a provider to make an informed decision about the risks and benefits of prescribing the drug. And for the patient to successfully and safely and effectively use the drug. Promotional labeling is generally any other labeling other than FDA required labeling that's devised for promotion of the product. And that can include printed, audio, or visual matter that's descriptive of the drug that is disseminated by or on behalf of the drug's manufacturer, packer, or distributor. And so that's important key as well. So just a few highlights from the draft guidance. Whether or not something is a PDURS, whether or not something, the output of a certain software is PDURS is independent of device status. So I don't have a lot of time to go into all of the intricacies of when FDA does and doesn't regulate software. There's a lot of guidances out of CDER, but just recognize that there are some software functions that are device functions that FDA regulates. There are some software functions that are not device functions, and we don't regulate those. And so the output of the software, depending on whether it's a device function or not a device function, could still be PDURS. So it's regardless of device status. When we're trying to decide if that information gets put into required labeling or promotional labeling, we're going to consider whether the function is essential to the safe and effective use of the product, whether there's an evidence of clinical benefit from use of that software, and whether it relies on data transferred directly from a device constituent of a combination product. The guidance also explains when and how to submit that PDURS information to FDA. And again, this clarifies it does not apply to software developers that are not affiliated with the drug sponsor. So third party, this doesn't apply to them. Now, as I said, the output could be PDURS, but the software may meet the definition of a device. So you have both cases here. In those cases, you may need to submit a device application for something that is also a PDURS. So what that means is that when CDRH is reviewing that device application, they're also going to consider questions related to the representations about the drug, and they're going to consult with CDER or CBER as appropriate for that. That means that if the output of that device is PDURS promotional labeling, then you would not necessarily need to submit a Form 2253. This is what we have in the draft guidance right now. But if you make changes to that PDURS and you don't need a new 510K because the changes didn't require a new 510K to CDRH, then you would submit that information on the Form 2253. And regardless, the 2253 is not a replacement for the device marketing submission and vice versa. So 2 examples here. The first on the left is a medication tracker. So it is tracking when injections are given. This is not a CDRH regulated device, but it is PDURS when the promotional labeling is in there by or on behalf of the sponsor. If it's displaying the name of the drug and may be considered part of an integrated system. So the 2253 might be appropriate in this case. On the right side is a dose calculator. The dose calculator is a device, and it might need a 510K. If it is also marketed by or on behalf of the drug sponsor, then it would also be considered PDR's promotional labeling and would need to follow those requirements as well. Again, if it's not marketed on by or on behalf, it's not PDR's. All right, moving on to another hot topic. I'm not going to spend any time on this really because you had a wonderful talk full of AI. I just want to point out that, again, combination products are in the AI space as well. And so this publication was published and it covers, these 4 different areas of focus about how we are working on AI. And so as you heard from Tala, combination products is right in there as well. The next guidance that I want to talk about is the compliance policy for certain ophthalmic products and part 4. Now, to talk about this guidance, I first have to tell you about a court decision called what we call the Genus decision. In brief, several years ago, a case went to court about whether barium sulfate contrast imaging agents were devices or drugs. And the court decided that FDA does not have the discretion to regulate something that meets the definition of a device as a drug. So if something meets the device definition, it must be regulated as a device. And their decision was broader than just that of the contrast agent at issue in there. Fortunately, Congress later clarified that in Section 503 that any contrast agent, radioactive drug, or OTC monograph drug is deemed a drug and not a device. So that removed some of that regulatory uncertainty with those products that resulted as a result of the court case decision. So what does this have to do with ophthalmic products? Well, there was this odd exception in the regulation talking about devices packaged with drugs. Now, you heard me say devices packaged with drugs, chemically combined or in a co-package or combination products, right? 200.50C had some language that didn't jive with what I told you earlier. And it said specifically, eye cups, eye droppers, and other dispensers intended for ophthalmic use are regulated as drugs if packaged with the drugs with which they are to be used. So that was this weird thing happening there, right? Combination products, if it's an eardropper, not a combination product if it's an eyedropper. Genus removed that regulatory like uncertainty, or not uncertainty there, but that regulatory exception. And so we now are regulating those eye cups, eye droppers, and other dispensers as devices, meaning that when they're combined with the drug, they are drug device combination products. So in doing that, that meant that manufacturers had to change what they were thinking about with these and stop thinking about them as drugs, start thinking about them as combination products, and apply the combination product regulatory scheme to that, including 21 CFR Part 4, which provides for CGMP requirements and post-market safety reporting requirements for combination product. So the guidance explains our approach to calling them combination products now, and also provided a 12-month grace period where we were not applying any action with respect to those because they were not previously regulated as combination products. That period actually ended a year ago. The other important thing about that guidance that is still in effect is that it's explained that for lower risk device constituent parts, like those really simple drop-tainers and ampules that administer the drug directly to the eye, FDA is actually still evaluating how the 820 requirements for quality systems are going to apply to those device constituent parts. And so we don't intend to take any action with respect to non-compliance with the 820 requirements for those types of combination products. Instead, we're going to continue thinking about it, figure out what we're going to do, publish some current thinking when we get there, and then provide some timing expectations to come into compliance depending on what the outcome is. Now, really helpfully for that, CDER held a panel meeting about ophthalmic devices in November of 2022 to talk about and make recommendations on the classification of ophthalmic dispensers under the CDER device classification scheme, which is class 1, 2, or 3 based on the risk of the product. And so they talked about classification of these. There was a recommendation at the end that they be class 1 exempt devices, but not GMP exempt. So as CDER moves forward with their thinking on how to move on from that panel meeting and what to do next, some of the outcomes of that will certainly inform our thinking about what Part 4 requirements are going to apply when these devices are parts of combination products. Next up is a guidance on the application of human factors engineering principles for combination products. This guidance is the final guidance. We're very excited this went out. It is not replacing either the CDRH or the CDER human factors guidance that exists for standalone drugs or devices. It's in a question and answer format, so we find that to be a little bit helpful. And what it does is clarify how the unique aspects of a combination product influence the considerations with respect to the HIV process for these products. So, importantly, the user interface for a combination product includes all points of interaction between the combination product and the user, including displays, controls, packaging, product labels, carton labeling, instructions for use, and even training if applicable. And so when looking at a combination product, we want you to be considering the use-related risks associated with the combination product as a whole, especially those that do not exist for the device alone or the drug alone, so where that convergence is of the combination product. And then apply that in looking at the what is needed from a human factors perspective. So the Q&A guidance includes items such as definitions of what is a final finished combination product. Well, that's the to be marketed product. It's best to do your human factors validation studies on that to be marketed product. So we talk about the timing necessary for that. Talks about how to identify what is a combination product critical task. The difference between a use-related risk analysis and other risk analyses. So, you know, a URA is essentially a subset of the overall engineering risk analysis that is done for a device. So you can get more information about our thinking on that in the guidance as well. And finally, the last guidance update I want to give is about controlled correspondence related to generic drug development. And this is a helpful guidance for developers of generic combination products because it provides information about the process that they can get for getting information from FDA about their drug development plan. And that's through submitting something called a controlled correspondence. So the guidance provides details and recommendations about what inquiries are appropriate to come in as a controlled correspondence. And helpfully for combination product developers, that includes questions related to the evaluation of the user interface of a drug device combination product. And so questions about combination products may be appropriate in a controlled correspondence. The guidance will go into more details about that, as well as explaining when it's appropriate to submit them in a single controlled versus two separate controlled correspondence, depending on what the questions are and whether they're related to separate elements of the generic drug development plan. Okay, and so that takes me to the third section of my talk today. Like I said, I'm going to give you a little bit of a how-to and then point you to some other documents that you can use for a little bit of self-help. So first thing, before you engage with FDA on any product development, you want to be sure that you know if your product is a combination product or not, and you want to know where the product belongs, which center it goes to. If that's ever not clear, you have a couple of different options. One of them is to take the pre-RFD or RFD route. That's called a request for designation. I'm sorry, I guess it's spelled out in the guidance at the bottom. A pre-RFD as a pre-request for designation or an RFD. And that is done through the Office of Combination Products. If you're not ready to put together a full pre-RFD or RFD document, you can always engage with one of the center product jurisdiction officers, one of them, for confirmation on where the product belongs. And it's also important, you do not need a pre-RFD or an RFD. It's only when there's uncertainty. Don't send us an RFD for a pre-filled syringe. You're going to CDER, okay? The pre-RFD, the difference between that and the RFD is the pre-RFD is not binding. It offers a more informal decision, although we do write down all of the reasons why we're giving you the decision we're giving you. There is a little bit more of an interactive process with that because there is not a firm deadline. There is a goal of getting responses back within 60 days, so we do strive for that. But because it can be a longer document, or we might do some more interactions, it could exceed that amount of time. An RFD, on the other hand, is a binding decision. It's best used when the product is already pretty well characterized and configured, like you know you're going to put it in a co-package. That way we know you're going to be a combination co-package. But if you're like, well, I don't know which device I'm going to use yet, and I don't know what I'm going to do with it, and that might be a little too early to tell if you're a combination product or not. It is not interactive. After the RFD is filed, we only have 60 days to issue that letter. That 60 days includes a lot of review time by the Office of Combination Products, review time by the center product jurisdiction officers, review time by center subject matter experts when appropriate. And so there is not a lot of time there. 60 days sounds long, but it really isn't. And the requirements for that are in our regulations, and there's guidance documents available on both of those. Okay, once you do know which center you're going to work through, and this goes back to what I said on one of the earlier slides, you're going to work through the center-based mechanism of the lead center to get feedback on the combination product. So if you're in CDER, you're using their Q submission program or their Q sub program, and they have several different ways that you submit through that and different types of Q subs depending on the nature of the inquiry. If you're in CDER, you're going to follow our formal meeting guidance depending on the product that you're developing. So if it's a PDUFA product, you follow that guidance. If you're a biosimilar, you're following the BsUFA guidance. If you're generic, there's guidances based on meetings on complex products or meetings on product-specific guidances. And then there's this overarching guidance document that really points to all of these other guidance documents that already exist, talking about more explicitly requesting feedback on combination products and what is helpful in there. And so some of the best practices identified in that guidance include, again, submit all your meeting requests to the lead center, even if all of the questions are about the non-lead center constituent part. Why? Well, you only have questions about this really novel device, but it's going to be pre-filled with the drug, and we want to keep all of that information in the same file, and we don't want to be trying to find information in one file in one center, information in another file in another center, and trying to put the pieces together when ultimately it's going to be a single entity combination product. Again, all communications should be through the assigned point of contact. That way we can keep that administrative record very clear and have that information so we can go back and look at it. If 5 years later you say, hey, this happened, I talked to so-and-so, and we don't have that in the record because you didn't go through the primary point of contact. Also, clearly identified as a combination product. You would think that's obvious, and there is a check mark in the 356H or the 1571 forms for you to tell us it's a combination product, but don't just do it there. Put it in the cover letter or put it in the summary of what the product is. Tell us up front it's a combination product. There's nothing worse than looking through a pre-IND meeting request and finding out on page 150 that you're planning to do this device, because we've just spent a lot of time not consulting CDER because we didn't know until we got to page 150 that you were planning this device. Again, this is obvious for pretty much any area of product development, but ask specific questions related to the appropriate stage of product development. A lot of times we are looking at sponsors who want to start bridging from a prefilled syringe to an auto-injector or something when they're changing their device midstream. So ask us those questions. Let us know that you're planning to do that. And then as you get to the point where you're starting to get ready to do that, then ask us the more specific questions about the appropriate way to do that. Provide sufficient information for FDA to consider. You know, if you're just saying, well, I'm going to use this with this device, this auto-injector, but you don't describe the auto-injector, you don't explain anything about it, it's really hard for us to then send that to CDER and say, hey, there's an auto-injector here, can you give us feedback? And they're going to say, no, there's no information. They should, you know, do device stuff with the auto-injector, right? So provide us enough information so we can give you constructive feedback. It's also really helpful if you are to have multiple development programs going on and you're using the same device across multiple programs, let us know that. So if you're submitting a pre-IND meeting request because you've got a pre-filled syringe for a drug, keep using pre-filled syringes, but those are the easy ones, pre-filled syringe for a drug, and you're like in the anesthesia division, and then you've got the same device and you're using that in the oncology division. Let us know that so that we can leverage the information from the relative files, make sure that we're being consistent in the information we're giving you. It's very helpful if we know that that same device is being used elsewhere, because we can look back at some of that information that we already have on file. Also, A lot of devices might be 510K cleared or PMA approved before they get added to a combination product. And so if we have that information, that can be helpful as well, especially if it's in a clinical trial or an IND setting. If you're telling us you're going to use a specific device for delivery of your drug, that you may not be developing a combination product necessarily, but if you're using a specialized catheter that's only for delivery to one part of the body and it's only cleared for that, but now you're trying to use it for delivery to another part of the body, we might need to take another look at that device. But having the 510K number, the clearance number, really helps us confirm that the device is appropriate for the use that it's being used for in that study, or if we need additional information about the combined use of the drug and device. So FDA promises to identify a primary point of contact. You get that every time you reach out. Provide timely communication. We have goals under all of the user fee programs. We work very hard to meet all of those goals. We will engage the relevant expertise from the other centers when needed. And again, that's why it's really helpful if you tell us up front you have a combination product. We'll provide comprehensive feedback as best we can based on the current thinking at the time and the information that you provided. And you can request certain staff and be included in those meetings. It's especially helpful if, in addition to saying, hey, I have a combination product, say, hey, I think CDRH might want to be at this meeting too. Like, tell us that. That does help us. And you can also request that the Office of Combination Products or the center's PJO staff attend those meetings as well. And we can provide helpful assistance in those. Okay, so up to the challenge questions. You are developing a novel implantable device that is pre-filled with the drug, and that drug was approved for the same indication years ago and is a well-understood drug. Which center, I went too fast. Which center is responsible for the combination product? Well, I hope you were going to say CDER, because that's the way where it goes. I didn't even think I hit a button. Oh, wait, it's moving by itself. Why is it doing that? I didn't move it. I wasn't even touching it. Anyway, the device is pre-filled with the drug. It is a novel device. We've never seen it before. Drug is well understood. It's still a pre-filled drug device combination product. It still goes to CDER. Challenge question number 2. If you have an approved combination product in CDER, for example, an NDA, who would you ask about the type of supplement needed if you're making a change to the device constituent of the combination product? Would you ask the Office of Combination Products? Would you ask CDER? Oh, again, I didn't touch it. I didn't know I had a timer on these. Anyway, you ask CDER. Don't ask CDRH. CDRH doesn't know anything about NDA supplements. CDER will decide what is the appropriate type of what is the appropriate type of NDA supplement that would be needed. They might consult with CDRH because they want to make sure that the supplement type is commensurate with the type of review required. So if you're making a pretty significant device change, a CBE 0 is probably not appropriate. Maybe it's a prior approval supplement. Maybe it's a CBE 30. It really just is going to depend on what that change is. Okay, now move without me. If you have questions, when you're in the library, you ask the librarian. When you're at FDA or needing to work with FDA, you ask us. You can talk to me. My address is up here. All of the center product jurisdiction officers have their own address. It's really easy to remember. It's CDER, CBER, or CDRH, product jurisdiction at FDA.gov, and that will get you to the product jurisdiction officers. I promise you, we are in those mailboxes multiple times a day. So your email is not going unseen. It's not going into a black hole. If you email those. And the same with the Office of Combination Products, combination at FDA.gov. That is not a typo. They don't have the HHS in their name. I don't know why. And thank you. I'm happy to answer any questions you might have. I guess.
Hi, thank you for the presentation. A question related to guidance documents. The Combo Product Summit last fall, the FDA mentioned there was work being done towards a guidance that would include something about essential performance.
Essential performance requirements, yes.
An update?
Yes, I am very excited that that will be coming out this year. Yeah, I can't say when, but it is. That's to say it's imminently by the end of the fiscal year, so not even the end of the calendar year, you should see it. Probably well in time to talk about at the Combo Summit. No promises.
Can you share with us where to find the latest FDA guidance on combination products that incorporate nanomaterials? For example, an IV administered drug comprising of a nanoparticle carrier plus a covalently linked therapeutic agent. In this case, is the nanoparticle a drug or a device?
Excellent question. And it's going to depend, it's going to be very product specific on what that nanoparticle is, whether it has additional functions or whether it's, really just part of the carrier the same way, saline is a diluent. So that would have to be something we'd look at on a product specific basis. And all the guidance can be found just searching FDA guidance. I don't think there's one specific to that question.
I'm expected to do a performance now. Speaking about guidances, we have thank you, Jeff. We have quite a slew of guidances out there on our exhibit table. Those were pertinent to presentations from today's schedule. So if anyone catches your eye, please feel free to take them home with you and give them a good home. Okay, next question online, please.
What is the threshold for determining if Peters meets software as a medical device requirements? Should industry refer to guidance on mobile medical applications, device software functions, and clinical decision support software?
Yes, all of the above. So as I said, PDERS is separate from device status. So if you want to know if something is a device and it's software, you are going to refer to all of those CDER guidance documents, which will tell you which ones are devices that are subject to enforcement discretion, meaning we don't need to see an application for them, and which ones require an application. And then depending on if you are buyer on behalf of the drug sponsor, the output of that is PDERS, or yes, if you're on buyer, is PDERS, regardless of whether you meet one of the device definitions.
Next question, please.
If an investigational assay is being used for patient selection in a drug clinical trial, what kind of regulation and processes does the sponsor need to follow, and how to get more advice?
So I would recommend in that case, so that's not necessarily a combination product, but it is a co-development, sounds like a co-development of an in vitro diagnostic and a drug. And questions about that can be directed to CDER as part of a pre-IND meeting request, or to CDER as just to ask questions about the assay itself. Depending on what it is, you may need to have both an IND and an IDE in CDER and CDER for a drug and a diagnostic.
Next online question, please.
If leveraging an approved drug device combination product for a new indication, so no change in dose or route of administration, how should a sponsor approach risk in terms of human factor studies?
That's an excellent question. And so again, it's a new indication, but same drug and device, correct? That was the question?
Yes.
Yes, new indication.
New indication, no change in dose or route of administration.
Yep. Yeah, so that is one of the things that the human factors guidance talks about. I think it gives an example of a case like that, but you are considering, when you're looking at use-related risk, you're considering not just the drug and the device and the dose and route of administration, but also the patient population. And so if you, and the context of use, so whether it's an emergency use versus a non-emergency use is going to matter. So even if the product is the same, if you're changing from emergency to non-emergency, you're going to have some differences in critical tasks that might be there. So you need to go through that use-related risk analysis.
Next online question, please.
Does the FDA plan to update post-approval guidance for drugs/biologics to include device constituent changes, such as auto-injector or pre-filled syringes?
I do not know the answer to that question. I know that we have been working on a guidance about post-approval changes for combination products specifically, but that has been in the works for many years and hasn't made a lot of progress.
Let's take 2 more, please. Online questions.
Who do we talk to about bridging approach for the auto-injector and used across multiple different drug molecules as the drug sponsor?
Yeah, so that's one of the challenging questions, and it's a really good idea to where you are in your development program, wherever you're first going to attempt that bridge, to include information telling us that you're doing this in other development programs and what your plan is, and we may be able to provide you advice that across a little bit more of that in just one. It's also a good time to just reach out to the Office of Combination Products and talk in some generalities about the bridging. That is definitely something that we have a lot of questions about. I will just also mention that there is a draft bridging guidance out there, and we are working through the comments that were received and working towards hopefully putting that guidance out in final at some point.
Last question, please.
If there will be an FDA inspection on a combination product managed by Office of Combination Projects, which agency will complete the FDA inspection?
Right. So if it's a combination product, the lead center will work with the non-lead center constituent to decide what type of inspection needs to be had. And they're going to look at the information that's submitted in the application about the manufacturing facilities, the type of facility it is, and determine what inspection type needs to go out there. And then there is a guide for inspectors for doing those combination product inspections.
Okay, that's our last question. Thank you. Thank you very much, Dr. Lauritsen.
Thank you.
Well, that brings us to the end of today's schedule. Time for a quiz, anyone? Okay. Tell me, somebody, what was the most applicable lesson or point that you learned today that you're going to take back home to your office? First person gets a prize. Nobody wants a prize. Or did nobody learn anything? Come on, tell me something. All right. Oh, yes, ma'am. Excellent. Thank you very much. Commander Lim, can you give this attendee a prize, please? Okay, so this brings us to the end of today's schedule. A couple of things for the on-site attendees. Make sure you get your parking stickers before you leave, so you get a discounted rate of $10. What else? Online people, thank you very much for staying with us. I don't even have a number of online, but I know it's up in the thousands when we consider the online and, everyone else through the various mechanisms online. Unfortunately, our scheduling, the speaker's scheduling had conflicts, so we are not able to offer that one-on-one session we were hoping to offer this afternoon. So we will be adjourning early, but that means you can go down to the networking in the lobby area a little bit early and get a head start over the other 2 centers. And tomorrow we'll see you bright and early at 8:30 a.m. Thank you very much.